Dr. Sneha R. Ambwani is the Professor and Head, Department of Pharmacology, All India Institute of Medical Sciences, Jodhpur, Rajasthan. She has a teaching experience of more than 27 years and has published an MCQ-based book titled Review of Pharmacology. She has 80 publications in various national and international journals and has been awarded the Certificate of Appreciation from Dr. S.N. Medical College, Jodhpur for Best Performance and Dedication as a faculty member in the year 2013. She is also the chairperson for various committees including Drug Trial Committee, Animal Ethics Committee and Biomedical Waste Management Committee at AIIMS, Jodhpur. We welcome you, Madam.
We also have with us Dr. Punna Gai, Professor and Head, Department of Pharmacology, SRMC, Chennai. She is a member of Government of Tamil Nadu Etymological Committee, member of Board of Studies and the Curriculum Committee of MBBS and MD Pharmacology at SRMC. She is the member of Ethics Committee in SRI HER and Aravind Eye Hospital. She has been invited as speaker in many GCP workshops and national conferences. She has organized the Rapid Review course Aspire since 2019 for a period of 4 years and also various clinical pharmacology and medication safety conferences. She has published more than 40 original research publications in indexed journals, along with being editorial member of National Journal of Basic Medical Sciences. We are happy to have you, ma'am.
We request our chairpersons to kindly take stage and introduce our honored speaker for the plenary session, Dr. Kiran Haridas, who will be speaking on Pharma Industry Practical Exploration of Know the Unknown.
Good morning all. I am very happy to introduce Dr. Kiran Haridas, who is MD and MBA, Senior Director, Asia Pacific and Europe, Medical Management and Scientific Services, Syneos Health International Private Limited, Bangalore, India. He has held a lot of higher positions in various global companies. He is a Senior Director, Medical Management Scientific Services, Syneos, and former Medical Director, Medical Affairs and Clinical Development, Pan Global in Biocon, and former Senior Manager, Medical Affairs in Pfizer, and Clinical Trial Investigator and Duty Physician in Akunova, and has vast experience across therapeutic areas, multiple generic and IP products at global levels, and he's also had more than 15 scientific publications.
Thank you for your introduction. Very good morning to all of you. Honorable dignitaries and my friends here and colleagues. It's an honor to be part of this event. Congratulations to all the organizers for trying to put in the whole community together and try to bring out something worthwhile. So it's great to see that we have this event being organized. So let's begin the presentation. I see quite a few postgraduates interested in more breakfast. Looks like a wonderful breakfast probably. I missed it because I wasn't told that I should have a breakfast. I was told I should make a presentation. But breakfast wasn't mandatory for me, so I missed it. So I presently work with a clinical research organization, a US-based organization. I head the Asia division for them. I am an MD pharmacologist. It's been about 15 years for me in industry. I have worked with various segments, include pharmaceuticals, clinical research organizations, strategic organization, analytic organizations, and I also initially joined as an assistant professor in medical college as well. So I've touched upon all of these things. There's a reason why I say this. One big question directly to speak, should I join healthcare industry? I know it's a very broad question here. So may I know how many postgraduates are here now? Okay. How many of you want to join industry in some part of your career? Okay. Right. I see someone having difficulty in raising the hand. Is that because your professor is watching you? Don't worry, you can raise your hands. Good that you have an idea about what you are looking at in near future. Should I join industry is not something I should address, it's something which you should address. You should be very clear about what you want to do, but then I say explore. Do not take a decision at day one. Do not listen to anybody who hasn't joined industry, who hasn't worked in industry to go for an opinion. Those who do not know swimming can't teach you swimming, right? In principle, try to reach out to right people to understand whether you would want to join industry or no, to make a decision of what you want to do. You know, when I say this, today, when I wanted to make this presentation, I really wanted to talk about the things which you don't hear very regularly. You might enter somewhere, I mean, there are conferences or meetings where you might have job opportunities, a list of jobs available. Correct, but how do you reach them? How do you go to these jobs? How do you get these jobs? Or why, I'll put it in this way. Why many pharmacologists who have completed their MDs say this? It took me time before I got my first job in the industry. They rejected me. They offered me a lesser important role. Why do they do this? Now here are a few dominant beliefs what people have. When I say people, I talk about the pharmacology world, the MD pharmacologists or the people who are doing the post-graduations, the surrounding people. The dominant belief of what most of these people think. Can see here? Okay. Here's my pointer. It's working. Thank you, it's working. Okay, so first and foremost, they think academic is of no help in industry. If somebody says this, and you are never a good person to be in the industry. Trust me, the first point at which somebody will make an evaluation on you is based on what you have done in your academics. Because at the end of your 3 years of academics or teaching, whenever you want to join industry, the only thing that you bring with you is the academics. And if you say I did not do much because I was always interested in industry, I researched everything about industry, but fine, but what is your efforts that you have done during your academics? So first point, academic is of no help, remove it from your mind, concentrate on your PG, post-graduation and your education. Till now, if you haven't done, whatever time left, concentrate on that. Interviewers cannot track my background. Once I remember one person from one of the very good colleges moved back. During his introduction, his CV was his biography, as if he was getting a Lifetime Achievement Award. So many things. Each and everything was mentioned, and everything that fits got into internet and filled everything. When we inquired, also felt he might have done. But very easily, you can go back to the college, you can go back to the professors, you go back to the teachers, and you will know what exactly is happening. So if you're found to be something which is falsifying the results, then you will be the last person to be selected. And they won't tell you that you're blacklisted or something, but you won't be very easily recalled for any interviews. So that's one important reason. So be very, very honest in your approach whenever you reach out to industries. Now here's this, I need to apply for a job before completion of my MD. How many of you already have applied for a job in industry? Anyone? No one? Good. You should not apply before you have a degree. Nobody in the industry is waiting for anybody on earth. These are profit-making organizations. When I say profit-making organizations, there are financials behind that. So nobody is waiting for anybody, it's a game of availability. If you are available at that time, if the company has a job at that time, so you would be the best person suited for that job. So applying 3 months before, 4 months before, 2 months before, blocking it like a bus ticket, it doesn't work that way. So you have to complete your MD. You have to take those 2 or 3 months off, or 2 or 3 months do something what works best for you. Wait for an opportunity till the companies get back to you. And trust me, MD pharmacology is a very hot job in industry. People are looking for it, but at right times. Okay, this one is very interesting. Many of them feel, you come to know about a job and you apply for a job and you feel you are the only one who has applied. You have 4 postgraduates and other 3 postgraduates. None of them know about this job. It happened to me multiple times. All 4 were there for the same interview. None of them know that they have applied for each other. So it doesn't go that way. Be competitive, inform your friends, all of you give a try. The best definitely moves up. Or you never know, these are not specified government organizations. You might end up getting 4 jobs for all 4 of if they're so good. So that's why this will also help in spreading a word about the jobs among you all, and the problem that is addressed, how to approach, where to find these jobs also reduces. This one, they will offer me salary in crores. How many of you think they'll offer salaries in crores? I should ask before. I'm late to ask this. How many of you feel they offer jobs in crores? They do, but not after MD Pharmacology. So you need to start very humble. You have to start at zero. You have to establish yourself in the industry, in your own community, in your own field, in your own establishments. As you grow in your career, definitely there isn't anywhere to stop. There isn't any level to stop. At this point, I also want to add this. I think somebody sent me a question on Facebook. I'll address that. I'm sorry, I don't remember the name from the group itself. They said they call sometimes MD pharmacologists who are in the industry as glorified medical reps. Have you heard this? Right? All of you heard this? Hear me carefully. That is utter nonsense. Okay, that's first. Second, nonsense, I'll tell you why. These reps what you're talking about are the next CEOs of those organizations. They are excellent in their jobs. Their positions are great. What medical does is completely different from what others do. It is an exclusive field in industry. You do your own work in which you are qualified to, unless and until you want to go beyond your zone. You are not glorified anybody. You are only MD pharmacologist. You will be treated as MD pharmacologist till you are good in what you are doing. Now, when it comes to industry, there are many segments in industry. What I spoke about was medical affairs, where people usually have marketing and sales objectives. So that is a little different from clinical research. So remove that thought in your mind that people say that. If somebody is saying that, probably either he isn't aware about industry or he hasn't got into any of his post-graduations. Companies are waiting, I address this, waiting for us to join. Nobody is waiting for anybody to join, but yes, there are positions for all. You just need to find a right position. Is there a right age to enter industry? Is there a right age? Do you think is there an age to join industries? Anybody feels so? After post-graduation, maybe 10 years later. So I'll address that. There isn't any specific age for you to join industry. Every experience counts. And there are jobs and roles which suit each and everybody at their age. Where do we fit? When I talk about post-graduation in pharmacology, where do we fit? Do we fit specifically into one particular role? Trust me, at the beginning, you don't fit anywhere. You don't even fit in medical. We don't even fit in medical when we start. We're very good academicians. So that's our strength. We have to build upon that. So at zero, when you join, when you go for an interview, I'll have points where I'll help you to build up a craft at the end of it. So one thing you must be very clear is your greatest strength is MD pharmacology, what you have done, your academics, your work, what you have done. Use that as your base for all the interviews. Do not copy anyone else. Interviewers are different. Job roles are different. When I say this, as an MD pharmacologist, one greatest advantage people like me, many of them who have had, was you can fit into any therapeutic area. You don't need to be a neurologist. A neurologist can do a trial in neurology. Oncologist in oncology. What about a pharmacologist? He can do trials in any of these therapeutic areas. He is fine to fit into all of this. So in order to do all this, his basic foundation has to be strong. So your work, your studies, your efforts, what you put in understanding the concepts of drugs and drug-related aspects during your post-graduation are extremely crucial. I remember once when long back, initially when I applied, it took me about 8 months before I joined industry. I was in a medical college then. When I applied, there was one industry, without naming it, the person had actually given me MCQs for an interview. So I was surprised. I said, why do we, though I wasn't selected for that, I was rejected. I asked him, why are you giving you MCQ? Why are you giving us MCQs for this interview? So this was about 15 years back. So he told me, you people are best at this now. So this is how I'm judging you. You're best at what you're doing. You are in, otherwise you're out. And that holds good for all. Remember, people who interview are all MD pharmacologists. They know what you did and what they do in their post-graduation as well. So these are some of the positions what you can see here. All of this you can fit. You can fit into marketing as well. Don't go with the wording. You can fit into anything of this. Okay. So here we have a healthcare industry. You can go through it. These are all the places where are the different arms of healthcare industry, where you can have your positions. All are fancy. Some of them, I don't know whether you heard about it. Medical analytics. Have you heard of this? Here is what you will make crores within no time. Medical analytics. And then some of this which you might probably, real world analysis or real world. There are organizations which only deal with real world. Now, medical analytics, here I put the point. What is a strategy? What is all this marketing, sales? They talk about, we are strategizing this product, we want this. Ultimately, what they're trying to do is, they're going to bring some sense in what they're selling. They're not selling chocolates. Here there are some products. They have to be backed by absolute evidence. At the same time, the claims can be fancy, but they have to still say the same thing what the studies or what the approvals say. And that ends up in positioning a product. That is the strategy which they make. Medical analytics. Medical analytics, I'll tell you an interesting thing here. In medical analytics, there are people who spend years together, 10, 15 years before they become experts. Trust me, you are experts at day one. They have not been into medical field or anything. They collect the data throughout the world and come to a conclusion like, for example, depression is treated by a psychiatrist. You know, for this, they need to spend a few lakhs of amount to make sure that they come to this conclusion. This is a very basic level I'm saying. This is how this analytics world works. So in that way, when you go into the core of analytics, we are the best at it. So this is another emerging field which is playing an extremely great role with computation, generics, biosimilars coming up in the market. So you could look at this. So here are some of them which you can look at. The rest of things I think are all familiar, which you must have already heard. Okay, is this clear? Okay. I'm just trying to put a hierarchy here. Just to let you know that this may not be the same hierarchy in all organizations, but in similar, you would find hierarchies like this in different organizations. The reason for why I put this is, what is the growth in industry? We join, what next? How do we grow ahead? Whether we should choose to change industry. They're all personal questions which come up as you evolve in your studies. But in a hierarchy, you usually, you can see at the top most your interns. So many of them even join as interns, but usually MDs are taken up directly at the level of assistant managers. The name itself suggests here, assistant manager means you are an assistant to manager. You're not still having the ability to take decisions. And these positions and grades, what you see as you progress towards directors and presidents, these are not time driven. These are based on your expertise driven, your, your hard work driven. I would not put it as hard work, smart work. You don't need to work hard, you need to be smart. So smart work, how good are you in your work, what you're doing? How good are you in your skills beyond your therapeutics? You will have to interact with people. Now here interesting part which you must all know. You will know how to, you should know how to interact with people who do not have similar degrees, who have higher degrees than you, could be investigators, could be regulators, or people who are lesser degrees, like just, they could be just simple basic degrees and they are doing it. They are in the part and parcel. Sometimes you might find your CEO to be hardly educated. He is the one who runs the whole pharmaceutical company, and you have the highest degree. You don't even report to 10 people below him. How is this happening? So you should be knowing how to interact with all of them. So these are a sense of learnings that we get and people lack when they join the industry. I remember one person in my team, I think 15, 20 days down the lane, there was a corporate meeting in which he had to make a presentation. 4 or 5 questions were asked, and I seen him crying. He came out and started crying. I said, "What's the issue?" He asked me 4 questions, I answered only 2. So it's a completely different world. It's a different world. It's a world of trade-off. So it doesn't matter. It's about addressing the issues there. So your medical knowledge is extremely important, but it might be like 100-page what you read ends up with one single line. That should be your ability to communicate. Right? This, you can grow up to anything. I would say you can go up to the president level, which is the head of your own department from directors. But if you're really good at it, there's no stopping. You can be regional head. I work with, on regular basis, MD pharmacologists who are heading the organizations. I'm not talking about medical, who are heading the organization, who are heading beyond medical. They are in the ranks of CEOs. MD pharmacologists, humble background, humble colleges, same as what we did. Okay, now this is something which we all must understand. The future is not about what pharmaceuticals dictate. It's not about dumping whatever they have and the doctor should prescribe it. It's a patient-centric and evidence-based world. It comes from the bedside and not from the bench side. Moreover, why I'm trying to say this is completely patient-driven. Every single information that you get from the patient side is an exclusive information. So going ahead, people with MD pharmacology who have extensive information and knowledge about drug products, including the history, including the way they are analyzed, they're written, makes a big difference because you have those good presentation skills. You need to work on that. So this is the whole future what you see. So assuming this, more and more doctors would be interested with the type of money people get as they grow. People with just medical degrees also slowly keep moving into industry. So where does your MD pharmacology comes? It comes here. You have an MD pharmacology, you have an edge over everybody, each and every tries to enter into industry. Your MD pharmacology will make a big difference when you join industry. And I can tell you what, 15, 20 years back, people weren't much aware in the industry as well about MD pharmacology. They were wondering why many MD pharmacologists do enter, but now, I was surprised, I can tell you this. About 3 months back, there was a talk from our CEO, and I was having a discussion, so he told me we need to hire 7 people as early as possible. So I did not say anything. He is based in US. So his center, why you don't have MD pharmacologists coming out now? I was amazed to hear that he said industry is a synonym for MD pharmacology. Now, people are recognizing it. Let's build up on this recognition. I have done MD pharmacology. No, I have done MD pharmacology, right? On the peak of your voice. You have to speak up your background very nicely. Many people do not have, I don't know why, many people do not have that strength to spell out MD pharmacology. I have 5 minutes more. Thank you. Okay, spell out more. That's the most important thing you should do. Disruptive points for you. Here are some of the disruptive roles. I have addressed a few of them. You can read them. I wanted to take a few questions, but so 4 minutes left now, 1 minute over. So, right. Job roles are therapeutic area specific. If you have worked in oncology, if you have worked in specific areas during your MD, so don't try to stress upon that when you go to industry. Be independent, just go and join and be open for therapeutic areas, any therapeutic areas. Do not project yourself as an expert at zero. And these 2 points, be very, very honest and humble when you go for interviews especially. And this will take you a long way. Disruptive points to build your craft, you can read these. These are something which are very important. If you can learn some IT skills while you do your MD, that will really help. When I say IT skills, they should be really analytical IT skills, not just making presentations, not just putting slides, PowerPoint presentation. There are people in the industry who will do presentations for you. So you should, a few analytical skills will make a big difference. Now here, two things I always wanted to address this, so I'll quickly address this. When you apply for a job, job might not be there, but after 4 months, you might get a call from 4 different companies. That time you must see, I have 4 calls from industries. They accept all 4, reject 3 of them, and take 1. And they're very proud about the fact that they've rejected 3. Remember, your rejections are noted there. Your rejections are noted, and after accepting, never reject an offer. So what you must do is, before accepting an offer, make sure what decision you're doing. You want to go or you don't want to go. As simple as that. So never reject an offer after accepting it. So this is something which I see very common in present day pharmacologists. They take up, in fact, the one person came with the job offer of another company when he came for the interview. So I made sure he had a good job in the same company where he brought that from. That's the only possibility you can do. Now, this is just slide tells you about how role of we people here sitting have to play in building up the community for us. We have to bring in good environment. Remember here on the right side when you see the key stakeholders, 4 of them, academics is an independent stakeholder in bringing up the best pharmaceutical industries. So 90-95% of the people in industry are non-medical. A very small percent of the people are medical, paramedical, related medical. So it's a very important role in academia where you have to make sure that the people, the students are well trained in what they're doing. I think I should be having a half minute. So let me conclude this by saying all those people who did not raise your hands as well. Good to be in academia, and I would love to be in academia as well. But I'll tell you, give an honest try before you say no to pharmaceutical industry. Give a try and see how it works. Give a try for clinical research and say, you're sure you want to be somewhere, but for something which you don't know, just give a try and make sure that you've explore it, because as MD pharmacologist, we have an exclusive opportunity to explore things over and above academics. I repeat, over and above academics. So we should make utilization of that. Unless we do that, we will be failing in a very good opportunity that we all have. While I say this, I'll complete my presentation. I wanted to take some questions, if there are any. Thank you.
Thank you, Dr. Haridas, for nice presentations. And it's true for all professions. The key of success is always hard work, smart work, and honesty. That is for all. So, for his forum is open for questions. Yes.
Thank you, sir. Thank you very much. My name is Dr. Sushil Sharma. I am the professor and head of Department of Pharmacology at AIIMS Mangalore. And as a newly established AIIMS, we are actually about to start our MD program next year. So in your opinion, because you've been an MD pharmacologist and you've seen the kind of training that is provided to them, do you think we provide sufficient training to our MD students so that they are a good fit to the industry? Or do you feel that there are a few lacunas that exist in our system and maybe there is an opportunity to plug them during our training?
Thank you, sir. I did not want to address this, but thank you told this, because many academicians would be unhappy if I say this. See, I'll tell you, the curriculum is excellent. MD pharmacology is excellent because we have all studied that and we reached here through the academics what we learned. I still have a respected sir's book, the recent most edition as well, which I keep brushing up myself very regularly and then keep myself updated. But I think we have a sufficient 3 years of time, a little bit of time, maybe 6 months or maybe 8 months dedicated towards learning not pharma industry to learn some specific financials or important things which will help them make viable in industry at start. Now, what do you mean by this? You know, you might know hundreds of drugs, we might know hundreds of things which are related things in it. But in a pharma, what matters when we do our MD pharmacology is, what we forget, or most of us forget is, to do a research we need fund, right? Somebody has to give us money. If we have 100 rupees, we should know how best we utilize this 100 rupees. So when I say financials, there should be a literacy that should be developed, advocacy or literacy developed, where they can understand a little financial literacy about how funds are handled in case you have it, whether it is researchers or whether it is using it in particular things. That's one. Second is, there are industrial internships that do happen everywhere. They might not guarantee you anything like a job, but they would definitely help. There are many of these internships now, they are available. I think one 2 months, 3 months course, small course, which is embedded in the curriculum, where they give them a complete exposure towards non-academic healthcare, which will be very crucial. I think ma'am will also be talking about clinical research when she'll be having a session. So that will also be very crucial when you look at how do you progress in it. But as you said, sir, if I have to give a single answer, we aren't equipped for it. If somebody has to select somebody from an industry based on only MD pharmacology, a non-MD pharmacologist interviewing him will never take him to the industry. As simple as that.
It was an excellent question, sir. In fact, this is the right time to address it because the PG curriculum is getting revised. The CBME curriculum is, the new curriculum is being introduced, and everyone wants to introduce something like some industry exposure into it. So that the
That's excellent, ma'am. I think that's an excellent move if that happens. And I suggest, very honestly, partner with people who are working in industry for this and try to get a feedback from them and then build a curriculum which is more specific and relevant for global industry. So that's excellent. That's nice to hear that something has been happening here.
Yeah, hello.
Hello, sir.
It's a very nice talk. I'm Dr. Dinesh Padyal from CMC Ludhiana.
Hi, sir.
And new curriculum, there is no option that you have a postings in industry also. 2 months up to, you can go up to that. We want people who are from industry to open them and offer these training periods. Because right now, which are available are competitive, some of them which are, only few get selected, very few. Right now, we are open.
So I have
we want more opportunities to coming from the industry side.
Sure.
So then I think this is a very good platform right now. Everyone is ready for that.
Great idea. I have a question for all, one thing for the professors. How many of you approached industries for such an opportunity? So answers were no? No for it? Okay. So I suggest this. Rather than that, I would say the best thing would be,
Sir, can I say something? Sir, I'm Dr. Sarita, Professor, D.Y. Patil Medical College, Pune, Department of Pharmacology. We have this industrial posting policy since 2014. And all PG students, 6 PG students per year, they go for industrial posting for 2 months in their curriculum. So it's helping them a lot, and most of them, almost 100% of them, they get absorbed in industry because by even the social contacts and the exposure, they can decide whether they want to join pharma industry or not. So it's helping them a lot. But at the same time, when they apply for internship, many of the pharma industries, many of them, they say, we don't have this policy of internship. So why can't, the MD pharmacologists who are sitting at higher position there, they can change the policy and they can allow these MD pharmacology residents as an interns for at least 2 months so that they can get good exposure.
I think it's a good start. While many of these questions might not hold good for people staying in Mumbai, Delhi, Chennai, Kolkata, Hyderabad maybe, where you have direct pharma exposures. For people who do not have these pharma exposures in these places, what should I do? Now I tell you something here. What could be the reason when they say no? These have to be very strong policies between your college and the industry. Industry will have some, very confidential information, and people who come for internship for 2 months, 3 months, when they leave back, they are worried about this confidentiality, so that could be a problem. The only way to this is, reach out to the right people in the industry, which is medical. medical people, try to have a strong documentation between your department or your college and the industry. Nobody can refuse once you have that. And these are honorary, neither nobody is going to get paid on either side, so it's very easy that you will get through it. But have these documentations done. We have tried this in few of those. We're successful in that. PGs do benefit immensely with this. They love their jobs after this. I think as seniors, if you get together and form this, the coming community will be grateful to all of you. They do. Great. Sure.
Good morning.
Hello.
I am Dr. C.M. Kamal, Professor Pharmacology, Government Medical College.
For a change, nice to see you directly, Dr. Kamal. We only met through emails.
Sir, as you have already, probably you have explained it already, and people over here, probably I am happy to share is that Dr. Kiran Haridas and me have worked together in Manipal Ekinova in industry. So there is a policy in the country, several countries, that where there is a population less, like Israel, that they have to pass certain period in army. So similar trend can be applied to that also. After passing immediately MD, students are looking for cozy jobs where they are comfortable. Academics is a little bit comfortable as compared to the industry. So my opinion is each and every student can try. There are hundreds of the jobs available in the pharma industry, and as you have already told, that if they can satisfy, they are satisfied for their particular job, they are absorbed, they can continue with it. Otherwise, I have done work with you more than 6 months. I feel not much comfortable, so I again can back to the academic. So same thing can be done by the students. It is a better way, but the students probably may not want to compromise on the salary. Initially, pharma industry are giving lesser salary. So they have to compromise at least at somewhere they need to compromise.
True.
In that problem probably can be solved. Otherwise, getting a department and industry relationship, seems to be a wild job, but it is a very difficult one also. Okay, yeah. What is your opinion on that?
Thank you. Though thank you for that, sir. I think that was the dominant belief overall what I was addressing. One thing is jobs in pharmaceutical industries are. I'm from industry. I'll be biased, so I'm sorry if I'm biased, but it's good. It's not very hard. It's not hectic. Now tell me, is this hectic? On the lighter side, you get to travel on some of the best flights. You go to best restaurants in the world. You will never go with your family. You'll go and stay in the restaurants and places where you and me would like to rob something from the hotel. What else I can say? You will be respected in any part of the other world when you go, when they know you're done your MD. You will be the face of that company for regulators. You talk these name of these regulators, you're standing in front of them. You go and meet some of these PIs who have missed up on their opportunity there. You're trying to teach them out there how this rightly it's done. You will be the face of that organization where you go. Your expertise in MD pharmacology matters. Your card reads MD pharmacology. You will be in the medical world. You tell me, how is this less interesting? How is this more tough? I don't understand. What is more tough? Just on lighter side, 8 hours making 4 publications is tough, isn't it? Nowadays you have to make publications in industry, in academics. Dr. Ashok Shanes a few days back, I just gave him a call and I said, the day I get bored in in industry, you please give me a job in your department. He said, he promised me, I will give you. He said, why do you want? It's much easier. I said, who the hell said it's easier? So I said, sorry sir. Then I realized how difficult it is there as well. So that's what my last slide was. Give an honest try to say, before saying no to industry. You are right fit to the industry. You are not right trained for now. It will take 2-3 months for you to be trained, not more than that, 4-5 months, max to max. So you're not fit for now, but definitely going to be fit for it. So take that opportunity. Don't lose it. Don't never lose it. 15 years later, oh, I should have done it. Very bad. 20 years later, very bad. Postgraduates here, take it. This is what I will say from my point of view.
So with this, we end the session.
Sir,
if you
okay.
have any question,
Yes,
please discuss this during
Sir, good morning, sir. Sir, I am from Andhra Pradesh. Sir, here, sir.
Please.
Sir, I am final year MD Pharmacology.
So it's the last question we'll take. Right.
Sir, unfortunately, I have 3 queries, sir.
See, one is, I'll conclude. One is this honorary session, I'm not taking any charge. Okay. I'm finishing it time, it's already over time. We'll speak personally.
Oh.
Sure, we'll talk, we'll connect.
Yes, sir.
Oh, is that for me or for the public question?
It's for the entire our academics.
Ma'am, you want to take that? You have a very interesting session now,
Sir,
more interesting. Spare your questions for
sir,
ma'am's
I will
session.
ask 3 queries, sir. You may answer
Good, good.
now or later, sir. Your wish.
Go ahead.
Sir, my query number 1 is I suggested my junior first year freshers to join some MBA online course for 3 years so that they will have dual degree by the end of MD Pharmacology. Will it be any useful to them? Query number 2 is what is the best department for us to join in after MD Pharma, like medical affairs or medical analytics whatsoever, so that after 1 year we can shift our curriculum, our job profile from employee to employer, not as an employee. What is the best way to join so that we can promote ourselves to improve ourselves to become employer? And my query number 3 is we heard a lot from many people, sir, that pharma industry have no longevity. After the age of 50 years, they will throw you out.
Throw you where? Throw you where?
Out. We heard it that they don't. They will remove you so that how expert you are.
I'm very scared now.
Actually, we heard it, sir. Unfortunately, I heard it from a person who joined industry and he came back.
Never reach back to him. He's the wrong connect. Don't. I was talking about such people only. Don't connect them back. Nobody is going to throw you back. You said something about MBA.
Yes, sir. MBA and employer. Yes.
Quickly, I'll answer now. They all gave good comments. Now comments will go bad because I'm over time. See, first of all, we'll discuss this. I'm here around. Don't do an MBA at 0. Only MD Pharmacology is sufficient. 1. MBA is a training. MBA is an experience. For us, you have to take that as an experience. I did an MBA 12 years after my MD Pharmacology, clearing all exams, free of cost, doing it with entrances. Do it once you have little experience and you reach a stage where you can take responsibility beyond medical. When you want to go into marketing, sales, when you want to become a CEO of an organization, which you will be, because you made sure that your question was asked. So if you want to do that, do an MBA 10 years later after you start. First do well in the basic part of this. Starting MBA will not have any value because you have an MD degree. It's like doing KG class after completing 10th standard. Thank you.
Thank you, sir, for that enlightening session. I now request Dr. Sneha R. Ambwani to present a memento to Dr. Kiran Haridas as a token of our appreciation. Thank you, ma'am.
I now request Dr. Rakesh Kumar Dikshit, Professor, King George's Medical University, Lucknow, to kindly present the mementos to our chairpersons, Dr. Sneha R. Ambwani and Dr. Punnagai, as a token of our appreciation. Thank you, sir. Dear delegates, we have some fun questions for you, and we request your attention and kind participation as we have goodies to give away to those who fulfill the conditions of the activity. So let's begin with the first one. As ladies go first, the first one is for our beautiful ladies. A lady among you who is wearing the maximum number of earrings. A lady among you who is wearing the maximum number of earrings. First come first. So, you can raise your hands or... Max. Okay, we have a... Awesome. Okay, we have 3. Anybody else? Anyone more than 3? 3 it is. Okay, we have 3 plus 3. Dr. Haifa has the maximum number of hearings. I request. Yeah. Okay. Please give a big round of applause for Dr. Haifa. Thank you. We have with us Delta Healthcare, who offer medical simulation services and are into developing skill labs across India. They would like to introduce a new technology which will be demonstrated here. Over to you, Delta Healthcare.
Good morning. I am Bharat from Delta Healthcare, and we are a company that offers simulation services and set up medical simulation laboratories across India. With more than 200 customers and more than 40 distributors, we are the fastest growing company in this space in India. And the engine which drives the pace of our growth is innovation. And now I have come here to introduce a new product that we have launched. And here you can see, can anybody guess what this could be? Just a wild guess. It could not be any... Whatever comes to your mind, just see how it appears. Based on its appearance, can anybody guess what it could be? Yeah, monitor is one answer. Another one? Yeah? Defibrillator is one answer. And what else? What else it could be? Ventilator is one answer. Actually, it is everything. That is the thing. The idea behind this product is compressing a complete simulations laboratory in a yellow color bag. Imagine that you can just bring a child's toy or any cloth store mannequin or anything in your disposal, and you can convert it into a simulated patient and run any kind of scenario at any environment. Imagine that you can get a complete simulation facility in a bag and you can take it to your classroom or anywhere. And imagine the effect it can bring to you, to transferring your knowledge to your students. And that is what this device is. And we are just right opposite to our stall is just right opposite to this auditorium. And I invite you all to come and have a look at this device in our stall. And I take this opportunity just for the invitation. Thank you.
Thank you, Delta Healthcare. Now, we won't leave out the gentleman here. So the next one is for our gentleman. A gentleman who has his wife's photograph in his wallet. So obviously, it's for the married lot. If we don't find any husbands who fulfill this criteria, the question jumps to the eligible bachelors. Any? Okay, yeah, okay. First, yeah, raised hand is... Okay, I think we would give away the goodies to all those husbands who had the wife's photograph in the wallet. Yes, please come forward, sir. It should be the wife's photograph specifically. Okay, wife and kids photograph is also there, so you get double goodies. And yeah, kids photograph is also there. All the kids photographs are also. We have Dr. Rakesh Kumar Dikshit, Dr. Badiyal sir, Dr. Ratnakar sir.
No cheating, no cheating. That's what Dr. Kumar. That's what. How do we confirm you have? You should also have a photo with
Dr. Nishant and Dr. Manjunath. We have 6 winners. Please give a big round of applause to all our wonderful gentlemen here who carry their wives. Okay, we have one more. Please don't borrow somebody else's wife's photograph and put it in your wallet. Sir, you have 2 pictures, sir. amazing. Please give a big round of applause to all these wonderful people. Thank you so much. Thank you so much for your active participation. I'm glad you enjoyed this activity as much as we did. We shall begin with the next plenary session for the day. We have with us our esteemed chairpersons, Dr. Rakesh Kumar Dikshit and Dr. Ranjan P. Nerurkar, who will be chairing this session. Dr. Rajan P. Nerurkar. Dr. Rakesh Kumar Dikshit is a professor at King George Medical University, Lucknow. He is the General Secretary of National Association of Pharmacology and Therapeutics, member of the Medical Education Unit, member and coordinator of the NMC SSRs, member of National Board of Education for Examination and chairperson of Ethics Committee. Other than this, he has given several guest lectures, published books and book chapters, has 178 scientific publications and 6 fellowships, reviewer in 12 scientific journals, and also has received the Best Teacher Award in 2020 and 2022. We welcome you, sir.
We have Dr. Rajan P. Nerurkar, is the professor and head of Department of Pharmacology at Topiwala National Medical College, Mumbai. Sir has received COVID Warrior Award for the work done for getting donations for the Nair Hospital during the pandemic. He also has set up a well-equipped CAL lab, first of its kind among all government municipal medical colleges in Mumbai, and also the skill lab of the college. We are proud to have you here today, sir.
I request our chairpersons to kindly introduce our honored speaker for the plenary session, Dr. Nitya Gogte, who will be speaking on setting up a clinical research division in medical institutions, opportunities and challenges.
A very good morning to everyone. Now I feel honored to introduce Dr. Nitya Gogte, who is actually pioneer in many fields, in many clinical research. She is professor and head, Department of Clinical Pharmacology, KEM Medical Hospital, Mumbai. She is the first female editor of the Journal of Postgraduate Medicine, one of the most reputed journal in the medical science. She is one of the former member of advisory board of the British Journal of Clinical Pharmacology. We actually salute her. Chairman of Ethics Committee, Tata Memorial Hospital. Her work on the Vivax malaria has been recognized everywhere, not only in India, pan-globally. She and her team has worked with the several vaccines in all phases of the drug. Many times she is involved in the clinical trials, and because of that she has been given a topic which is actually related to the clinical research. So I am sure that she will enlighten us regarding the clinical research setup in the medical institutes. And one thing is more, that chikungunya vaccine for the childhood immunization, her role is also in that particular vaccine. Thank you, madam. And I request you to please give us some enlightenment regarding the setting up of a clinical research unit in our medical institutes. Thank you, madam.
Good morning. Respected chairpersons, thank you for a very kind introduction, and ladies and gentlemen. I'd like to begin with a study which was published in the Journal of Internal Medicine. This is a study from a clinical research division in Sweden. In the late 18th century, King Gustav III decided that coffee was poison, and he ordered a clinical trial. The king condemned a convicted murderer to drink coffee every day, and the control was another convicted murderer who was condemned to drink tea every day, and the two of them were identical twins. The outcome of interest was death, and 2 physicians were appointed to determine the outcome. If you look at the results of the study, the 2 doctors, they died first. The king was murdered. The convicts enjoyed a long life. The tea drinker died at the age of 83. and the age of death of the coffee drinker was unknown. This is a true story. The conclusion was that none was possible. External events and biases may have confounded the results. Coffee was forbidden in Sweden in 1794 and yet again in 1822. And it was decided that kings should not set up clinical research divisions. So who should set up clinical research divisions? Over the next maybe 17 or 18 minutes, I'll take you through a personal journey of the clinical research division which exists in our institution. And I'll also do a SWOT analysis, strengths, weaknesses, opportunities and threats, and perhaps address some of the questions which were asked of the earlier speaker, Kiran. I'll start with a disclaimer. All the views expressed are personal. I have no intent to disparage any organization or individual, and my spouse works in the pharma industry. All right? This is going to be the outline of my presentation. I'll start with the beginnings, the setting up of the CDRI unit in the late 80s. I'll then talk about being in the right place at the right time with our early studies in infectious diseases. I'll talk about strengths and opportunities at that point. How does one get funding from the pharmaceutical industry and government agencies? What are the weaknesses and threats that we pharmacologists face actually on a day-to-day basis? And end with some of my own personal perspectives and how my journey has really taken me up to this day. I think if the Department of Clinical Pharmacology at KEM Hospital exists today, if the Phase 1 unit, if the Clinical Research Division exists today, it is only because of the vision of the Government of India. The Central Drug Research Institute, CDRI, was established in 1951, about, I think, 13 years after 1947 when we got our independence. So when CDRI started developing molecules, they did not know where to test these molecules, right? So CDRI had the vision of setting up a clinical research division in the pharmacology department of the KEM hospital. So anything that we are today is full credit to our government, right? And later on, Dr. Hamid from CIPLA, along with CDRI, actually signed, very few people know this, they signed a memorandum of understanding with the Municipal Corporation of Greater Mumbai, and they supported a 3-bedded unit for a long time. So we had a mixture of CDRI funding, which is the government funding, and the vision of the pharmaceutical industry to invest in a clinical research division right in the late 80s and early 90s. So that unit looks like this today. We call it a Phase 1 unit, but you will still see that the CDRI name is mentioned there. Because it's a Phase 1 unit, we have all the emergency facilities, an emergency trolley, we have a defibrillator, we have 3 multi-para monitors, and we have like a hi-fi ECG machine which gives you multiple corrected QTC intervals as per the industry requirement. So to my mind, I think setting up a clinical research division, I mean the greatest strength and the greatest opportunity, the fact that you are in a medical college, the fact that your medical college is attached to a hospital which does patient care, patient service, I think is the biggest strength and the biggest opportunity that all of you have to set up a clinical research division. The next thing that I'd like to talk about is actually being in the right place at the right time. Now, in the early 1990s, the city of Bombay, it is Mumbai now, there were 2 problems that we were facing. We had the problem of multi-drug resistant typhoid fever. In those days, we used to use chloramphenicol, which is called chloromycetin. Yeah, that's what we used to use. And it started in a suburb of Bombay called as Dombivli. So it used to be called as Dombivli fever. The second problem was all the successes which India had in terms of reduction in malaria came back up with the resurgence of falciparum malaria. So much so that I took training for 1 year to identify, this is Plasmodium falciparum actually, So identify malaria. So what happened is, Dr. Nilima Kshirsagar, she said, let us give the reports quickly. At least the patients can be treated. At that time, remember, we did not have artemisinin derivatives. We just had chloroquine and we had quinine. That's all we had, right? So Madam set up the OPD, which still runs today. And we used to give the reports within 30 minutes so that you had early diagnosis of malaria. The second thing that happened was that we realized that Vivax malaria had started coming back. So Madam had the vision to actually set up chloroquine levels. And with a lot of courage, we wrote this letter to editor that despite having chloroquine levels in the therapeutic range, Plasmodium vivax malaria was actually coming back. So this was a letter to editor which was published in the Transactions of the Royal Society in Tropical Medicine. The next thing that we did is, because we were working with diagnosis of falciparum malaria, today all of you must be using it routinely. It was uncommon then. You have what is called as the RDTs, Rapid Diagnostic Tests. And one of the first RDTs was this parasite F test, right? And there were two very big people in malaria. They're still very big people, Professor Nicholas White and Professor Jundra Carbong. They used to publish a lot of work on the use of this parasite F test for the management of falciparum malaria. But what we found in Bombay was that we did the parasite F test for diagnosis, but as we went forward, the parasite F test continued to be positive. So the parasite had died, but the parasite DNA was being picked up by the test. So what we said was, it is not a good prognostic indicator. And again, we had the courage to write this letter to editor, which got published in the Annals of Tropical Medicine and Parasitology. So what happened after all of this is Sandoz, now that's Novartis, they actually came to us and said, will you do a study for us? Will you do a randomized control trial which compares chloroquine with artemisinin derivatives? And we did that. And when the study was being completed, actually, the WHO came to us and said that, will you do a study on filariasis, right? So this study was comparing diethyl carbamazine versus a combination of diethyl carbamazine plus albendazole. And this study we did along with Dr. B.S. Garg, the Community Medicine Department of Sevagram, Varda. And this is what I looked like at that time. And these were the best years of my life. I have not done community medicine, I've done MD pharmacology, but I learned a lot about practice of community medicine with this study. What happened after this, and this was 2000, 2001, so the floodgates sort of opened. Serum Institute of India came to us with a rabies vaccine. Bharat Biotech came to us with a chikungunya vaccine, and Sanofi came to us with a flu vaccine. And I am absolutely grateful to each and every one of these companies for the trust vested in us, the faith vested in us. Before me, you had an industry speaker. I think academia and industry are nothing but two sides of the same coin. We just do the same things in different places, if I may say that. Once these companies came to us, they said, "Arey, aapke paas to pharmacy nahi hai. You should have a pharmacy." So I said, "Hamare paas to paisa nahi hai." So okay, so they gave us money. We invested in a pharmacy. And then they said, "You don't have a deep freezer. You don't have calibration. You don't have this. You don't have this." I said, "We don't have money." Okay, so they gave us the money. And then we have refrigerators now, 228, with calibrators, and which is now backed on cloud. This term cloud I have learned in the last 3 years, right? So we have 24-hour power backup and a deep freezer. Then in the mid-90s, 96 actually, you had GCP coming up in a big way. Some of you may remember the first ethics committees in India were set up actually in 1986-87. It was not as big as it is today. And the company said, "Oh, but you don't have an informed consent room." So we went back to the dean. Yeah, our deans have been very, very kind to us. Set up an informed consent room. And then they said, "Oh, but you need to have place to archive everything." That time there were no clouds, no? So we needed to have physical space. So then we created an archiving room for that. So that's how the clinical research division sort of grew. And if I have any take-home message for you, I said we were in the right place at the right time. You can always find something which is right for you, right? You find your niche, whatever it is, and you sort of expand on your niche. One of the most important things I think all of us need to remember is pharma industry funding is never adequate. Remember what Kiran said, it's a profit-making organization. So the moment the study is done, they will pull out, right? So your MyBap, your father and mother, is actually the government of India, yeah? So we applied to the Indian Council of Medical Research in 2000 and we got an advanced center. And the reason is we wanted to also develop on the academic front. So thanks to the government of India, Indian Council of Medical Research, we have an HPLC today. And one of the things that studies actually demand, and our KEM people used to tell us, "Aapka to private hai na sab. Why do you want to send us samples to biochemistry? You do your own analysis." So we set up we had an automated biochemistry analyzer, we have a semi-auto analyzer, we have an automated cell counter. And along with all of this, what we did and what all of you should do is actually publish. The more you publish, the more you get recognized actually. Our work in malaria particularly, we've done a lot of studies, got published in the British Journal of Clinical Pharmacology. Our work in TDM was recognized by Therapeutic Drug Monitoring. And I think the crowning glory in our careers actually came. We have 2 systematic reviews on artemisinin derivatives in Vivax malaria. This was a Cochrane systematic review. That's something that the entire department is proud of. And we're able to average about 10 publications, 10 original articles in a year. So what this does is, when you do that, I think that's the next thing, it draws in collaborators. So other academic institutes actually want to work with you. So we had the TIFR that came to us and said that, would you like to work with us on metabolomics of malaria? So we did that. And then we had Indian Institute of Technology, IIT. They said that we'd like to work on proteomics of malaria. And then Bombay College of Pharmacy and Manipal College of Pharmaceutical Sciences, MCOPS, with whom we have a long-standing collaboration. So we do work with them on population pharmacokinetics. So when you do this and you publish, you actually attract academicians as well. And then we decided that funding from Indian Council of Medical Research alone is not adequate. So we applied to the Department of Biotechnology. This was when Dr. M.K. Bhan was heading the Department of Biotechnology, and with him was Dr. T.S. Rao. I remain eternally grateful to Bhan sir because he set up this Clinical Research Training Center, CRTC. We still have the center today. And that brought in more funds, and it also brought in more government grants. So my one advice to all of you would be to support and sustain yourself through government funding. You might want to ask, what does your own hospital do for you? And I'll answer the question right at the end of the presentation. While all of this was going on in the background, we've always been doing pharmacovigilance. Some of the seniors sitting in the audience would remember that pharmacovigilance in India actually started with ICMR. ICMR set up a lot of pharmacovigilance centers, and then the DCGI, that is the Drugs Controller General of India, set up his centers. And I remember Shirsagar madam was quite upset because we were not selected as an ICMR center, but later on we got selected as a center for the Drugs Controller of India. And I think today we are what is called as an AMC, ADR Monitoring Center, and a regional training center for the PVPI. And our highlight actually came last year. This was a national conference, and we realized that we were ranked 3rd in India in terms of reporting adverse reactions to the government of India. So the effort that started in the late 80s and 1990s actually bore fruit much later. So my take home here is you need a lot of patience and you need a lot of perseverance with what you're doing, because your fruits are not going to come immediately. Just like Kiran said, you're not going to get crores as soon as you give your first interview. For those of you who don't have a center and would like to set it up very quickly, you need to link up with your closest ADR monitoring center. They write to the PVPI, currently headed by Dr. Rajiv Singh Raghuvanshi, and he's very, very proactive, and he gets back to you and says that you are appointed as a ADR monitoring center. So that's how the system works. The next thing, and this is this old adage, no? Keep your friends close and your enemies closer. You have to network, and I'm really, really, really grateful to each and every one of these heads of department over the last 25, 27 years that I have been in KM. Whether it is medicine, whether it's pediatrics, OBGYN, community medicine, we are really grateful to all of these clinical people, heads of department, for their outstanding support in everything that we do. Pharmacology, whether we like it or not, remains a dependent department if you want to do clinical research, unless you have something really strong of your own. So networking with these people, I think the most important question that one needs to answer with clinicians, what is in it for me? मेरे लिए क्या है इसमें? So if you are able to add value to what they are doing, whether it's therapeutic drug monitoring or pharmacovigilance, they will automatically buy into what you are doing. And of course, we continue to apply to the government, and currently we are sustained by the PKPD, the Pharmacokinetic Pharmacodynamic Center of the ICMR and the Product Development Center of the ICMR. I just want to, I have a few more slides left, to say that everything that I have said, everything that I've presented, it's really not mine, you know, it's the legacy. Professor U.K. Sheth was the father of clinical pharmacology in India, and Dr. Nilimak Shirsagar was the person who actually set up the first, separated the department on the 1st of June 1995, and then it was taken over ably by Dr. Urmila Thatti. I have been head of department only for the last 2 years. So everything that I have spoken so far is due to true, very, very strong women whom you can see here, and of course their teacher. Nothing that we do would be possible without students. And we are really, really blessed to have some of the most excellent students. When I say students, it's MD, it's DM, it's MSc, it's PhD. We have people who rotate with us for 6 months, 1 year, we have observers. So I'm grateful to all of these students because they help you drive and sustain what you are doing. And as you can see, they've also been my strength and my support during the pandemic. And if I have to give you one advice for setting up a clinical research division, please have women. Yeah? The more women you have, the stronger your department will be. And of course, students also help you to organize workshops. This is our canvas of clinical pharmacology workshops, which we have run successfully for the last maybe 3 years or so. Finally, I haven't talked about weaknesses, and I haven't talked about threats. Right? When Dr. Thatti took over as a head of the department, she said, Nitya, we should get an HPLC from the municipal corporation. Madam retired. We are following up the files for 12 years. We still haven't got an HPLC from the municipal corporation. So I remember Madam retired during the pandemic, and she said, Nitya, jara wo HPLC ka file jara follow up karna. Yeah? I'm still following up the file. It's 14 years. We still haven't got an HPLC. So what I'm trying to say is, in a medical institution like KEM, that's why I started off with a disclaimer. Remember that patient care will always be a priority. Aapko kyun HPLC chahiye? Aapko kyun auto-analyze? Why do you need something like that? No, so patient care is front, and then you have teaching, and then research comes somewhere at the back. So it is your biggest strength and your biggest opportunity, but it's also your biggest weakness and your biggest threat. So I'll leave you with that. This is a personal opinion. This slide actually shows you MS Subbalakshmi. MS Subbalakshmi, some of you may know, was born into a family of Devadasis. Yeah? And I read 2 books about her. One is a book which I finished recently. It's a book called Incarnations by Sunil Khilnani. And the other book is, I think, A Life in Music. That's the book. And one thing I learned from that book was that before MS Subbalakshmi gave a concert, like before she would come on stage, she would have 3 full rehearsals with orchestra, 3 full rehearsals. So can you imagine? So somebody asked her, why do you have 3 rehearsals, no? So that when she came on stage, everything was perfect. So she said, that's the distinction between, so the term in Tamilian is toril. Toril is work, the job that you do. But she didn't consider it a job. She said it's toril dharma. It's my duty, my devotion to God or to music or whatever. The second person, this is a very nice YouTube channel for those of you who are interested in music and dance. It's called as Notes to Myself. And I read an interview, listened to an interview with T.M. Krishna. Some of you may recognize him. And he said that my teacher would never praise me, even if I did very, very well. But today, I have to praise my students. So I think we are living in a very different generation. And I'll tell you why we are living in a different generation. We are living in a sort of, I wouldn't say generation, living in a time where there is so many distractions around all of us. You have WhatsApp, you have Facebook, and right now I'm speaking, but somebody somewhere is checking WhatsApp, like as if something very important has come in the last 10 minutes. So we're living in different times, and I would recommend that all of you see this social dilemma. It's on Netflix. Yeah? It's a very nice statement there that if you're not paying for the product, if you're not paying for WhatsApp, you are the product, right? So I'll leave you to ponder over it. And this is my last slide. In 1997, I was in Delhi airport, ICMR meetings, and waiting for the famous Air India. Air India in Delhi, earlier at least, used to work like this. The 4 o'clock flight would become the 5 o'clock flight. The 5 o'clock flight would become the 6 o'clock flight. 6 o'clock would become 7 o'clock, and finally it would take off around 8:30. That's how Air India used to work. I'm not sure how it works nowadays, but that's how it used to work then. Madam was there and I was there, and we were sitting because we were just waiting for like 4 hours for the flight to take off. We had a 4 o'clock flight. And there was a person whom I will not name. He was sitting. Madam recognized him. He was a former municipal commissioner of Bombay. And they chatted, of course. I was a nobody. And then after some time, somebody came, "Arey Nilima ji, aap kaise hai?" "Badhiya, badhiya." So madam said, "Okay." And then one more person and one more person and one more person. I said, "Now this is Dr. Shirsagar." And somebody came and said, "Arey Nitya madam, aap kaise ho?" I said, "Chalo, at least one person recognized me in Delhi airport." And then this Mr. X was just sitting. Nobody recognized him. So I asked madam, when he got onto the flight, I said, "Madam, so many people came to see you." She said, let me recollect, "Swadeshe pujyate raja, vidwan sarvatra pujyate," right? I think I learned a very important lesson that day. And I'll end with this quotation from Barack Obama. When he became president, somebody asked him, how does it feel to be a president? He said the presidency is actually a relay race. There was somebody before me who passed on the baton. I have taken the baton. My job is to pass it on to the next person. This is the biosafety laboratory which we have set up. I could say I have set up in the last 2 years. I have taken on the baton from Dr. Thatte, who took on the baton from Dr. Shirsagar, who took it from Dr. Yokesh. What I can do, what advice I can give you, is to do the best you can and run that part of the relay the best you can. Yeah? Thank you, ladies and gentlemen, for your kind attention.
Thank you, Dr. Nitya, for a very excellent presentation using SWOT analysis. You have, in a nicely summarized the journey of one of the best clinical pharmacology department in India over the last 30 years or so. You are so meticulous about each and every step by which your department has grown to this level, and you are carrying the legacy of Dr. U.K. Sheth, Dr. Nilima Shirsagar, and Dr. Urmila Thatte, and now she is one of the stalwart of clinical pharmacology. Now, any questions from the audience?
Ma'am, there
Yes,
is
sir.
the first step to set the clinical pharmacology lab. That is, I think, that is the biggest challenge.
Absolutely.
Because the most resistance in our mind setup, and that is how we start and we just stop immediately. So, how to cope up with that? What are the strategies which can be utilized in a college like AIMS, Mangalagiri is a new college, but has potential to develop clinical research unit. So how he or other department they can start? Because the first step, if you have started, then the journey is started and you reach the destination. Most of the time, I think the person think that clinical research lab should be established, and they think only. So how to cope up with that?
So you're very right, sir. And I think the most important thing that I think is just perseverance. You can't give up. Dr. Thatte tried for 12 years. I am trying now for the last 14 years. If I were to quote Albert Einstein, what he said is, it's not that I'm so smart. It's just that I stay with problems longer. We just have to keep at it, persevere, and not give up soon. It is easier to give up. It's a lot harder to persist with something. I think with persistence, patience, perseverance, it can be done.
Anybody wants to start a clinical research unit in his or her institute, so can you guide something, or can you train something in your institute?
Yeah, so we do have training programs that we offer, and from all over India, many students come, and they used to come pre-COVID, and we hope to start it now. We do offer training programs in pharmacovigilance. These are 2-week training programs, how to get adverse reactions, how to fill it, how to put it into Vigilize, how to analyze the adverse reactions at one. We also offer training programs in therapeutic drug monitoring. So I know for a fact that many institutions in India have actually purchased an HPLC, but it's like a white elephant sitting somewhere in the corner, and then people don't have the expertise to use it. So we do offer training programs. I think one of the most important things in clinical research divisions that people, pharmacology departments everywhere, is that skilled manpower, which I talked about in the last slide. Manpower, technical, competence, and staying with that instrument or staying with the department to take it forward. That's something that people have to figure out how best to do that. Training programs like what we do will only take you so far. Ultimately, you have to build on that capacity and capability that you have built and sustain it the best you can.
Thank you.
Yes, sir.
Yes, Professor.
Madam, first I would like to congratulate you such an excellent session that you have made.
Thank you, Professor. So before I forget, I'm here only because of him.
It is not like that. We are very much thankful that you are over here, and moreover from your presentation is probably whole of the audience will accept it. The way of presentation is so excellent that we are literally overwhelmed. Thank you very much for accepting our invitation. You are having very, very busy schedule.
Thank you
I
so much.
have worked in the industry. I am into the clinical practice. I am in the academia also. There are probably 2 domains. One is clinical research lab. Another is clinical research division. So probably, maybe your opinion, I would like to know that initially we can begin, as you have already said, that manpower is important thing, and our colleagues who are from the clinical department, liaising with them is very, very important, because we are little bit dependent on them. And there are 2 categories of the people. If we are junior one, we can be taken as on the post of the investigator. And pharmaceutical companies are searching many places. I have worked in the pharmaceutical industry along with the Kiran, and we have literally searching. So in a medical college which is located like Kannauj in UP, far away, it will be very difficult to set up with the equipments. So initially what they can do, they can set up a particular room, make a board of clinical research division, get trained all of your people in good clinical practice training, in ethical training, and same training could be imparted to your clinical colleagues. I am quite sure that at least 50% of our department from clinical colleagues don't know probably much about good clinical practice, what are ethics, ethical guidelines. Train them, then pharmaceutical companies, and show them, approach them, pharmaceutical company, that we are having a established clinical research division, not lab. You can approach to us with particular clinical trials. They are searching for pediatric trials. It is quite common for many trials in the medicine department, gynae ops department. They are searching and they are looking for the trained manpower. It can be done, and funding is done by the pharmaceutical industry. So it is probably an easier approach, and when the work is going on, later on the equipments can be established, as you already told. So probably this is my opinion, it is the easier way.
Yeah, that's a good point. The other thing which I may just add to what you've actually said is, many of the clinicians, whether it is medicine or community medicine, gynec, at least in our institution, they want to do trials. I'm sure it's true everywhere, but they don't have the necessary support. So pharmacology departments offer support to clinicians. They themselves will grow in the process. I'm not sure about many of you, but I find it very, very hard to get funding from within the institution, right, to sustain ourselves. Applying to government also, I've made it sound easy. It's not easy even to get money from ICMR or DBT. It's a long drawn process. So one has to, you have to think in a multi-pronged approach, whether it's ethics, whether it's division, whether it's laboratory, people, manpower. You start small and eventually grow big. But you find a niche and then you sort of take it forward. But all your points are well taken. Yes, sir.
Ma'am, good morning. My name is Dr.
Good
Sushil
morning.
Sharma.
Yes.
I am in
Yeah.
AIMS Mangalore.
Yeah.
And as I've just said, we are just 3 years old. And I think your point of approaching the government agencies is very, very valid. Like in the last 3 years, we have approached DHR, we have approached DBT, and both the times we have been rejected. And right now we have put up a proposal to the ICMR.
Right.
So I think we plan to be at it till the time we get something. And your point about being at the right place at the right time. I remember reading somewhere, to be at the right place at the right time, you have to be in a lot of places at a lot of times.
Lot of times.
Only
Well
then you
said.
can be at the right place at the right time.
Well said.
My question, madam, here is, pharmacology department, I'm not very sure, but it straddles two places. Like, for example, we are not a laboratory sciences department.
Correct.
At the same time, we are totally not clinical also.
Correct.
So, so the manpower which you've got, so if we have to actually handle laboratory sciences, instruments, it becomes very, very difficult. So that is one limitation which I feel, like even in our MD curriculum, we are actually not trained hands-on on any of these equipments, as compared to people who have done biochemistry or microbiology or pathology. That is one place where I think we feel handicapped.
Yeah, sir, I thank you for bringing out the one point which I missed actually. When I pressed the last slide, I realized I'd forgotten to address that. So one of the things that we do, I have personally trained in HPLC. When we bought an HPLC, Dr. Shirsagar had signed a MOU with the company. So I had gone to Connecticut and I trained in HPLC. So when the HPLC breaks down, I know what is to be done. I don't work with the HPLC on a day-to-day basis. I think the point he's made is very valid. We have a master's and a PhD program. Let me just start with that. So we have MSc in applied biology. We have MSc in pharmacology. We have PhD in pharmacology, clinical pharmacology, and applied biology. So if you have masters and PhD students and you get recognized as a guide for those students will take your instruments forward. And like I said, you should offer niche services which are not offered by the biochemistry department. Right? So the biochemistry departments, at least in Mumbai, I can say, do not offer this therapeutic drug monitoring services. So we found a niche in that, and so much so that we have set up anti-epileptic drug estimations which are not offered in the private sector. like levetiracetam or lamotrigine or gabapentin. So you find something which will draw people to you. But having master's programs, PhD scholars with you, I think can help you take your instrumentation forward, sir.
Thank you, Dr. Nitya,
Thank you
for
so
an
much.
excellent session.
Thank you, ma'am, for that insightful session. I now request Dr. D.D. Gupta, Professor and Head IGIMS Shimla, to present a memento to Dr. Nitya Gokte as a token of our appreciation. Thank you, sir.
I request Dr. Sushil Sharma, Professor and Head, AIMS Mangalagiri, to kindly present the mementos to our esteemed chairpersons, Dr. R.K. Dikshit and Dr. Rajan P. Nerurkar, as a token of our appreciation.
Thank you, sir. Thank you. Medimation Education Private Limited was founded by Dr. Mansi Bhavkar and Maithili Bhavkar in 2013. The company, since then, has supplied visual educational content for more than 170 medical colleges around India. Medimation education is constantly evolving, and its ability to integrate technology in medical education has been vast. They have been the winner for the Economic Times Power of Ideas in 2017, also the winner of the National Entrepreneurship Award in 2018. We shall now have a video of Medimation Education Private Limited.
Understand by Medimation Education. According to NMC guidelines, pharmacology experiments on animals can be demonstrated by computer-aided education using computer-assisted modules. The three components of competency-based learning, as described by NMC, teach, perform, and assess, can be effectively conducted by our tutorial, practice, and exam modes respectively. Our current product list. We have a one-time purchase policy for all our products. Let us take a quick look at the features of each of them. Experimental Pharmacology Tutorial Mode is a series of 17 video modules that cover key topics in animal experiments of pharmacology. Each video vividly showcases the experimental procedure, including dissection of various animals. The experimental setup and the devices used are explained in detail. Accurate graphs and charts help students to understand the experiments and draw conclusions. A quick glimpse at the relevant theoretical concepts ensures an in-depth understanding of the topic. Next up, we have the Rabbit Eye interactive desktop application, which has a practice mode and an exam mode. In the practice mode, students can prepare the rabbit and perform experiments at different times. Students can measure the pupil size, perform light and corneal reflex tests, and understand the effects of the drugs. In the examination mode, students have to identify a randomly assigned drug based on its actions. The professors can view the students' answers and grade them accordingly. Lastly, we have effect of drugs on dog blood pressure, an interactive desktop application with practice mode and exam mode. Medimation Education presents Wonderstand Interactive Dog BP Software, a desktop application that runs without an internet connection. Learn about the actions of individual drugs on dog blood pressure and heart rate. Study phenomenon like tachyphylaxis or Dale's vasomotor reversal. An examination mode lets you test if students have understood the material. A simple admin login enables professors to get detailed student results easily. Our products have been designed keeping in mind the needs of Indian medical colleges. All our products have a one-time cost. They run offline without an internet connection or any complicated hardware requirements. We provide accurate and verified content along with high-quality photorealistic animations. Ideal for all medical institutes offering experimental pharmacology. And that's not all. Very soon, we have a host of new products coming up, all suggested by professors like you. Firstly, we have bioassay of guinea pig ileum as an interactive desktop application with an exam mode and a practice mode. This application will help students plot the dose-response curve, identify S1, S2, T, and arrive at the correct concentration of the test dose. An exam mode will test the students on their understanding. Then we have routes of drug administration, a tutorial mode which will cover the various routes of administration of drugs, their advantages and disadvantages, do's and don'ts, and also touch upon advances in drug delivery systems. Also in the pipeline are two interactive modules, Effect of Ions on Perfused Frog's Heart and Mammalian Experiments. Medimation Education prides itself on developing content that is suggested by professors for professors and their students. So, for any suggestions for new products or queries about our current products, please do get in touch with us. Thank you so much for your valuable time and attention.
We shall now break for tea. I request all of you to assemble here at 10:50 a.m. for the upcoming YPS talks. Thank you.
25 clinical trials and Principal Investigator, NIH Center of Excellence 2009 to 2014. He has authored many book chapters, received various prestigious grants, and has more than 185 publications, along with being a reviewer in several esteemed journals. We welcome you, sir.
Dr. Manjunath is Professor and Head, Department of Pharmacology, Sri Siddhartha Medical College, Tumkur, Karnataka. He is the President of Karnataka Medical Pharmacological Society, Convener of Medical Education Unit, Member of Academic Council and Scientific Review Committee and IQAC, along with being the Coordinator for NAC accreditation. We welcome you, sir.
We have with us our first speaker, Dr. Paul Mathai, Associate Professor, Department of Pharmacology, Malabar Medical College, Calicut, who will be talking about functional inhibitors of acid sphingomyelinases, an evolving concept.
Good morning. My name is Dr. Paul Mathai. I am an Associate Professor of Pharmacology in Malabar Medical College, Calicut, or Kozhikode, whatever way you want to say it, Kerala. I'm here today to talk to you something about functional inhibitors of acid sphingomyelinases. It's an evolving concept. So, in November 2019, the world as we knew it changed forever. The Chinese discovered some new disease. They didn't discover it, they were afflicted with some new disease. They didn't know what it was about, they had no idea. So in December 2019, they informed the WHO. And later on, in February 2020, this disease was given a name, COVID-19. The etiological organism was SARS-CoV-2. In March 2020, the WHO declared this a pandemic. And we know how the situation evolved from then to the point where we are right now. We know that vaccines have been developed with variable efficacy, but we also know that till date there is no drug, no effective therapeutic drug for this disease as of today. So in the face of a lethal pandemic of global proportions, there is no time to waste. We can't spend decades of time developing a new drug. We need to develop the drug quickly. We need to have data regarding the safety of this drug that we're investigating. And ideally, the investigated drug should reveal new targets and new pathways that can be exploited further. In this sort of situation, drug repurposing is the strategy that helps to achieve these targets quickly. So it's very easy to understand what drug repurposing does. We identify new indications for drugs that have been in the market previously for other non-related indications. Right. So as you can see from this slide, many drugs have been repurposed for COVID-19, and it all started with hydroxychloroquine. You all must remember the then President of the United States, Donald Trump, endorsing this drug very strongly, and under huge pressure, the US FDA approved it, gave emergency authorization for the use of hydroxychloroquine, but just a few months later, the authorization was withdrawn. Once it was quite clear that there was no evidence whatsoever to support this claim, they withdrew it. So many other drugs have been introduced, or repurposed rather, for COVID-19, but none with significant success. Okay, so now I'm going to move on to a very, very rudimentary and brief, I'll walk you through a very rudimentary overview of SARS-CoV-2 entry into a human cell. So there's a cell membrane, there's the intracellular portion, that's the extracellular portion, and that's the viral particle, SARS-CoV-2. We know that it attaches to its receptor, ACE2, with the help of its spike protein, and that's how it enters the cell. Now, we know that there are structural lipids. I don't know why this has come like this, but yeah, I think when sending it by email, something has gone. So we know that there are various structural lipids in the biological membrane, and one such structural lipid are sphingolipids, an example of which are sphingomyelins. And we know that sphingomyelin is hydrolyzed by a group of enzymes called sphingomyelinases. And the sphingomyelinase that I shall be talking about would be acid sphingomyelinase. So, sphingomyelinases hydrolyze sphingomyelin to ceramide and other products. And this is significant because, coming back to this diagram, there's the cell membrane, it's got sphingomyelin, ACE2, and acid sphingomyelinases are there, which is going to convert sphingomyelin to ceramide. It coalesces, ceramide coalesces to form physical structures that we call ceramide platforms. And ceramide platforms are capable of attracting various proteins, notably ACE2. And when that happens, there is a far greater likelihood for SARS-CoV-2's spike protein to attach to ACE2 and enter the cell, as you can see there. So the logic is you block acid sphingomyelinase with a drug and thereby inhibit the formation of ceramide platforms and therefore reduce the likelihood of the spike protein of SARS-CoV-2 to attach to ACE2 and enter the cell. That's the whole point of, well, that's one point. that functional inhibitors of acid sphingomyelinases can achieve, or phyasmas. There are plenty of phyasmas in the market. Most of them, or rather all of them at present, are already approved for other indications. So this is a drug which is being repurposed. The most commonly known phyasmas include dextromethorphan, maprotilin, nortriptyline, sertraline, and notably fluoxetine and fluvoxamine. These are drugs which are gaining a lot of attention for its potential use in the treatment of COVID-19 and other indications as well. But the question remains, is there sufficient evidence to support the use of fluoxetine or fluvoxamine or any phyasma for that matter in the treatment of COVID-19? I take this opportunity here to hammer this thing, which I feel is very, very important, not just in pharmacology, but any medical person. It's a philosophical razor. Extraordinary claims require extraordinary evidence. This should be the cornerstone for any medical man in this country or wherever he is or she is. At present, there is insufficient evidence for or against the use of these drugs in COVID-19. So what do? We generate evidence. And there are clinical trials going on a global scale whose answers or whose conclusions we await with abated breath. Right. So, if these drugs are found to be effective, if the evidence supports their use, this is important and significant because these drugs are freely available, their cost is low, their safety margins are high. These are very attractive targets, not just for COVID-19. Now, there are other potential uses of phyasmas. For example, as I told you, sphingomyelin gets converted to ceramide with the help of acid sphingomyelinases, and ceramide has been found to have apoptotic promotion and it arrests cell growth. So, if you can block acid sphingomyelinases, you could expect this drug to have anti-apoptotic and therefore neuroprotective effects, and this translates into potential use in various conditions such as Alzheimer's, Parkinson's, post-stroke, and these kind of diseases. So, to conclude, phyasmas are a group of drugs which have got serious potential. They are under serious research. They are under the microscope. And if the evidence supports their use, it's something that's worth looking into in the future. Thank you.
Yeah, the way you are presenting is excellent. I think you have been in the teaching profession since a long time. Looks like you are a veteran. Okay, so what is your experience about this? Is it you have done any studies regarding this, or it is purely a thought process?
Sir,
yes, it can be.
So there, among those 3 clinical trials which I showed, there is one called NAPTICON. So I am the principal investigator of that study in the institute where I work in. So that got me interested in the topic. It hasn't started yet, but it's about to start.
Okay.
So,
any questions from the audience?
Yeah, while questions from the audience comes, Dr. Paul, the question or the trial that you're doing, what are the sample size and what are the outcome you're looking at? I know, I mean,
okay,
COVID patients are very few, but
yeah,
and then what are the drug you're looking at? There's a list of drugs, right? You're doing fluoxetine or?
So the sample size is around 1,150. It's not a big sample size. It's small. And it's spread across 12 centers in India. The endpoint is basically SpO2. So the anti-COV is about whether we can prevent COVID-19 from progressing from mild to severe.
Okay.
That's the whole point
Right,
about.
got it. Thank you. Questions from the audience? Just keep it to a question, no comments, please. Yeah, give him the mic here, please. Just put up your hands when you have a question, then we'll reach the mic to you. Yeah.
So, this is only under research so far, and because a lot of this is a very common drug, fluoxetine, fluoxabine, a very common antidepressant drug. And any pilot study, you have any kind of, so that lot of the people, of the patient, they are taking this drug. Any protective effect during the COVID-19 on those patients? Any idea about that?
Sir, there are trials regarding fluvoxamine, not fluoxetine, fluvoxamine. And they have been shown that apparently the sample sizes were small, but apparently they do prevent COVID-19 from progressing to, they prevented intubation. They reduced the rates of endotracheal intubation. So that way they concluded that this might have some better property. And in addition to its ability to functionally inhibit acid sphingomyelinase, it also has SIR1 inhibition also. Some anti-inflammatory effect also it seems to have. So these are the two hypotheses that have been put forward in favor of fluoxetine.
All right, all right. You can continue the discussion at the break. Thank you for the question. Thank you, Dr. Paul.
Thank you, sir.
And hopefully at the next NAPTICON, you can present results of a few patients that you have.
I hope so, sir. Thank you.
I now
very much.
request Dr. Dennis Xavier to kindly hand over the certificate to our speaker, Dr. Paul Matai. Thank you, sir. The next speaker for this session is Dr. R. Lavanya, Assistant Professor, Department of Pharmacology, Sri Lalitambigai Medical College and Hospital, Chennai, whose talk is on the future of biomimetic co-formulation therapy, a boon to diabetics.
Good morning. It gives me an immense pleasure to present the YPS talk on the future of biomimetic co-formulation therapy, a boon to diabetics. As we all know that diabetics is the most important disease which is being prevailing in India, and India is the second largest country with diabetic population. So what would be the boon to diabetics? A new innovative therapy which would help the diabetic patients to survive their future. So my objectives will be diabetes introduction and types. And the introduction, as we all know, diabetes is a chronic metabolic disorder characterized by hyperglycemia, glycosuria, hyperlipidemia, and negative nitrogen balance due to defects in insulin secretion and or action. So, as I said earlier, globally, fourth leading cause of death is diabetes, and global prevalence is 422 million persons. So, as you all know, diabetes is divided into type 1 and type 2 diabetes. First picture, you can see the healthy pancreas secreting insulin, and there is glucose uptake through the insulin receptors. In the second picture, we see the type 1 diabetes, where is absolute deficiency of insulin due to autoimmune destruction of beta cells of pancreas. And the next type is type 2 diabetes, which is more common in India. And here we have insulin resistance in the liver and skeletal muscle, along with elevated levels of insulins. So what is amylin? Dual hormone secretion of beta cells of pancreas. All the time we speak about diabetes, we think about beta cells of pancreas and insulin. But we forget about the other hormone, amylin, which is also co-secreted along with insulin. So we can see amylin is also a hormone secreted by the beta cell along with the insulin, and it also helps in reducing the blood glucose levels. And as you all know, insulin action is decreasing the hepatic glucose output and increases the glucose uptake. We should also know about the amylin action. It increases the postprandial satiety, suppresses the postprandial glucagon secretion by alpha cells and suppresses gastric emptying. On the whole, it decreases the blood glucose levels along with insulin. So why do we need an amylin analog? In the first picture, you can see the healthy patient. Amylin and insulin is being secreted, and we have all the three actions: gastric emptying delayed, increased glucose uptake, and there is inhibition of glycogenolysis and gluconeogenesis due to the suppression of glucagon. Whereas when you give insulin for a patient, only the action of the insulin is that there is increased glucose uptake. There is no effective glucagon suppression. So why do we need is that amylin replacement, which also causes suppression of the glucagon and is critical to fully restore the metabolic signaling and constitute a true hormone replacement therapy which occurs physiologically in our body. So here we comes postprandial insulin analogs and amylin analogs commercially, which is available. Prandial and insulin analogs rapidly acting such as Lispro, Aspart, and amylin analog pramlintide. Of course, it's very costly and very few patients use it due to its cost. And both of them differ in their onset, duration of action, as well as both of them have the pharmacokinetics doesn't overlap. That is the major difference between the commercial preparations of prandial insulin and amylin analog. So this is why we are not able to give both the analog preparations together under a single syringe, and when patients or rarely patients need both the analogs and they are given in separate pumps or separate insulin syringes. So there is lack of overlap of insulin and amylin pharmacokinetics, and this hinders the synergistic effects which normally occurs synergistically in our body physiologically. So how does it is evolved? Insulin only was discovered initially, and then insulin and pramlintide, separate pumps, separate injections, and then a covalent pegylation, polyethylene glycolation was done. This extended the pharmacokinetics. There was some overlapping, but immunogenicity was more due to covalent pegylation. Then came the curcubutyryl enhanced stability by macrocyclic compounds like curcubutyryl, which causes more of pharmacokinetic overlap, decreased immunogenicity, and more stability. So that is why the advantages is that stabilization of the co-formulation. By adding the supramolecular curcubutyryl, we are able to stabilize the compound. Even pegylation helps you to stabilize a compound, and biocompatibility is being increased. By reducing the hydrodynamic size, we are able to increase the diffusion rate of both the compounds, insulin as well as pramlintide. The most advantage is that these two synergistic effect causes less hypoglycemia and more compliance. Here there are preclinical studies of both ultra-flat insulin pramlintide co-formulation. In the first picture you could see that there is no overlap of pharmacokinetics, whereas the co-formulation of Lispro and pramlintide has a overlap of pharmacokinetics, and this has been initially done in diabetic rats. Secondly, clinical studies was first tried in type 1 diabetic patients, and this also proved that it was well tolerated and reduced postprandial blood glucose compared with the separate preparation of insulin and pramlintide, and this is the study. So way forward, what are we going to do is that comprehensive assessment of the biocompatibility and immunogenicity of curcubutyryl polyethylene glycolation of insulin and pramlintide co-formulation. Thereby, in the coming decade, we can envision even more powerful scientific synergies at the interface of structural biology, molecular physiology, and therapeutics. Thereby, bringing both the drugs under one single syringe, we will be able to improve the compliance, decrease the side effects, and thereby it will be a great boon to the diabetic patients, and India is filled with diabetic patients in the current scenario. So this is the most advanced innovation which is being going on. Several clinical studies are also under the way, and let us hope for the brighter future with a less cost. So thank you for giving me an opportunity. Thank you.
Thank you, Dr. Lavanya. Any questions from the audience?
Sounds promising. But what is the data on the preliminary studies that were done? You showed the pharmacokinetics, but you said clinical studies are ongoing. What are the results from that?
In the results, they have proved that the co-formulation has reduced the prandial blood glucose levels. When compared to given separately, they have done 3 groups of studies, one receiving insulin only and one receiving...
Any of the studies done in India?
No, so far no studies have been done in India. This is the study being done in USA. So in future, probably India might do.
Sounds promising. Any questions from the audience? If not, thank you very much, Dr. Lavanya, for a nice, lucid presentation. And you can just stand by to receive your certificate. You can go, Dr. Manjunath.
I thank the NAPTICON for providing me this opportunity.
Thank you, Dr. Lavanya. I now request Dr. Manjunath to kindly hand over the certificate to Dr. Lavanya.
Thank you,
Thank
sir.
you, sir.
The next speaker for the session is Dr. Kirtana B., Assistant Professor, Department of Pharmacology, Government Kilpauk Medical College, Chennai, whose topic for today is PROTAC.
Good morning to one and all present here. Thank you for providing us this opportunity for the young speakers. So my topic for today is PROTAC. I'm Dr. Kirtana from Kilpauk Medical College, Chennai. So we'll go on with the contents. I'll talk about my introduction. What are the different parts of PROTAC? History, a little on ubiquitination and the mechanism, and how was it different from a traditional drug, how to develop a successful PROTAC, the clinical trials, and a few advantages and challenges which are faced regarding the PROTAC. So the expansion is, it is proteolysis targeting chimeras, and another name for this is also called as snipers. It is a specific non-genetic IAPase inhibitor of apoptotic protein based protein erasers. So finally what we need is that we have a protein which we are going to degrade it completely. So it is a promising approach which can help us in crossing the hurdles with present drug discovery. So the parts of the PROTAC. It is a hetero-bifunctional molecule where it has 3 parts. One part is a molecule or the protein which is the target of interest, and the other one, the other end, it has E3 ligase which will be connected through a linker which connects both the moieties. So one end is a protein molecule, the other end is an E3 ligase, and in between is a in between is a linker. So this is not only for the protein molecules. When it is seen with other drugs, say in case other endosomes, if it is an endosome, it is called as LITAC. When it is going to target an RNase, it is also called as RiboTac. So similarly, the names differ. So just a basic note on what is a proteome. And we know that nearly about 20,000 proteomes are seen, and only 3% of it has a proper drug which can target and degrade or inhibit a protein. So the yet 97% is still unexplored, and that can be targeted, or the resistance can be overcome with this type of PROTAC molecule. A little about the history. It started nearly 20 years back. The thing is that it was mostly a proof of concept title, but it became clinically important only in the past 2 or 3 years, where new molecules have been started developing. So PROTAC started to roar, or in the starting of 2000s, it gained more importance when ubiquitin degrading protein, which was one Nobel Prize by Aaron Avram and Irvin Rose. When these people got the Nobel Prize for ubiquitin degraded, only this PROTAC started having more importance. A little about ubiquitin is it's a very small molecule, and it participates in both synthesis and also destruction of the defective protein. So what happens here is that this PROTAC molecule will help in ubiquitination. So finally, a 26S proteasome will be formed and the molecule will be degraded. So the enzymes involved are nearly E1, E2 and E3, ubiquitin activating enzyme, a binding enzyme, and finally the E3 ligase, which leads to formation of a ternary complex, and the ternary complex will lead to formation of 26S proteasome. Finally, the protein which we are going to target will be degraded. So the mechanism is mainly based on the ubiquitin protease system, which will target specific protein, and finally degradation happens. So what all the target proteins can be? It can be a nuclear receptor, it can be a protein kinase, any type of translation pathway, anything can be there. And in case of E3 ubiquitin ligases, mostly the ligases involved are VHL, MDM2 and the cereblon. And finally, the PROTAC molecule will be a ligand which will connect both the molecules, a linker molecule. So how it happens is that it first enters the cell, it combines with the protein of interest, and then the E3 ligase, ternary complex, the ubiquitination will happen where all the three molecules of ubiquitin will join together. This will trigger the degradation process, and the final PROTAC molecule, after the degradation started to happen, the linker of the PROTAC molecule which we have introduced will again enter the cycle. So it doesn't degrade itself. It will be there again and again. So just one small molecule is enough to degrade the whole of the protein. We need not give more dosage. So how is it different from a traditional drug? Always an active site is needed for a traditional drug. We just here needs an active site is not needed. Just the protein which we want to degrade is needed. And it is a transient and less durable one. Here it is sustained degradation. As I said, it will enter multiple cycles. It doesn't degrade in one cycle. There we need high drug exposure. The amount of drug due to resistance, everything, we need to increase the dose, but it is not necessary here. Stronger binding is needed for a normal drug. Here even it need not bind, it just want to be there, it has to be present. It targets mostly one function. Here it targets the protein as a whole. And it will only inhibit a protein subunit. Here it degrades the subunit, whole of the protein. So why it can have or it can gain importance is that here it can be sensitive to drug resistant targets. Already we have drugs. When resistance develop, this can have a drug resistant target, and it will be better selectivity than the classical inhibitors. Here chemical knockdown happens with a different speed. And unlike other gene editing tools like say some CRISPR or something, where genetic component will be involved. Here even, as I said in the sniper word, it is a non-genetic inhibitor of the protein erases. So even if there is some genetic polymorphism also, the final protein, it can be degraded invariably. So where the targets can be, when it is an undruggable genome, when resistance is more, and when a lot of scaffolding is there, this can be a target for the PROTAC molecules. So how to develop this one? What we need to know that you'll have a protein, you should know whether this can be degraded by a proteasome or whether it can be ubiquitinated. Only that is needed. You need to know whether the permeability and affinity of the proteome, the PROTAC is sufficient for the target, and whether the target can be, whether it will lead to formation of the ternary complex. Finally, the degradation will happen. So few trials which shows that Arvinas, the drug has the name ARV-110. It is there in phase 2, and the same company has another molecule which is Arvinas 471, which will degrade. The first one is for the androgen receptor. It is used in treatment of prostate cancer, and the second one is for the estrogen receptor in the breast cancer. These two molecules are as of now in phase 2 trial, and they are oral molecules. The others also in development stage. As I said that ARV-110, it is known as bavdegalutamide. It is in phase 2 stage. So advantage of PROTAC, expected to overcome drug resistance, it will improve the selectivity, and it has a huge potential to develop in even for undruggable targets. It has a curative effect which will last for a longer period of time. So challenges, it is actually in a budding state. So more efforts are needed to have an insight on the efficacy and safety of the PROTAC, which will be used. And also more target binders. Usually only the E3 ligases like VHL, the cereblon, MDM2, only these three are used. So other E3 ligases also need to be found. It is an expensive one, and proper screening technology is not available for the PROTAC even if it is developed. So take home message is that it has two parts. One is a ligand for the target protein, the other is an E3 ligase. The linker which joins is called as a protein. So thank you.
Okay, good talk, Dr. Kirtana. Any questions from the audience? Maybe while audience is thinking of questions, among the list of clinical trials that you showed, of course you didn't have time to present anything, what is showing promise?
The ARV-110 molecule, sir. It is for...
In what condition could we use that?
Come again.
In
It
what
is for
condition?
prostate cancer, sir.
Prostate cancer.
The name is bavdegalutamide, similar to bicalutamide. And that one has come even till phase 2.
Right. Audience, please be involved, ask questions. Okay, maybe then I should ask the next question as well. So do you think this approach will get us more newer medications quicker?
Yes, sir. Being a small molecule and we can have more trials. So we can expect more number of agents if it is successful, sir.
All right. Maybe the next session, if the audience don't ask questions, we'll ask them questions, correct? Okay, please be involved and, yeah. All right, so thank you, Dr. Kirtana. Please stay on to receive your certificate.
Yeah, I now request Dr. Dennis Xavier to kindly hand over the certificate to our speaker, Dr. Kirtana.
Thank you, sir. The next speaker for the session is Dr. Anuj Chandran, Assistant Professor, Department of Pharmacology, Malabar Medical College and Research Center, Calicut, whose talk is on myosin modulator, MAVA Campton.
You want hand mic for this? Hand mic.
This is spelling mistake. I want to correct it also.
Good morning, everyone. I'm Dr. Anu. I'm from Malabar Medical College, Kerala. Now, today I'm going to talk about a drug that is myosin modulator, MAVA Campton. And I'll start my talk with a study that is an Explorer HCM study. It is a randomized, double-blinded, placebo-controlled phase 3 trial. In this trial, more than 18 years of age patients were taken, and the patients with hypertrophic obstructive cardiomyopathy and NYHA class 2-3 symptoms were enrolled in the study. Total 251 patients were taken into the study, and we have one control group and we have test group. So in the test group, our drug, that is MAVA Campton 5 mg, was given daily along with the conservative therapy, that is beta blocker and calcium channel blocker. So total 123 patients were put on that group, and on the next group we have placebo along with the previous therapy, that is beta blocker and calcium channel blocker. So this was done for hypertrophic obstructive cardiomyopathy patients who were having NYHA symptoms, class 2 and 3 symptoms. Okay. Now, this duration of the study was 30 weeks. After, this is a multi-centric study, and it was done in 68 clinical cardiovascular centers in 13 different countries. Okay. Now, following 30 weeks, after 30 weeks, we have two endpoints. We have primary endpoint and we have secondary endpoints. So, the primary endpoint was more than 1.5 ml per kg per minute increase in peak VO2, peak oxygen consumption with more than 1 NYHA class improvement or more than 30 ml per kg per minute increase in peak VO2 with no worsening of NYHA. That was the primary endpoint, and 37% of the patients who were put on the mavacamten group, they attained that endpoint. Then you have next secondary endpoints, which were LVOT gradient, PVO2, and more than 1 NYHA class improvement, and KCC questionnaire. So these were the secondary endpoints, and it also came to the conclusion that mavacamten was found to be superior than placebo. So this is the interpretation of the study. Among the patients with HOCM, mavacamten was superior to placebo, and this medicine, that is mavacamten, has improved the functional capacity and health status, and the mavacamten was well tolerated with no significant long-term side effects. So here I'll tell what is the side effect. The most commonly seen side effect of this drug was dizziness and fainting. That was seen, and also some of the patients experienced allergic reactions, and 10% of the patient experienced secondary cardiac failure. That was a long-term side effect. So that is the interpretation of Explorer HCM study of mavacamten. Now, 28th April 2022, very recently, this drug was approved for symptomatic New York Heart Association class 2-3 obstructive hypertrophic cardiomyopathy to improve the exercise capacity and symptoms. So this is why it was, this was the condition for which this drug was approved, and this is available in the market with the brand name Camzyos, and it is available as 2.5 mg, 5 mg, 10 mg and 15 mg tab doses, and the current recommended starting dose is 5 mg. 5 mg is the recommended starting dose, and it is developed by Bristol Myers Squibb. And one more thing I want to say is it is marketed with a boxed warning for the risk of cardiac failure. Now, I'll explain the mechanism of mavacamten, and so I'll just explain the normal physiology. We all know we have heart has got atrium, we have ventricle. So in the right side, right atrium, from there the blood passes to the ventricle, and from there it goes to the pulmonary arteries, and on this side you have the left atrium, left ventricle, and the blood is going to the aorta. We are all familiar with this. Now let's see what is happening in HCM. Patients with HOCM, what happens is the ventricle becomes thickened. There is thickening of ventricle, it becomes very thick, and what happens to the size of the ventricle? The lumen becomes very narrow, and because there is thickening and the more stiffening of the ventricles, the blood is not able to be pumped out properly from the heart. So it might block the blood flow out of the heart. This is what is happening in HOCM. Now, these cardiocytes become hypertrophied, and along with that, there will also be fibrosis of the cardiac muscle. Now, I'm going into the molecular level. There is a picture of cardiac myocyte here. We know that cardiac myocytes have got voltage-sensitive calcium channel. Now, I'm going a little more detail into the cardiac myocyte. So here, I've drawn a gap junction between two cardiac myocytes. They are just the membrane channels through which ions pass. So if you see there, when the action potential reaches the gap junction, it moves along the membrane, and then it reaches the T-tubules of the cardiac myocyte, and from there it reaches the voltage-sensitive calcium channel, and this will trigger the entry of calcium into the cardiac myocyte. And this entry of calcium will trigger the movement of the calcium from the sarcoplasmic reticulum. So when there is too much calcium in the cardiac myocyte, what happens to the sarcomere? It will become shortened, and this is how the cardiac myocyte will contract. Now I'm going into little more detail into the sarcomere. So we know sarcomere has got thick filament and thin filament. So I have drawn myosin here, myosin with myosin head, and then we have actin on both sides. So actin has got tropomyosin in between the actin, which is covering the actin binding head of the myosin binding head of the actin. I'll just stop with the mechanism. That is this mavacamten. What it is doing is it is decreasing the binding between actin and myosin. What happens in HOCM is there is too much contraction. When there is contraction, there will be binding of actin and myosin. Too much binding of actin and myosin. See, I have drawn too much connection between actin and myosin. So patient will experience shortness of breath, chest pain, palpitation and all that. So if you give mavacamten, mavacamten is a selective, allosteric and reversible inhibitor of cardiac myocytes. So if you are giving this drug to the patient, the connection between actin and myosin decreases. So when there is less connection between actin and myosin, what happens? There will be relaxation of the cardiac myocyte, and so the heart will be able to pump properly. That is a mechanism of mavacamten, and there are other studies like the Valor HCM study that has also come to the conclusion that if the patient is prescribed mavacamten, the current therapy that is the septal myomectomy and alcohol ablation therapy, the need for these procedures can be decreased if the mavacamten is prescribed to the patient. So that is about mavacamten. Thank you all. Thank you all for listening. Thank the organizers for giving me this chance.
Thank you, Dr. Anu. Any questions from the audience? Yeah, there is one question.
Effects that they had reported.
Adverse effects were fainting, dizziness.
What were the rates and how bad was it? Was it bad enough to stop the drug?
No.
Okay.
It was not bad enough.
All right, thank you. Just stand by to get your certificate. Go ahead, Dr. Manjula.
Thank you, Dr. Anu. I now request Dr. Manjunath to kindly hand over the certificate. Thank you, sir.
The next speaker for this session is Dr. Sneha, Assistant Professor, Department of Pharmacology, MS Ramaya Medical College, Bangalore, whose talk is on marine pharmacology.
A very good morning to one and all present here. I'm Dr. Sneha from MS Ramaya Medical College. So the next 8 minutes, you would be listening a talk on marine pharmacology. So I'm sure many of you would be wondering why has she chosen marine pharmacology when there are a lot of recent advances going on, a lot of new molecules coming up, and why marine pharmacology? So I'm reminded of my ancestors saying that salt water is the cure for has the cure for every disease. It may be in the form of sweat, or it may be in the form of tears, or it may be the seawater. So when I read between the lines, I realize that what they are trying to say is sea or ocean is has a variety of diverse organisms. And India has a very rich coastline which stretches to around 8 kilometers, and there are a lot of biodiverse organisms, maybe ranging from it may be the plants or the animals, it may be the seaweeds, it may be the different types of fishes, sponges, mollusks, tunicates, or a lot of organisms are there, and they are they vary from region to region, from one coastal region to the other coastal region. So these bioorganisms release a lot of bioactive amines or polysaccharides or carbohydrates, either for their self-defense from the predators, or for their food habits or reproductive habits, or for their survival in the extreme environment, like for protection from the extreme sunlight or the extreme oxygen variation conditions. So for these, they secrete a lot of biological active amines. And these amines have pharmacological potential actually. let's look into it with this brief introduction. I go on to the definition of marine pharmacology, which is a branch of pharmacognosy, which focuses on the substances with active pharmacological properties which may be present in the marine species. It may be a plant or an animal. So this biodiversity of marine environment ranges from the sponges, coelentrates, opisthotonal mollusks, echinodromes, bryozoans, marine algae, and a lot of marine microorganisms. To begin with the evolution. So Bergman and his company in the 1950, as early as 1950, identified spongiocin as a nucleoside. So from then, the journey began. And in 1969, we had cytarabine, which we still use in therapy in treatment of leukemias. And then there were discovery of various cytotoxic properties of various turbinata tunicates species. And in 2004, there was ziconotide, which is an analgesic drug in chronic pain. And of course, we have the omega fatty acid. I know the role is controversial, the dose and the type, the subtype of the omega fatty acid. The controversy is always there. And then the next decade, between 2010 and 2020, lot of new molecules came into the market, mainly which focuses on the cancer chemotherapy or the antiviral drugs. So this is the classification. I would deal the drugs in detail in the next few slides. Coming to the toxins, the tetrodotoxin or the saxitoxin, conotoxin. So they are from the pufferfish or from the snails. They mainly used in the experimental studies to induce neurotoxic damages in the experimental tissue. Then there are dyes which are obtained from the red algae, that is phycoerythrin, which stains red color to the tissue, which is used in the histopathological studies. We have aquarin and the green fluorescent protein. These are actually from the jellyfishes. So they have self-illuminance property. These dyes which are secreted have a self-illuminance property. So they are used in fluoroimmunescence assay studies. Next, we have the enzymes like Wen DNA polymerase. So the advantage of this Wen DNA polymerase from the conventional DNA polymerase is that it can sustain high temperatures. So as high as 95 degrees, which is done as a priming before the PCR chain reaction begins. And the source of this enzyme is a thermococcus. Next, we have the nutraceuticals like the fatty acid, doxahexanoic acid, that is the DHA. So there are different types of omega fatty acids in therapy. It might be the ethyl esters or the carboxylic acid moiety, or it can be a enolic ethyl ester omega fatty acid. Then we have the mucoadhesive polymers like the chitosan and the alginic acid. So chitosan is actually the exoskeleton of the crustaceans and the arthropods. It is a fibrous tissue which confers the stability. So all these agents are being tried as an excipient to confer the stability to the drug formulation. Next, alginic acid, it provides the jelly-like consistency, so it's already there in therapy. In the treatment of GERD, where there are formulations which contain this algic acid. The advantage is that it gives a jelly-like consistency and it forms a protective barrier in the GERD. So as I told, let's look into the drugs, anti-tumor and the antiviral drugs. All these are already approved and there in the market. Cytarabine is a pyrimidine analog, so it is used in AML and CML. Vidarabine is a antiviral drug. Ziconotide mainly acts on the calcium, voltage-gated calcium channel in the neurons, and it is helpful in chronic pain. Panobinostat is been a newer drug in the treatment of multiple myeloma. Lubrinectin and trabectedin are the alkaloids which are there, and these are the origin of these are from the tunicates. There are a few drugs in the pipeline, that is litidepsin, tunicate in origin. So it has been tried in COVID-19. It is undergoing a phase 3 trial. The rationale here is it prevents the viral replication. Tetrodotoxin is from the pufferfish. So this is a toxin which is released from the ovaries and the liver of the pufferfish. So it actually blocks the voltage-gated sodium channel. So it is being tried in neuropathic pain. Marizomib is another ubiquitin proteasome inhibitor. So it might be an alternative to the bortezomib if the trials prove safety and efficacy, because it is more potent and selective when compared to bortezomib. We have the mucoadhesive polymers. So the advantage of this marine origin mucoadhesive polymers is that they have strong interchain bridges which confer stability to the molecule when added as an excipient. And then we also have polymers which are already used in dentistry in the bioadhesive tablets and the gels and the paste dosage forms. So mainly it is the chitosan and alginic acid which has been used in this mucoadhesive polymer films. So to summarize, so ocean pharmacology, the marine pharmacology is a sunrise industry. So the postgraduates here, don't be surprised if you get a question on this marine pharmacology in your final exams. So yeah, we need to focus on the development of drugs for the marine sources. So I conclude my presentation. I thank NAPTICON a lot for this wonderful opportunity for the budding pharmacologists, and my HOD, Dr. Anuradha ma'am, who's always been a constant support for me. May that be official or a non-official thing, she's always there as a backbone to provide solution. Thank you, ma'am.
Good. Thank you, Dr. Sneha. Questions from the audience? So, Dr. Sneha, would you recommend in our new CBME curriculum that PG students also undergo training in scuba diving?
So I have no recommendations, but MCI has come up with an idea to tag with the regional institution and develop a course.
No. I mean, really good talk in such a short time. You were able to cover so many things. Any questions or any experience?
Thank you, sir.
From people? All right. Okay, so please stand by for your certificate.
Thank you, sir. I now request Dr. Dennis Xavier to kindly hand over the certificate to our speaker, Dr. Sneha. Thank you, sir. We have a small announcement by Dr. Kamal.
Good morning, esteemed pharmacology colleagues. This is an open announcement for postgraduate pharmacology students. Postgraduate pharmacology students, those who are listening to my voice, please come near the dais. After 15 minutes, we will be having an open interaction session with the Honorable President, Dr. Padamja Uday Kumar, with the respected Secretary, Sir, Dr. R.K. Dikshit, and myself, Dr. C.M. Kamal, National Coordinator of the National Association of Pharmacology and Therapeutics. And if you are listening since yesterday from the speakers, from us, we want to listen from your mouth. What are your various suggestions? What are your various expectations from NPT? We will be listening and we will be trying to work them as strongly as possible. Thank you very much. Please open up and participate in this very, very important session. Again, this is an announcement that NPT have been created keeping in view the expectation of the young generation of pharmacologists. We are here for you. You are our future. All the efforts are dedicated to you. Please give a big hand to the future of pharmacology. Thank you.
Thank you, sir. The next speaker for the session is Dr. Abdul Rashid Razia from Melmaravatar Adiparashakti Institute of Medical Sciences, whose talk is on dual orexin receptor antagonist.
Good morning, everyone. Thank you, NAPTICON, for giving me this wonderful opportunity. So today, my topic is about dual orexin receptor antagonist, shortly called as DORA. As you can see, you want to sleep, trying to sleep, you can't sleep, call DORA. At the end of this session, hope you will understand why this is quoted. Now, can you please tell me how many of you have suffered from insomnia? At least you have gone once a day for a tablet. And can you just name the drug, what you have taken? Any one of you? Yes? Yes, sir, what you have taken? Okay, sir. So the first, usually we go in for benzodiazepines or other OTC drugs. Now there is a change. What is the change? So FDA recently approves daridorexant, that is oral tablet for insomnia. So moving on to the definition of insomnia by DSM, it is difficulty in initiating or maintaining sleep, frequent awakenings, and you have even problems in returning to sleep after awakening. And what is the prevalence? At least 30% of the general population, they suffer from insomnia, which could be due to stress or irregular sleep schedule, poor sleeping habits, mental health disorders, of course, anxiety, depression, physical illness, pain, especially cancer patients, and few medications, that is those who are on SSRIs, especially fluoxetine. Okay, and we all know this neurotransmitters involved in sleep promoting and wakefulness. At the last, you can see the peptide, that is hypocretin or oroxin. The rest are acetylcholine, dopamine, norepinephrine, serotonin, and histamine. So why this has to be treated? So why do you think this insomnia has to be treated? Because it affects your thinking ability, and it can cause some neurological dysfunctions like mood swings or hallucination, and even this has a higher risk of developing obesity, diabetes, and cardiovascular disease. And what are the recent drugs we have for insomnia? As Sir said, is it drugs that is non-benzodiazepine or benzodiazepine groups, and you have melatonin agonists, heterocyclic compounds, anticonvulsants, OTC, that is over-the-counter drugs, mostly here antihistamines. So why this benzodiazepines or others, it should not be, why it should not be preferred? Definitely they given for residual sedation and memory and performance impairment, falls, and undesired behaviors during sleep, and you have somatic symptoms like malaise, fatigue, and drug interactions, especially with the barbitons. Why? Now, why DORA? That is, they reduce the sleep onset latency. That is the time period taken to fall asleep, and they increase the total sleep time, that is at least 7 hours, and they increase sleep continuity and sleep efficiency or sleep quality of time. So what do they do? This is oroxin, it is a precursor of pre-pro-oroxin, okay, and this binds with OXR1, OXR2. This oroxin A, it binds with both, whereas oroxin B, it binds with only OXR2, okay, which is located almost in all the hypothalamus, locus coeruleus, amygdala, which is involved in the neuronal pathway. So this basically acts by GQ pathway, and also there can be a biased signaling, that is alternate beta-arrestin pathway. So what are the residual actions? It is clear that they bind with either OXR1A or B or the both. Okay. The residual action is it will increase the intracellular calcium, which leads to the activation of sodium calcium exchanger. When it binds to both, there will be increased expression of NMDA receptors and inactivation of G-couple inwardly rectifying potassium channels. Now what are the DORAs? They are almorexant, filorexant, lemborexant, suvorexant, and daridorexant. What does this preclinical studies say? They are tried in rat mice. They have proven that they have decreased the wakefulness and they have increased non-REM REM sleep, and the cataplexy, it is not accounted. The same with all the drugs. In daridorexant, again the cataplexy, it is not reacted to it. So coming on to the clinical studies in human, which is REST-ORA, restore your normal physiological sleep with the oroxin receptor antagonist. That is, Swiss pharmaceutical company tried this with almorexant phase 3. And why this DORAs can be used? They have high genetic conversion, less serious side effects, and they have the newer mechanism of action. And the results, that is, this almorexant, after the clinical trials and studies, it has been abandoned because it has led to the elevated abnormal liver enzymes, and this filorexant, it was also discontinued, again leading to increase in creatine phosphokinase. And the reason for using daridorexant is, you can see the T-half is 8 hours, whereas in lemborexant, it is 17 to 19 hours, and suvorexant 12 hours. So the adverse effects are headaches, somnolence, sleep paralysis, hypnagogic or hypnopompic, which means falling asleep or waking from sleep, and like cataplexy-like symptoms. And where we should not use this, that is in patients with narcolepsy and the strong drug interaction with CYP3A4 inhibitors. And definitely when we recommend this, we have to give a medication guide, that is 30 minutes before going to bed, and they have to stay in bed at least for 7 hours, and the effect is delayed if it is taken soon after the meal, and patients has to be warned about CNS depressant effect and daytime impairment. What are the limitations? This antagonist, it has not been tried in comorbid conditions like liver and renal impairment, and the agonist, they have not entered any clinical trials. Again, this needs further exploration in substance abuse and reward pathways. And what are the future targets? This can be tried in other sleep-related disorders, mood disorders, addiction, neurodegenerative diseases like Alzheimer's, inflammatory condition, obesity, colon cancer, and apoptosis. These are my references. So it's time to sleep with Dora. Thank you.
Questions from the audience? While the mind teaches you, Dora is actually an explorer, right? Who never sleeps. Dora the cartoon.
Yeah. But Dora is very active asset.
You mentioned that in DORA drugs there is no residual effect on the next day, but half-life is, I think the minimum half-life is 8 hours.
8 hours.
And when we compare the jet drugs, zolpidem 2.8, 3 hours. So how is it better than the jet drugs?
That
because
is the residual effects, daytime
a residual effect, but half-life is 8 hours. There should be residual effect on the next day.
And also,
DORA drugs.
the reason is the sleep latency. You can fall asleep as early as possible.
Okay.
And the sleep quality is also increased, and definitely 7 hours you can sleep.
Okay, sorry, same thing, just to clarify, is there a head-to-head study comparing?
Sorry, sir?
Is there a head-to-head study with zolpidem? Quality of sleep and adverse effects, because that list of adverse effects looks pretty bad to me. No head-to-head studies.
No.
Yeah.
And
they should be coming soon.
Second, is there any effect on anxiety? Can we take an anti-anxiety drug in low doses?
Still in the pipeline.
Okay, thank you.
Thanks.
All right.
Is DORA available in India, and what about the price like?
India, I'm not sure, sir. But FDA is approved in January. US, it is in the market.
Okay, I think India still we don't have availability in the market. But even if it is available, it should be very expensive. So that is another factor we need to consider actually. So cost-wise, how effective it will be. Anyway, thank you for the lovely presentation.
Yeah, that was good. So thank you very much, Dr. Razia. So just stand by for your certificate.
Thank you, Doctor. Thank you, Dr. Razi. I now request Dr. Manjunath to kindly hand over the certificate to Dr. Razia.
We have now come to the end of this YPS session. I once again thank the speakers for their brilliant presentation and our honored chairpersons for their valuable time. I now request Dr. Sainath Iyer, an accomplished pharmacologist with over 50 years of experience, to kindly present the mementos to our chairpersons, Dr. Dennis Xavier and Dr. Manjunath. I request Dr. Sainath Iyer to kindly hand over the mementos to our esteemed chairpersons. Dr. Sainath Iyer is one of the most senior accomplished pharmacologists with over 50 years of experience. We are very proud to have him today. Please give him a big round of applause. I request our beloved HOD, Dr. Padmaja Uday Kumar, to kindly hand over the memento to Dr. Sainath.
Yes, he is a retired pharmacologist, and just one WhatsApp message from me that we are conducting a conference, he immediately registered and is here. Thank you, sir, for your...
I now request Dr. D.D. Gupta to kindly hand over the memento to our beloved chairperson, Dr. Dennis. Thank you, sir.
Thank you, Dr. Dennis and Dr. Manjunath. As we express our gratitude, we must never forget the highest appreciation is not to utter words, but to live by them. With these words, I now request Dr. Padmaja Uday Kumar to kindly give a memento of recognition to the Governing Council members.
Those MD students who are outside.
Those MD students who are outside?
They will not be awarded certificate. Please come in. Please come. We will take attendance of MD students. And then only we'll distribute certificate. Please. Faculty is okay.
Faculty are also invited. They need to hear what are the expectations. Faculty members are also invited as well. So we will be giving chance to speak to postgraduates, but faculty members are also requested to listen the expectations of the future of the pharmacology. Okay, all the faculty members also please come inside. Can we start? So, my competent and esteemed future of pharmacologist. I feel the clap should be loud. This is for you. This is for the future of pharmacology. Please let's hear the word that pharmacologists are also having a lot of energy. We can show to them. Please. So,
just to boost you, just say pharmacology in slow tone. Just say pharma. Pharma means a mother of every specialty is pharmacology. Pharma. Pharma is mother of the every specialty. So tell pharmacology. Okay.
Thank you very much. I will be having some idea, taking some idea from which part of the postgraduate studies. I was having a look on the list that Madam Honorable President has provided to me, and I am very proud to say that I have prepared a map of the country, and from each and every part of this country, postgraduate student, pharmacologists, our colleagues are participating in this conference. It is really a matter of pride for all of us. From which medical college you are?
State GS Medical College, Mumbai.
State.
Maharashtra, sir.
Thank you. Your good name, your state?
I am Dr. Sarang. I am from Mumbai, Topiwala National Medical College.
Okay. Which state?
I am Dr. Shakti Prasad Sodangi from Bhimsar, Bulla, Odisha.
Which state?
Odisha.
Odisha. So my young postgraduate colleagues, before I can open up the other sessions, with your kind permission, only with your kind permission, if you give me the permission, we are having certain certificate or mementos of honor. If you allow me, we can also, it will take nearly 5 minutes. If you allow me, we can also finish that. And then we will be free of mind, we will be able to listen to you. Can you allow us? Thank you. I feel Dr. Nicole can take. Quickly distribute all these mementos and certificates, please. These certificates and mementos are for the efforts of the organizing committee also. They are putting such a hard work day and night for past 1 month. Of course, some of the mementos are for the governing council members who are putting efforts behind the scene. This is also to motivate that today they are sitting, I am standing, and tomorrow you will be standing over here, you will be sitting there. This is just to motivate you all also. Okay? So with your kind permission of all, I will be handing over this mic to Dr. Nicole Pereira to quickly distribute all these mementos and
Thank you very much. Thank you very much, sir. With these words, I would now request Dr. Padmaja Uday Kumar, the President of NPT and the Organizing Secretary of NAPTICON 2022, to kindly give a memento of recognition to the Governing Council members. Dr. C.M. Kamal. Can we have a big round of applause? Dr. R.K. Dixit. Can we please
further
give sir a big round of applause again?
I am devoting all these certificates, all these token of mementos to all my future pharmacologists. Thank you. I dedicate you with honor and pride. Thank you very much.
Dr. R.K. Dixit. Dr. S.P. Daneria. Dr. P.K. Sharma.
She is the student. Due to unavoidable circumstances, he could not join us.
Yes. Thank you. Major Dr. Jitendra Singh. Kindly give sir a big round of applause. Thank you, sir. Dr. F.A. Khan, I kindly request a representative to kindly collect it on behalf of Dr. F.A. Khan. Thank you, sir. Thank you, sir. On behalf of NAPTICON, we'd also like to thank Dr. Ratnakar, who's our KMC observer, and I now request Dr. Padmaja Uday Kumar to kindly give a memento of appreciation to sir.
Thank you, ma'am. With these words, I kindly request the founder chairperson, Dr. C.M. Kamal, to kindly felicitate the organizing team. Dr. Nicole Pereira, Joint Organizing Secretary. Dr. Chandralekha N., Joint Organizing Secretary. Dr. Skandashri K., Chairperson, Scientific Session. Dr. Vinita K., Co-Chairperson, Scientific Session. Dr. Mangala S., Finance Secretary. Praveen from A1 Logics. I kindly request a representative from A1 Logics to come forward. A representative from A1 Logics to please come forward to collect.
Thank you, sir. With this, I now request our National Coordinator, Dr. C.M. Kamal, to kindly give a token of appreciation to our the wind behind the wings of NAPTICON 2022, Dr. Padmaja Uday Kumar. Please give her a big round of applause, everyone. Ma'am. Thank you.
Now, before I can proceed, I wanted to make an important announcement. Since I am listening for past 2 days, that you have all witnessed a huge success of NAPTICON. It will be, you are the better people who can judge the success of the NAPTICON. Are we successful? Are we successful? I am not able to hear any voice. Thank you all. So, many messages from across the country, from Kashmir to Kanyakumari, from Himachal to Arunachal Pradesh, Gujarat to Arunachal Pradesh, messages are coming, where is next? So, I wanted to announce it very gracefully, and I wanted to extend my thanks to the next organizer from the core of my heart, from the entire team of the NPT, those who have gracefully accepted, and based on the performance of this team of NAPTICON under the able guidance of our Honorable President, Madam Padamja Uday Kumar, and respected President Dr. R.K. Dixit. Many are attracted and many have volunteered. In fact, we have to choose one. We are gracefully announces that next NAPTICON is going to be announced in the city of Nawabs, Lucknow. And fortunately, he is a source of inspiration for young generation of pharmacologists, including me. I would like gracefully to invite the organizing chairperson of next NAPTICON, Dr. Amod Kumar Sachan, on the dais. And I would like to request our President Madam and Secretary Sir. See, there is a tradition in the country that whenever a responsible thing is given, responsible post, responsible work is given, we are giving a pagadi, a cap on the head. So I would like to request Madam President and our respected General Secretary Dr. R.K. Dixit to give him the honor. This is the seat of responsibility, and he has gracefully accepted. He will say some few words about the NAPTICON, about Lucknow also. I would like to request, sir, that please complete your speech within 2 minutes. Thank you.
आप नवाबों के शहर में आने वाले हैं और इतनी जल्दी, वहाँ तो इतनी जल्दी में तो लोग ट्रेन भी नहीं पकड़ पाते हैं। पहले आप में ही 2 मिनट निकल जाते हैं। I am really delighted to accept this opportunity to really serve you in our city of Nawab. It is a historical city. Since long, in 1856, when there was a second independence, the War of Independence took place in Lucknow. That was in Residency. This time, we have really wonderful things to see in Lucknow. It is a historical city. There are many monuments in the city, like Bada Imam Bada, Chota Imam Bada, Rumi Darwaza. There are so many historical things. Historical buildings are there. And there are extraordinary medical institutes also. We have got Sanjay Gandhi PGI, we have got Ram Manohar Lohia Institute, and that they all are nationally reputed institutes. King George's Medical University, which was established long back in 1905. It was the fifth medical college of the India, of undivided India. It's a great institution, and it has produced so many doctors who are spread all over the world, and they're known as Georgians. And we are having a special tradition that we always respect our seniors, whether they are here in India or anywhere in the world. Whenever I go anywhere, we get immediately a Georgian, and he looks like my senior person, and if he's a senior, he will behave like a senior even if it is 1 year senior. The King George's Medical University is one of the oldest university, and it is really a center of eminence. Institute of Eminence recognized by government, and we are getting these things. Beside this, Uttar Pradesh is also known for many good things. Most of the prime ministers, they have come from Uttar Pradesh. Even the present prime minister for the last 2 terms is from Uttar Pradesh, from a city of Varanasi. The city of Varanasi, where Lord Shiva is praised like anything. The morning of Banaras is a really wonderful morning. The Krishna, the city of Vrindavan and Mathura, where he has spent his most of the time, and most renowned, most popular name of Ram Lala. Ram Lala, Bhagavan Ram ki dharti, that is Ayodhya. And all these places are not far from Lucknow. This time, we will have a special desk for travel desk. We will give a responsibility to somebody who will organize these tours for you starting from 2 months advance. And that person who does this work is doing for us for last so many years, nearly 30 years he's doing this for us, and absolutely flawless. So we'll organize this thing, we'll keep a travel desk in the campus where the conference will be organizing. We are planning to get participation from the other institutes also. For example, the Lohia Institute, Ram Manohar Lohia Institute, the Sanjay Gandhi PGI, Dr. Ram Manohar Lohia Institute, Dr. Atul Jain is sitting here, and the director of SGPGI is my batchmate, and I am there in the King George Medical University. There are all people are there to really welcome you in advance. And thank you, Dr. Kamal, and thank you, Dr. Padmaja, that they have given this opportunity to serve you and to develop some new milestones in field of pharmacology. I again reiterate that pharmacology is a branch which is everywhere. We are in the house of any discipline in the medicine, and we are there. And we have to prove ourselves that we are the able people so that we can guide them, we can teach them the real medicine. So that is the most important thing which we have to develop in years to come. Thank you very much.
Thank you, sir. And thanks to all my postgraduate students who have given me the precious time. I have taken their kind permission, and we are opening this session with the postgraduate students. They can write their questions also. I will try to take up as many questions as possible. Our respected secretary, Dr. R.K. Dixit, will be noting it down on a writing pad. Probably you are ready, sir? Sir, you can take the seat. You can talk. Thank you. Thank you. Thank you. So our respected secretary, sir, will be noting it down, your suggestions. And madam is listening carefully. Myself is inviting the questions, expectations. And before I can take the questions, I would like to announce that many of the activities of the NPT probably you are not aware of. Probably you are thinking this is an academic organization. All the times who are having the academician, we have seen the leadership quality and organizing skills of the president also, secretary also. This organization is not limited to research activity on academic activity. I would like to know you all, those who are not aware of, that here in another conferences where there are other colleagues also apart from the pharmacologists, we were a little bit hesitant speaking about the pharmacologist, future of the pharmacologist. Okay, I am talking strictly about the appointment. We are totally against the appointment of non-medical pharmacologists without an MBBS degree in the medical colleges, because they are taking your rights. And for this purpose only, probably you are not aware that non-medical 4 organizations have a case in the court of law, Delhi High Court, against this decision. NMC has excluded, probably how many of you are aware that microbiology and pharmacology, the MD degree, without MD degree faculty have been excluded. Please raise your hands. How many are you are aware of? How many you are aware of this fact? That in Department of Pharmacology and in Department of Microbiology, as per the recent NMC guidelines, no faculty appointment can be done without an MD degree. How many you are aware of? Please raise your hand. Are you aware of the notification of the National Medical Commission? Those who are not aware of, please, those who are not aware of this fact, please raise your hands. Those who are not aware of, you are not aware of, and you will be surprised to know that 4 organizations have put a case in court of law, Delhi against the decision of NMC. And I am happily, very gracefully, happily announcing from this dais, from the, on behalf of NPT, that out of your all pharmacology organizations, I am not taking the name of any organization, any organizations, apart from our research activity, apart from our academic activity, we are taking interest in your rights, in your future also, and we are 100% devoted. We are the only organization which has filed a case of intervention against that. We are a party. On the information website of Delhi High Court, information is available, court case number is available. National Association of Pharmacology and Therapeutics is one of the party of that, who is fighting for the rights, your rights. Our main motto is that only MD pharmacology should be appointed at faculty position. Your future must be secured, because you, with lot of expectation, you have done MBBS, you have done work day and night, you have worked hard. You have taken out 2 entrance tests, NEET UG you have given, NEET PG you have given. So we cannot compete and compare. Only at faculty position there is a right of the MD pharmacologist. So with these words, I am inviting this session opening to all. Please raise your hands and please let us know your expectations from NPT, what we can do further.
Hello.
Yes, please. Please give your introduction, your name, from which state.
I am Dr. Vineet Chaturvedi from Gajara Raja Medical College, Gwalior. And I must congratulate the organizing team and committee that they have brilliantly organized this conference. So thank you very much for this. Ma'am, I have few suggestions. First, we have to work so that every pharmacologist in India should be a member of NPT. First thing. Because we have to make this a national organization, like other specialties have their national organization, so we should also try to do that. Every pharmacologist, every MD pharmacology student or faculty, whosoever is, should be a member of NPT, first thing.
I fully, sorry to interrupt, fully agree with him, and we have provided, it's very easy to become a member, and the price is also kept very low. It's just 2,000 rupees. If you go out and eat one biryani, you will be spending 2,000 rupees. So please don't wait for some time to come. Do it today itself. Please be member of NPT.
My second suggestion is to participate in this conference, particularly for the MD students, to present their papers, to present their posters. They should be the member of NPT first. This is my second suggestion, because all other organizations do that. If a PG wants to present their paper in the conference, he has to take the membership of that association. So this way we will make our organization huge, and if we are organized, then we can do many things in coming future. Thank you very much.
My dear friend, I would like to address this question. This is very, very important questions asked by my postgraduate colleagues, that we have not allowed, and without MD pharmacology, we are not allowing to be a member of national NPT. Okay. Due to some technical reason that I am not able to explain at this moment, we are doing this. We are having all the future plans for that. But for all practical purpose, if you are a student, you are fully allowed to take participation in any of the activity of NPT. Any of the activity of NPT, and please remember for that, if any person who is with the qualification of MSc, PhD want to participate, he is not allowed. You are allowed, but due to some technical reasons, at this juncture of time, we are not opening it. But as soon as you are getting your registration degree, see, your registration degree is the only evidence that it is a medical doctor. We don't want any kind of the percolation in NPT that any person without an MD or MBBS degree should enter. So to keep that very strict criteria, we are just observing that. But for all the future means our actions, we are keeping this in our mind. I hope I have given the answer of your question.
No, I was not asking any questions. I was just suggesting.
Yeah, suggestion.
Yeah.
Your suggestion is well taken.
Yeah. Sir, we can allow students to take part in the activities, in teaching activities, in training activities, in workshop, in everything. I think we make it compulsory to present a paper in the conference. NPT registration should be mandatory. I think that.
Okay. Your suggestion is well taken. Thank you. Any other student? Please, your name, your medical college, your state. Please feel free to ask your questions.
Hello.
Don't hesitate. Please come up. This is for you only. Yeah, please.
Sir, I am Dr. Santosh Saklani, native from Uttarakhand, Rishikesh, but at present I am in RMC Kakinada, Andhra Pradesh, doing my MD Pharmacology. Sir, my idea is, I don't know whether it is feasible or possible, but my idea and question is that, is it feasible and possible that a patient after being diagnosed for a disease or a group of diseases by any clinical or surgical specialty be prescribed minimum essential drugs most suitable for that individual by the specialist of drugs, that is by pharmacology, OPD. Means in future, is it feasible and possible that there should be a pharma OPD in at least tertiary care hospital so that all the patients after diagnosed from various clinical or surgical departments, they may be referred to the pharmacology OPD for best suitable drugs prescription.
You wanted to start a separate OPD for pharmacology department?
Yes.
I feel madam can also give the answer of that. And there are, in many medical colleges, there are specialized OPD that are being run by the pharmacology people. There is no restriction to that. But officially, a specific pharmacology OPD is not running, probably. We can write this suggestion to the government.
Actually, sir, what I wanted to convey is that after being diagnosed by other departments, clinical and surgical.
But clinical audit of the drugs, that is also a suitable activity, probably.
But that is not, sir, real time. Means the patient-oriented treatment, patient-centered treatment. After being diagnosed from various diseases by various departments, patients should be given minimum essential drugs which is best suitable for that patient, and that will be that decision is made by pharmacology specialist only.
I got it. Probably he is not aware of the recent NMC guideline. In recent NMC guidelines are saying that postgraduate students are also need to be posted in the clinical department, if I am right. So you can do that activity there also. It is officially allowed. Recently, they have revised the criteria, guidelines, and it is already allowed. You will be taken up, maybe to apply it across the country, each and every state, in all more than 500 medical colleges, it will be possible, but it will take some time because these guidelines have been received probably 3 months back, probably, sir. So it will take little bit time.
Yeah, in this we have to, I would just like to say word of caution. Let us not create conflict between our clinical colleagues and pharmacology, because the moment we enter, we'll be, kind of considered as an interference. So let us at this juncture not venture into that. We only be in an advisory capacity. That's the thing. Let us be in the advisory capacity. Let us be open to take advice. So we are open. You can have pharmacology. OPD, there is no need. But there should be a desk or a kind of a connection or line. Anybody can, any clinician can take opinion and advise. You know, whether I'm going to prescribe this or send sample prescriptions or copies of the prescriptions to the pharmacology department or to the pharmacologist for their opinion, or if it can be improved and things like that. But not direct intervention, because what is being suggested is that the clinician diagnoses and the pharmacologist prescribes. No, it will create a big conflict. Big conflict. So please do not enter this Pandora's box. Let us be in an advisory capacity. And we have been doing it. Not now, nothing new. No, we have been doing it for 30 years. Yeah, sure.
Yes, I'm from West Bengal, Muller Medical College, and we have a therapeutic clinic running there by pharmacology, Department of Pharmacology. Those who have DM, those who are clinical pharmacologists, they are entitled to run a OPD. And all those patients from medicine, diabetic clinic, those who have problems regarding drug-drug interactions, or they have reconciliation, sometimes this prescription is wrong, that's not rational. And we have the rapport with those departments, and they refer the patients to this therapeutic clinic, and it's run by this clinical pharmacologist of Muller Medical College. And also we have another institute at medical college, yes, tropical medical college, and that is also running. And so I don't think that therapeutic clinic is impossible by pharmacologists, especially clinical pharmacologists.
We are taking up the question from another student. He will please say your name.
Can I clarify a little one thing in this regard?
Yes.
There are already two things going on in all the medical colleges by the direction of Government of India. One is pharmacovigilance, in which we are handling the adverse drug reaction. Second is the program which is run by the ICMR, that is the prescription audit. What we should do in this, that we should collect the data from the OPD as well as from IPD, both the things, and a monthly meeting is done in which we tell that these are the things which are found irrational, and every month we try to improve our report that now this has improved in this aspect. And this is our not only responsibility, but this is the most important thing which a pharmacologist should and must do.
Yeah, your good name, your state?
I'm Dr. Nandana, final year MD Pharmacology from Rangaraya College, Andhra Pradesh. Before giving my suggestion, first of all, I would thank to A1 Logistics for creating beautiful website. I like the website and everything, this interface. And my recommendation is we can add a button for fundraising in the NAPTICON website, so that we can pay any amount, whatever we wish, for any court case, anything.
Okay.
And one more thing, we can add one more option in the website as a latest updates, so that when is our publications are going to published, and one more, and some YouTube tutorials how to write cover letter, and any advertisement promotions.
Okay.
So mainly I would like to suggest any fundraising option.
Okay. Your suggestions are well taken. Sir, please note it down. From the core of my heart, I am thankful to such of my students who are interested in the fundraising activities. Please give him a big hand. Because nobody wanted to ask for fundraising. He is thinking that for each and every activity, we need funds. Okay? And if the funds are coming, we are meaning that you are interested yourself, you are investing in your future. Though we are not asking, but it shows that your willingness, how you are interested in making your future secure. Any questions from this side? Students, please, your suggestions.
Good afternoon. We have been discussing this in the WhatsApp group, that there is a small suggestion that MD Pharmacology and Therapeutics we have to make sure that NMC reframes that as MD Pharmacology and Therapeutics. Because at least we are seeing cases and we are like clinicians, we can point out. Because the moment I tell pharmacologist, if I introduce to a layperson, oh, pharmacy, PharmD, that is the main thing which comes up. So it's a request from the association to please take up this matter with NMC. Right.
Sir, please note it down, this suggestion. Anybody from this side?
Thank you, everyone. Myself, Dr. Aruna, professor from Department Medical College, Chennai, Tamil Nadu. So my suggestions are in this new CBME curriculum, the first phase, second phase and third phase, pharmacology falls in the second phase. All of us are agreeing that pharmacology should be there throughout and along with the clinical subjects. Can we make from the association some suggestions, since we have the members of NMC with us and in the association? Why can't it be extended to the phase 3 along with the, because forensic had been extended like that from the second phase one part, and the phase 3 also forensic had been taken, and community medicine is there throughout, and they have included community medicine as one of the clinical subjects now in CBME. So any chance for proceeding pharmacology into the, along with the clinical subjects as a clinical part of medicine in phase 3? That would be a good outcome for us, as many of them suggested, including ourselves into the therapeutics.
Can we take the opinion on this from the colleagues? How many are in the favor of that? Please raise your hands. Everybody have listened his suggestion carefully, that she wanted to extend the duration up to final year or fourth year, third year.
Course of pharmacology to the UG students or something of that time. We want to put forth that it is along with the clinical subject.
It is along with the clinical subject. How many in favor? How many in favor? Sir, please write it down.
Sir, once it was 1.5 years course,
yes,
that can be done like in phase 2. In phase 2 we can have for 6 months, and in phase 3 the extension can be.
Definitely your suggestion is well taken. We will be writing officially from our official letterhead with the signature of Honorable President and Secretary, and you know it very well that registered organization can communicate effectively with the government agency and National Medical Commission also. We will be sending your suggestion to NMC for the suggestion. Thank you. Please open up your ideas, your suggestion. What are your expectations from NPT? What are your expectations? What you more want?
I'm Dr. Priyanka Banik from RG Medical College and Hospital, Kolkata. My suggestion is, like, we are getting the degree as of now, MD degree, MD in pharmacology. Like, can we get like MD in clinical pharmacology or MD pharmacology and therapeutics? MD pharmacology and therapeutics, like that. Okay. Yeah.
Clinical pharmacology and therapeutics are two different things. So it should be pharmacology and therapeutics.
Yeah, that is the name of Association of Pharmacology and Therapeutics.
And let me tell you, King George's Medical College, Lucknow, the department has been known for 50 years as Department of Pharmacology and Therapeutics. Am I right? Am I right?
One more question.
So it has been known as Department of Pharmacology and Therapeutics. Yeah, so it should be in the MCG, gadget. It should be a pharmacology and therapeutic, because the curriculum does include so much of therapeutics.
Yes.
It does include therapeutic. Treatment of snake bite, they have introduced new. What is that? Is it not therapeutics? Bites, and so many things, treatment of hypertension, everything. Treatment is there. So why it should only be pharmacology? Yeah. It should be pharmacology and therapeutics. So this association will take up this matter and change the name of the subject.
Okay.
And
how many in favor of change the name of pharmacology and therapeutics?
Once we change the name of the subject from pharmacology to pharmacology and therapeutics,
yes.
This will come automatically.
Yes.
This will come automatically.
We will be definitely writing to National Medical Commission on behalf of National Association of Pharmacology and Therapeutics that as early as possible, it is the demand of all pharmacologists from Kashmir to Kanyakumari and Gujarat to Arunachal Pradesh that change the name or modify their name, give the degree in the name of MD Pharmacology and Therapeutics.
Yes.
With these words, I would like to conclude these sessions. Those students who are unable to find the time for the question, how many you are member of our NMDP pharmacology group? We have created a separate group, please. MD pharmacology students, raise your hands. There are still many. So I would like to request that through WhatsApp, we are in connection to more than 1,000 of MD pharmacology students. Those who are not the members, for some technical reasons, we are not giving the link openly, but you can request from your colleagues, from your pharmacology colleagues. Okay, so all the MD pharmacology students who are in your contact, please join our WhatsApp group. It is free of cost. Okay, through this group, you will be getting free newsletter, free updates, and various important PowerPoint presentation. Moreover, this group will keep you motivated all the time. More than 1,000 of the MD postgraduates are member of this group. It is because the limited capacity of the WhatsApp group, probably more than 500 probably, or 556. How many it is? 556. So we have created a second group. We will keep on creating third group, fourth group, depending on your demand. We are not going to stop anywhere. We are trying to include almost all the MD pharmacology students of the country. Thank you very much.
Sir, one more idea, sir.
Those who have been unable to ask questions, I would like to that they can ask in WhatsApp group, or they can ask me personally during the lunch hour. Okay, thank you. With these words, I am putting an end of this session. Madam will announce. Thank you.
Excuse me, sir. Sir, I think it's...
No, the questions, please WhatsApp, please.
I think it's very valuable right now if I express it, sir, madam. Madam, my suggestion is we can make it as a dictum for all the MD pharmacology students. We can make arrangements so that we can purchase all the MD curriculum books from our website itself instead of purchasing in Amazon, Flipkart, so that we get more fundraising. It's an idea, madam.
Thank you, thank you. We'll look into it. We'll seriously look into it. Thank you. You should definitely join the industry.
Yes. Excuse me?
Thank you very much.
What is the membership fees of this association? Ma'am, what is membership of fees for this association? Can anybody tell? Live membership fee. Live membership fee. Just tell the neighbor. Just tell the neighbor. बगल वाले को बताओ. बगल वाले को बताओ ना. ऐसे करके बता दो हम. 2000. जल्दी बोलो 2000. और फिर आगे कहो अगली बार बढ़ जाएगी. So जितना जल्दी हो सके उतना जल्दी बन जाओ. Wonderful.
Thank you so much, Dr. C.M. Kamal, Dr. R.K. Dixit, and Dr. Panuja, ma'am, for this deliberation.
Hello.
Yes, sir.
I am trying to re-attire myself to pharmacology. There are a few suggestions I would like to make, if at all it is fit into your opinions. I have difficulty in vision, I have difficulty in hearing,
I'm 80 years old. I attend this function to be with pharmacologists, to be helping pharmacology, because I have worked 50 years in pharmacology. I just want to take you onto a different path line of thinking. We have been talking about therapeutics, yes. We have to give everything to, but we are teaching all the time to the students to become a doctor. We make doctors, and the doctors are going to become a powerful man, money-minded man, and a position man. This is part of life. We cannot say. But what are we doing to the patient? We are not anywhere to the patient as a pharmacologist. Has any one of us have any contact with the patient? We are in contact with government, we are in contact with other associations, MDs, but do we have any contact with patients? That is what I wanted to ask you. Let us start thinking how we can, the reason for that is simple. We don't want to become popular with them, but we want to be functional with them. We must be able to do things for the public. When we don't have contact with public, what can we do for the public? So what my point is, how are we going to contact the people who are suffering? So what is required is service. And how are you going to do it? I am not talking about politics, I am talking about service in our day-to-day life. How are we going to go? Now, many times it has happened, somebody asks me, what are you doing, doctor? I am a pharmacologist. They say, what? Pharmacologist. What is pharmacology? You are a pharmacist. They know pharmacist. They do not know pharmacologist. Why? Because pharmacist is in contact with the people. We are not in contact with the people. So if you want to be known, if our service should be known to the people so that they understand and follow our instructions, we have to contact with people. It's not there now. If there are so many reasons why we should do that. Now we talk about treatment therapeutics. The failure of treatment. The doctor has given perfect medicine, perfect everything, lovely medicines, costly medicines, but the patient is not improving. What is the main reason for that? Non-compliance. We know that. Non-compliance by the patient is the main reason the treatment is not working. What is the use of a good doctor, great doctor, and a great medicine? So what are we doing to improve the compliance of the patient? We are not contacting them. Do you know when you sell a medicine, are we telling the patients what are the information you should know while buying the medicine? The expiry date, manufacturing date, and manufacturing number. Why they should know it? Have you told them any time? Why not? I bring the medicines home and keep it at home. Do we ever tell the patient where to keep it? How to keep it? Now, why can't we organize a bit of, when the patient is buying this thing is supposed in an OPD, government OPD or a hospital OPD, why can't we ask our postgraduate student or our few medical students to sit with the patient when the senior person explains to them what are the things they should know about the medicine, what they should not do, etc. That will be a training for the interns and MD and also for students, what they should tell the patients. Why can't we tell how to dispose of the patient? You go to your house and ask them, you just ask the family to bring out all the medicine in the house. You will find there are so many medicines inside the house. How did it come there? How did it come there? Because of non-compliance. They are supposed to take it for 15 days, they took it for 5 days. 10 days medicine is there. What are we going to do with it? No one is bothered. So we have to start teaching. We have to start acting as a teacher, not for medical students only, for the public. So we have to reach out. How do we reach out?
Hello. Madam,
Yes. Just before we break for lunch, we want to give away the prizes for paper and poster presentation. They have trains and they have been requesting me for a long time. That's why I was in a hurry with him. Yeah, please come announce the prizes.
I now request Dr. Lopamudra Chowdhury to kindly give away the prizes for oral paper faculty. In first place for oral paper faculty, we have Dr. Ivy Abraham Varghese. Can we have a big round of applause for her? I request Dr. Manish to come forward and receive the prize on behalf of Dr. Ivy. Dr. Manish.
She has left, I think, yeah.
In second place, we have Dr. Nitin Gaikwad. Can we have a big round of applause for Dr. Nitin Gaikwad? For oral paper and postgraduate category, I request ma'am to give the first prize to Dr. J. Sylvia. Please give a round of applause to Dr. J. Sylvia. Second place, we have Dr. Arjun. Please give a round of applause to Dr. Arjun. Thank you, Arjun. Thank you, ma'am. Please give a round of applause to Dr. Lopamudra. Thank you. I'd like to call upon Dr. Bhuvaneshwari K. onto the stage to give out the e-poster prizes. At first place, faculty, e-poster goes to, any guesses? Dr. Veena Nayak. Please give a round of applause to Dr. Veena Nayak. Dr. S. Mohammed Mustafa. Please give a round of applause to Dr. Mohammed Mustafa. Second place, e-poster. Thank you, and a big round of applause to Dr. Bhuvaneshwari. I'd like to now call upon Dr. Chitra Kanvelkar to give out the e-poster prizes to our postgraduates. finalists at the e-poster prize we have. Any guesses? Dr. Archana M. Please give a big round of applause to Dr. Archana M. The second place in e-poster postgraduate category goes to Dr. Anand Alex. Please give him a big round of applause. Dr. Anand Alex. Congratulations, Dr. Anand, and a big round of applause to Dr. Chitra Kanvelkar. With this, we come to the end of the paper and the poster prize distribution, and we break for lunch. I kindly request all the delegates, the head of departments, and all our delegates to kindly go ahead, have lunch, and join us back at the session for the most awaited finals of quiz. So please come back for the quiz at sharp 1:35 at the hall.
First year resident from Government Medical College, Bhavnagar.
Hello, I am Dr. Tamil Selvi from AIMS Nagpur, second year resident.
Good afternoon, I am Dr. Abhirami Balambika, final year postgraduate from Tirunelveli Medical College.
Okay, Team D.
I'm Dr. Aravind B. From Kottayam Medical College, third year PG.
Be louder and clear.
Third year PG.
Dr. Christy Jashti, Government Medical College, Kottayam, second year PG.
Okay.
Dr. Komal Kurade, final year PG, SET GS Medical College.
Okay.
Hello, good afternoon. I am Dr. Shoham Sena, first year postgraduate student from SET GS Medical College.
Great.
So, can we give a big round of applause to all these teams, because 36 teams have participated in the prelims, and these are the 5 teams which have qualified for the final. So a big round of applause to these teams.
So here we begin for the landmark, the first quiz of NAPTICON. So before I start the first round, let me give you some brief rules about the quiz. This quiz will have 6 rounds, and rules of each round will be briefed to you before the round begins. In case we have a conflict with you, the decision of the judge will be considered the final decision. Okay? So these are the general rules. Let's go ahead with the first round, and this round is called as You Choose. In this round, each team will be asked one question. The first team, I am going to start with team A. First team will be given 30 seconds to answer this question. In case you answer the question correct, you will be given 10 points. If your answer is wrong, or you're not able to answer this question, the question is not going to be passed to team B. This question is going to be passed to the audiences. Okay? So there is no passing for this round. Are we ready? So the first question to team A: who coined the term antibiotic? Alexander Fleming, Salesman Waxman, Robert Koch, Louis Pasteur, Paul Ehrlich. Your time starts now. Timekeeper.
Option B, Selman Waxman.
Are you sure?
Yes.
Lock.
Lock.
The answer is correct. The Selman Waxman, he was a scientist who also discovered which antibiotic? Streptomycin, and he also coined the same term antibiotic. So 10 points to team A. Team B, are you ready? Yes. This is your question. Afrezza and Exubera, these are the 2 examples of drugs which failed. Because of what reason? Because of safety issues, because of efficacy related issues, pharmacokinetic related issues, cost of formulation, or was it lack of acceptability? Your time starts now. Team B. Team B. Timekeeper.
Option E, lack of acceptability.
Lack of acceptability. Lock? You're right. Exubera was the product from the Pfizer. Apreza was the joint collaboration between Sanofi and Mankind. Both of them, they discontinued the inhalational insulins because there was not, cells were not picking up. There was no reason of safety, no reason of efficacy. Just because the cells were not picking up, they discontinued. Team C, ready? Okay. Which of the following drugs may inhibit hepatic microsomal P450 responsible for warfarin metabolism? Is it amiodarone? Is it ethanol? Is it phenobarbital? Is it carbamazepine? Or is it rifampicin or rifampine? Time starts now. Are you sure? Lock करें? You still have time. The answer is wrong. Answer is amiodarone. Amiodarone is an enzyme inhibitor. Rest all are enzyme inducers. Carbamazepine is an inducer, phenobarbital is inducer, rifampicin is inducer, ethanol also is kind of inducer. Next team, team D. Are you ready? Question for you. Which of the following is an example of non-parametric test? Is it ANOVA? Is it independent t-test, Pearson correlation test, Mann-Whitney test, or is it paired t-test? Time starts now.
Option D, Mann-Whitney U test.
Mann-Whitney U test. Lock करें? Answer is correct. All of the other tests are parametric except Mann-Whitney test. The question, last question for this round goes to team E. Which of the following drug is used to induce diabetes in animals? Is it alloxan? Is it furosemide? Is it xylitol? Is it saccharin or aspartame? Time starts now.
A, alloxan.
A, alloxan. Sure? That's correct answer. So here we complete round 1. May I request the scorer to tell us what is the final round at the end of round 1.
So team A is at 10 points, team B is at 10 points, team C is currently at 0, team D is at 10 points, and team E at 10 points.
Can we have a round of applause for all teams? So we go to second round, and I would request Dr. Melvin to take the second round.
Switch off.
Okay. So this round is all about seeing something and giving the answer to it. Big quiz. So here we are going to have 2 rounds. So each team is going to get 2 questions. So I will start with team A, B, C, D, E, and again go back in the reverse direction, E, D, C, B, A. In this round there is passing. If you get a question right directly, you get 10 points. If it's a past answer, you get 5 points. Is it clear? No negative marking. Okay, we'll start. So question to team A. Are you ready, team A? This book, Bottle of Lies, that you see on the left side, highlights the scandal with respect to a particular pharmaceutical company which failed multiple FDA good manufacturing practices audits. Which is the company specifically that this book alludes to? Your time starts now. Your time is running out. Okay, no answer.
You don't have
Asked.
time.
So, no time. Team B? No negative marking, please. No. Team C? No. Team D? Wrong. Sorry? Sipla. Wrong. Anyone from the audience? Yes, Dr. Saibal says Ranback C. Please give him a chocolate. Please clap for Dr. Saibal. Ranback C is the answer. Now, question to Team B. Identify the name of the organization which has this logo. This is for Team B.
Novo Nordisk.
Novo Nordisk is the perfect answer. Team B gets 10 points. Question to Team C. Identify the name of the organization which has this logo. I can't hear you. Mike, please. No promptings from the audience. Dr. Reddy's is a perfect answer. Team C also gets 10 points. So, question to Team D. Name this historical event that took place in 1846. The painting that you see. What is this painting referring to? Just name the event.
W.T.G. Morton's discovery of ether.
Perfect. William Morton's demonstration of ether anesthesia in Massachusetts General Hospital, Boston. Historic landmark event in the field of anesthesia. Okay, question to Team E. Identify this personality. Pass. Okay, pass. It goes to Team A. Pass. Team B. Pass. Team C. Pass. Team D. Sorry? No.
Audience.
Audience. Dr. Fauci is the right answer. Dr. Sharma, please, someone. Yeah. Yeah, so right throughout the COVID pandemic, you would have seen him standing next to Donald Trump and giving him advice about, giving the country advice about COVID scenario, the task force. He was heading the task force there. He's the director of NIAID, a very reputed researcher, medical advisor to multiple, chief medical advisor to multiple US presidents. Now we are going in the reverse direction. Team E.
Now, in case we are starting with Team E, if they don't answer the question, it's going to go to Team D. We are going in the reverse direction.
Reverse direction, okay. Okay, again for you. Identify this personality.
Audience, please keep quiet.
Audience, please keep quiet. No promptings. Pass.
Team D.
D.
Team D, question to you. Pass.
Pass. Team C.
Speak in mic.
VG Soman.
That's a wrong answer. Team B.
YL Subbarao, he passed away. YL Subbarao.
No. Sorry, it's wrong. Team A.
Dr. Dilip.
Dilip.
Baloch, second name, but
The final decision will ask judge to give the
Okay,
ORS.
I will show the answer. Now the judge has a... Okay, before going to the judge, anyone from the audience wants to answer this? Yes, ma'am, in the last row.
Yes.
Dilip Mahalanabis is the right answer. Please give ma'am
So he gave the name but not the surname.
Sir, they have said Dilip. what do we do with them, sir? Should we give them 2.5 for that, or 5 points, or we don't give them? 2.5? Okay, please give 2.5 for this team A as a past answer.
So we went to team D.
Now question to team D. Identify this personality.
Audience, please.
Audience, please. No promptings. Pass. Okay, question to team C.
Pass.
Pass. Team B. Pass. Team A. Pass. Come back to team E. Pass.
Dr. Ganesh has raised the
Dr.
hand.
Ganesh raised first. Perfect. Dr. Ganesh is Dr. Viji Somani, current Drug Controller General of India.
Question to team C.
Team C. Okay. Identify this personality.
Audiences, please.
Audience, please. No promptings.
No prompting, please.
Ramnath Chopra.
Perfect. Colonel Ramnath Chopra. That's the perfect answer. Who is he? Great. He was involved in starting the first medical pharmacology department and called as the father of Indian pharmacology. So, question to team B now?
Yes.
Okay.
Question B.
Team B, this is your question. Identify this personality. Pass. Team A. Pass. Team E. Pass. Team D. Pass. Team C.
Audience.
Audience.
What's his first name? What is his first name?
Okay, Cyrus Poonawalla. Who is he? The founder of Serum Institute of India. I think all of us, most of us would have got this vaccine from their company. So who should be getting the audience? So many people answered. So.
I think, who answered the question? Somebody
Okay, we are not sure who answered, so we are taking this chocolate. Okay, thank you. So last question now to team A?
Team A.
Okay. Identify this personality. Pass. Team E. Okay, team D. Pass. C. B. Anyone in the audience? Okay, I'll give a clue now. He has written one, he's one person who's, all of us know, his book, most of us, or almost all of us in this room has read. A pharmacology textbook. Yes? Bertram Katzung is the answer. So he's incidentally also an astrophysicist. He's standing before his own personal telescope. Okay? So this is Bertram Katzung. So we come to the end. Okay, there is one question now directly for the audience. This is not for any of the teams. Identify this personality. Anybody? Sir, on the last. Sorry? Dr. Kang is the right answer. Dr. Gagantip Kang. Who was he, sir? Who was she, sir? Yeah, so CMC Vellore, chief immunologist, virologist, one of the important persons involved in the task force for COVID-19. You would have seen her in the newspapers and the television right through the pandemic. So,
I think sir, we can give a chocolate to Yashasri madam.
Sir gets a chocolate. Okay, I think we come to the end of this round. Yeah, thank you. So
could you just help me? Could you stop the screen?
Yeah, Dr. Reshma, please, you can read the final scores.
Okay, the scores are Team A at 12.5, Team B at 20, Team C at 20, Team D
at 20 and Team E at 10.
Now we are ready for round 3, and this round is Who Am I? So I'll be giving you autobiography of a medicine, and you have to identify based on the clues that I will give you. So that's the round Who Am I? Okay, I'll start with Team A, and again, if you answer correctly, you will get 10 points. If you don't answer, the question is passed on to Team B, and likewise. There is only one question per team. So rules I already told you. So can we go to straight away to the question first? Okay. Question for Team A. My experience with heart failure is not even more than 10 years. After administration, I reduced the pace of heart beating and also the oxygen requirement. My name will give you a fair clue of what I do. Some patients, they complain of transient visual disturbances called as phospines after my administration. Who am I? Time starts now.
Is it levosimendan?
No. Passes to Team B. Team C. She's right. The answer is Eva Bradin. Bradin stands for bradycardia. So the name tells you what the drug does, and also the adverse event phospines is caused by Eva Bradin. Question to Team B. They call me as a beta blocker, and I'm similar to all of my classmates on the most of the terms, but I do not reduce the heart rate as much as they do, and my name rhymes with Sim. Who am I? Any answer. There is no negative marking, you can still try. Team C. Pass. Team B. Team D, sorry. Pass. Team E. There is no negative marking, you can still try it. Team A. Audience. That's right answer, Pindolol. Pindolol is a partial agonist and it has an intrinsic sympathomimetic activity. So the answer is Pindolol. What happened? So rhymes with sin and pin. It rhymes with sim and pin. So that was the clue. Okay. Can we reduce the noise? Question for Team C. Are you ready? I am a drug of choice for patients presenting with a specific type of arrhythmia. I am used when ECG shows superimposition of P waves with T waves. I do not fit any of the 4 categories given by William Vaughan and Singh classification. Who I am? Who am I? There is no negative marking. You can still try. Team D.
Adenosine.
That's the right answer. Adenosine is used for supraventricular tachycardia and PSVT is a drug of choice. That's a miscellaneous category. Category 1 is sodium channel blockers. Category 2 is what? Beta blockers. Category 3 is potassium channel blocker. Category 4 is calcium channel blocker, and this is miscellaneous. Atropine, adenosine, all those are miscellaneous. question to team D. Are you ready? I'm a drug of choice for patient presenting with, oh, that's over. I came to market with the hope of helping sneezers, those who have sneezing. However, because of my tendency to disturb heart, I was thrown out of the market. However, my younger sibling has come back to rule the market and is still ruling the market. Who am I? Team D.
Celastin.
No, it's wrong.
Cetirizine.
No.
Terfenadine. Terfenadine.
That's right answer. Terfenadine is the right answer. The derivative of terfenadine is fexofenadine. Fexofenadine is available in the market. Terfenadine has been discontinued because of risk of torsades de pontis. Team E. Last question of the round. Are you ready? No. Surgeons, obstetricians, as well as cardiologists, they need me. And I am kind to the babies during pregnancy. Some use me locally while some others, they prefer me as an injection. Who am I? Think. There is no negative marking, but only one chance. Say loudly.
Lignocaine.
What's that?
Lignocaine. Lignocaine.
Wrong. Team A, pass. Team B, you don't have time. Team C. Team D, audience. That's right answer. Magnesium sulfate used by surgeon as a maxillary dressing, used by obstetrician as a tocolytic agent in eclampsia, used as by cardiologist for torsades de pontis, and even as a antacid, not as magnesium sulfate but magnesium hydroxide. It's a versatile drug. So that completes round 3. Thank you. So can we have the scores?
The scores after round 3 are Team A at 17.5, Team E at 10, Team B at 20 points, Team C and Team D at 25 points.
At this point of time, Team C and Team D, they have a tie. Tough fight between Team C and Team D. So are we ready for next round, round 4? I request Dr. Melvin to take over for the round 4.
Okay, so you can switch on. So this round is cocktail round. So in this round, essentially you're going to have a lot of questions from different areas, need not necessarily be pharmacology, it could be clinical research, it could be regulations. Okay, so let's begin this round. In this you'll have 2 questions for each team. The first, if your team if you're getting it straight away, you get 10 points. If it is a past question, you get 5 points. No extra time is given for when the question is passed. And with this we go to the first question. Shall we start with this? Okay. Team A. Which country's regulatory body is named as TGA, Therapeutics Goods Administration? A country.
United Kingdom, UK.
UK is the wrong answer. I pass it to Team B.
Sweden.
Pass. Team C. India. India is the wrong answer. Team D. Norway. Team E.
Netherlands.
Netherlands.
Audience.
Audience. Yeah, right at the end. Australia is the right answer, sir. Please give the chocolate. Okay, so it is Australia. Now the question to team B. Okay. This is for team B. Name the ICH guideline which specifies how non-antiarrhythmic drugs should be evaluated for their pro-arrhythmic potential. There is a guideline on how these kind of studies need to be done, QT, QTC studies. So what is the guideline for these studies? What is the name of the guideline? Pass, team C, team D, E, A. Anyone from the audience?
Nitya madam is raising hand.
Yeah. Great, ma'am. E14 guidelines is the right answer. Dr. Nitya ma'am gets a gift. So ICH gives several guidelines for different scenarios. So for this we have E14 guidelines. The next is for team C. What is the statistical measure for inter-rater reliability for categorical variables?
Convergent reliability.
Sorry, team D. Sorry, loudly. Okay, I can't hear you. You have to be loud. Interquartile range, no.
What is it? Speak loudly in the mic.
You have to speak loudly in the mic. Okay, I'm passing.
Kolmograph-Smirnov.
Kolmograph-Smirnov is not the right answer. You're passing. Audience, anybody?
Joshi,
sir?
Yes.
Cohen Scapa is the right answer. Okay, so team.
Now team D.
Team D. What is the statistic used to measure association between two ordinal variables? Okay.
Standard deviation.
Standard deviation. Pass. Okay. Team A.
Linear regression.
Linear regression is not the answer. Team B.
Wilcoxon's.
Wilcoxon. Team C. Okay. Audience. Spearman. Anybody else? Yes? Intra-class correlation. Anybody else? Kendall's Tau is the right answer. Nerurkar sir can get the gift. Great. Team E. Name the first US law, in fact it was almost the first in the world, that required mandatory proof in the form of clinical trials for effectiveness and safety of a drug before it reaches the market. NDA approvals. Okay, pass to team A.
No, team D.
Team? No, it's going this way. Team A?
Helsinki Declaration.
No. Team B? No. Team C? FDA? FDA approval. Okay, team D?
The law.
No, law, it's a law. Anybody? Yes, it's basically Kefauver-Harris amendment. This is a landmark amendment which came about right after the thalidomide tragedy, when the American government, the US FDA rather, realized that they need to do trials before they approve a drug in the market. It is a landmark decision. Now we'll come reverse. Now what I'm going to do this time, although it's said
Team A.
Team A, it's actually this side, okay? This question is for you. So I'm going to do something. I'm going to tell you the first letter. Okay, the first letter, the answer, the first letter of the answer. Okay, this is the clue not only for you, for all teams. Okay, so are you ready? This is your question. Which pharmaceutical company was formed by the merger of Ciba, Gigi and Sandoz, which were two companies? Okay, do you want the clue? The first letter? Should I say the first letter? Okay, it starts with the letter N. Okay, the answer starts with the letter N. Novartis is the perfect answer. Team E gets 10 points. It was now the question for Team D. Which is the largest provider for non-governmental funding for scientific research in the world? And this is a UK-based organization. The answer begins with the letter W. This is for Team D.
Based in London.
Based in London. Yes? Okay. Oh, sorry? No. Team B?
Welcome Trust.
Perfect. Welcome Trust is absolutely right. So Team B will be getting 5 points for that. The question to Team C, right?
Question for C.
Yeah. Which was the first company to commercialize manufacture of insulin? This is for you. The answer begins with the letter E.
No negative.
Sorry? England. Company.
Company, name of the company. Company that starts with E.
Passing. Team B. Passing. A.
Eli Lilly.
Eli Lilly is the correct answer. So Team A gets 5 points for that. And now the question for Team B. Which is the oldest operating pharmaceutical company in the world? The answer begins with the letter M. Merck, absolutely right. Team B gets 10 points. And the last question to Team A. Name the pharmaceutical company which is involved in the largest ever payment of settlement for false claims. The answer begins with the letter G.
Glenmark.
Glenmark, I am afraid you're wrong. It should go to Team E, right?
Yeah, this way.
Team E?
GSK.
GSK is absolutely right. 5 points to Team E. With this, we are done with this round. So clap for all the 5 teams. I request Dr. Reshma to read the final scores of all the 5 teams.
The final scores after round 4, Team A at 22.5, Team C at 25 points, Team D at 25 points, and Team E at 25 points, where we have Team B at 35 points.
So the exciting round.
Okay, now is the time for, you must be wondering why there is a bulb kept in front of you. So this is for the buzzer round. So now is the time to exercise the muscles of your hand and be ready. Only caution is, only caution for this is, there is a price you pay if you get it wrong. Okay, this is the only, or shall we cancel the negative marking?
Yes.
We'll cancel it? Or you want the negative marking?
No, let it be. Let the negative marking be there.
Okay, so then everyone says we should have negative marking in popular demand. There is a lot of popular demand for negative marking. In fact, it should be there so that this is well controlled. So we are going as per schedule. There is going to be negative marking.
There are 8 questions.
8 questions, it's not 5. 8 questions. So if you know the answer, press the buzzer immediately. Okay, and the team which gets it right, they are gets it first rather, will be, their bulb will be glowing, the buzzer will work.
For the audience, there won't be any conflict because only one bulb is going to get started. Even if all 5 starts pressing, only one bulb is going to get lighted. So there is no controversy.
Okay. Yes. Minus.
Negative marks. Negative marks.
You press it. It's an immense responsibility.
And wrong answer will be passed on to the audience.
Yeah, wrong answer, no passing, it goes to the audience. Okay? So shall we start? Okay, which was the first COVID-19 vaccine to be approved by WHO?
Your 30 seconds. Yes, Team E. Team E.
Covishield, unfortunately, you get -5, right? Minus 5? -5 to Team E. It passes right to the audience. Pfizer BioNTech vaccine is the right answer. Doctor, please give doctor the gift. Pfizer BioNTech. So we go to the next question. The 2021 Nobel Prize for Physiology or Medicine was given for which discovery? 2021 Nobel Prize. What was it given for?
It's 2021, not 2022.
Not 22, 21.
Seconds. Time of 30 seconds.
Okay, I will give a clue.
Give a clue.
I'm going to give a clue. It's something related to physiology. Something that you read in first year. They teach you about it in neurophysiology.
Maybe senses.
Something to do with the senses.
Now the clue cannot be booked.
Yes, Team A.
Pain transmission.
Pain.
Physiology of pain transmission.
Physiology of pain transmission. I'm afraid the answer is temperature touch receptor discovery. So -5 to Team A. Anyway, good, you have tried. Now we go to question 3. Name the drug approved by US FDA in 1999 that led to an increased risk of myocardial infarction, which led to its withdrawal within 5 years of it entering the market. Okay, I'm going to give a clue. It starts with the letter R.
Team B.
Rapacurium.
No.
Curonium.
No, I have to give you -5. Audience, yeah, that gentleman. Rofecoxib is perfectly right. Please give it to the gift to the doctor. Okay, so this is one of the greatest tragedies. Thousands of people had sustained MI heart attack, and they had a lot of claims from the company which released this drug, Rofecoxib. So we will now go to question 4. Name the pharmaceutical company which had a major patent dispute in India with one life-saving drug for cancer, that is imatinib. Okay, anyone is clicking? If no one is clicking, I'm going to give a clue. Okay, the clue is it begins with the letter N. Who's going to take the risk? Come on, you should be risk takers in life. No? Nobody wants to try? Nobody wants to try.
Press the buzzer, press the buzzer.
Who? No art is the right answer. Team E gets 10 they get, right?
Yes.
10 points. 10 points straight in the team.
You should have pressed the buzzer.
It starts with the letter B. It is there in one particular state of India. Its name would be there in the company's name. It is actually started by the government of India. It contains the name of one particular state which is in the western side of India. Eastern side of India. I'm sorry. Anybody?
30 seconds.
You tell the name of that state along with common words used in for pharma companies. You might guess it.
Audiences are eager to
No,
answer.
audience is eager to answer.
I think we'll pass out to the audience.
They want some more clues, I think.
So time is over.
Okay, someone has pressed the bell.
Nobody.
Did someone press?
Nobody.
You pressed?
No one.
Okay, anybody from the audience this side?
Time.
Sir, please tell.
Press the bell and say West Bengal. Yes. I pressed it. No, I said West Bengal, then said Bengal. I said West Bengal.
What do we do? No.
The final
Afraid we
decision-
cannot give you. The answer is Bengal Chemicals and Pharmaceuticals. Okay? Okay, so the next question. This drug became famous after being...
He pressed the buzzer. Team A.
Michael Jackson.
That's right.
He's dancing to victory. It's Michael Jackson. Unfortunate death because of an overdose of propofol. Though there were many drugs found in the post-mortem report, but propofol was the main agent which supposed to have caused his death. Michael Jackson is right. They get 10 points.
Sir?
No.
They don't.
Not minus, not plus.
It's just annulled. Here we see a couple. Okay. Name the contribution of these scientists. Okay, they are scientists and they have contributed something. What did they contribute? What are they so famous for? Okay, I'll give a clue, Dr. Anand.
Yes.
Okay, it is something to do with something we have all been struggling for the last 3 years.
COVID vaccine.
You have to be more specific than that. Which vaccine? There are so many vaccines.
mRNA vaccine.
Which mRNA vaccine? There are two prominent mRNA vaccines. Which of them?
Modern.
Okay, it's Pfizer BioNTech vaccine. BioNTech company, they are the co-founders. They have discovered COVID-19 BioNTech vaccine, subsequently acquired by Pfizer. And these scientists, so I will ask the judge. So they have said mRNA vaccine, COVID-19 vaccine, but instead of Pfizer vaccine, they have said Moderna. What do we do, sir? Should we give them 50%? 50%. Okay, 5 points to team A.
This is last question of buzzer.
Okay, the last question of buzzer. What is this operation all about? what you see, Operation Warp Speed. What is this about? I'll give you a clue. It's again related to what you see in the center, which is COVID-19. So what is this? Something that was done to facilitate something. To facilitate the entry of dash into the market. What was the dash?
Timekeeper, 30 seconds.
Nobody wants to take a risk.
It goes to audience.
Okay, I will give this to the audience. Yes, ma'am. Okay. So what exactly happened? Okay, basically it's a US public-private alliance started by the US government where they aggressively funded the pharmaceutical companies which were doing vaccine research. Almost 10 billion US dollars were released. Is something that never happens. They had released to the major companies which were involved in vaccine research, and that is how Moderna and Pfizer got a boost in their drug development. So with this we come to the last round, which is the rapid fire round. I hand over to Dr. Anand, but before that, the scores from Dr. Reshma.
The scores after round 5 are Team C at 25 points, Team D at 25 points, Team B at 30 points, Team E at 30 points, and Team A at 32.5 points.
Tough fight, isn't it? So now this is the last round. Either you win or you have difficult time. This round is rapid fire. Please listen to the rules very carefully. I'm going to start with Team A. You will have 1 minute to answer as many questions as. Maximum questions are 7. Okay? If I ask you a question, if you know the answer, you answer. If you answer correctly, you will get 10 points. If you answer wrong, there will be negative marking. Okay? If you say pass, and I finish the 7 questions, and you still have time left, you can revisit the question if you have passed before. Is that clear? I'm starting with Team A, and then I will come this.
There's some doubt there. He has a doubt.
Yes.
No negative?
No, okay. So no negative marking. I'm so sorry. So no negative marking, you can still take a chance, but if you answer wrong, you cannot revisit the question. Okay? If you pass, you can still revisit.
Any questions from the teams? The rules are clear?
Rules are clear. So I'll start with Team A. Question 1. Question 1. What is the mechanism of autosiban? Name oral iron chelating agent for thalassemia patient.
Deferipiro.
That's correct. Name a drug used for the treatment of visceral leishmaniasis in India.
Pass.
Name antihistamine available as nasal spray.
Pass.
Name NMDA antagonist used in the treatment of amyotrophic lateral sclerosis.
Edavaro.
Wrong. Go no-go decisions are taken after which phase of a clinical trial?
Pass.
The last question. Name the country which used yellow card system for ADR reporting.
UK.
That's correct. You want to revisit any questions? Time is over. So they answered 2 correct questions right. Team B, are you ready for rapid fire? Okay. So I'll take this for audience. Those questions which are not answered. This question, anybody wants to answer? Yes, it's an oxytocin receptor antagonist. That's correct. This was answered by the team. Anybody for this question? Urea stibamine. Urea stibamine is the right answer for this question. Anybody for this question? There are two antihistamines, one is azilastine and second is olapatadine. NMDA receptor antagonist. Riluzole is the right answer, correct.
Phase 1.
Anybody apart from phase 1? Phase 2 is the right answer. Phase 2. This they answered. Team B, are you ready? The first question. Which derivative of ticoplanin is given once a week?
Pass.
Name the biphosphonate that can be given as a once a year injection for osteoporosis treatment.
Pass.
Which is the other name for fatal warfarin syndrome?
Can I go back to the previous? Can I go back to it? Aldrinone.
No, that's the wrong answer.
Pass.
Who is called as father of modern pharmacology?
James Black, Sir James.
No, that's the wrong answer. How much is the osmolarity of new WHO ORS?
308, 45, 308.
Pass. Incidence of patient's availability sharply decreases when trial begins and returns after trial completion. What is this phenomenon called as? Time is over. I'll go back from the question 1 for all the questions to audience. Which is this derivative which is given once a week? The answer is dalbavancin. That's correct answer. Zoledronate is the right answer. It's called as chondrodysplasia punctata, which is also called as the Conradi syndrome. It's a milder form of Conradi syndrome. That's correct answer. 245 milliosmol per liter is the right answer. Anybody from the clinical research who answers this question? Melvin.
It starts with the letter L. Okay, it's Lasina's law.
Lasina's law. Drug which is derived from a plant Pacific yew. That's the right answer. Paclitaxel. Team C, are you ready for the rapid fire? The first question for Team C. Which ion preparation can interfere with MRI?
Next.
What is the other name for centroman?
Saheli.
Come again?
Saheli.
No. Name neprilysin inhibitor used in the treatment of congestive.
Sacubitril.
Sacubitril is the right answer. What is the other name for hydrophonium test which is used to differentiate between myasthenia gravis versus cholinergic crisis?
Next.
No promptings from the audience, please.
Name the monoclonal antibody targeted against RANK-L used in the treatment of osteoporosis.
Denosumab.
That's the right answer. Which organization was previously termed as Indian Research Fund Association?
ICMR.
That's the right answer. How many chapters are present in NDCT 2019?
Next.
Time is over. So I'll take the questions to audience. Let's start with first question. Which ion preparation interferes with MRI? The answer is ferumoxytol. Which is the other name for centroman? Or meloxifene? This they have answered. Penicillin test is the right answer. They have answered this question. They answered this question ICMR. How many chapters are there in NDCT? 13. NDCT has 13 chapters, 9 schedules and 107 rules. Team D, rapid fire. Are you ready? The first question. Name an example of nitrates given by inhalation.
Pass.
What is the other name for folinic acid? Audience, please keep quiet. Audience.
No promptings, please.
Pass. Name orally active lignocaine derivative which is used as anti-arrhythmic drug. Which ergot alkaloid is associated with risk of pulmonary fibrosis as an adverse event? Which 5-HT receptor is ligand-gated ion channel?
5HT1.
No, wrong answer. Which Indian organization related to research was established in 1911? Name the organization which controls drug pricing in India. You want to revisit any question? Time is over. Audience, the first question, what is the example of nitrate? Amyl nitrate is the right answer. Leucovorin is the correct answer. Leucovorin. Mexilendin is the right answer. Methisergide is the correct answer. 5-HT3, that's right. ICMR is the correct answer. Yes, NPPA, somebody said NPPA is the organization which controls pricing in India. Last team, last round, last 7 questions. Are you ready? The first question. Name selective alpha-2 blocker which is used in preclinical models for seizures. Which class of drugs are associated with Monday disease?
Nitrates.
That's correct answer. Which event led to the development of Belmont report in 1979?
Pass.
Alfred G. Gilman received Nobel Prize in 1994 for medicine. Name the discovery.
Pass.
Which direct acting nicotine agonist is used as anti-smoking agent?
Pass.
Which drug is the choice for therapy of hairy cell leukemia?
Pass.
Pass. Which is the most famous contribution by Gamaleya Research Institute of Epidemiology and Microbiology in recent times? You want to revisit any question?
666, question 6, please.
Audience, the first question. Yohimbine is correct answer. Nitrates, I think they answered the nitrates. Tuskegee syphilis study, yes, that's right. Correct answer, G protein coupled receptors. Varenicline is the right answer. Anybody for this answer? Somebody said cladribine is the right answer. And the last one. This is Sputnik vaccine. Thank you all the teams. Can we have the final scores of the quiz?
So, we come to the end of the 6 rounds. A big round of applause to all the 5 teams. So, as can be displayed, we see Team B with 30 points, Team E with 40 points, Team D with 25 points. Okay, so team at 3rd place is Team E with 40 points. Team E, where are you? Okay, here. Okay, 3rd place, Team E. At the 2nd place, the runners-up is Team A with 52.5 points. And Team C emerges winners with 55 points. Clap for all teams, please. Clap harder. So at this point, we would like to specially thank our mark scoring person, Dr. Reshma. And we also want to thank all the volunteers. Great efforts that you have done to make this a successful one. And thank you, Dr. Chandralekha, for coordinating with us and helping us with all the arrangements, the sound arrangements, the team. Thank you all. And a special thanks to the organizers of this conference of NAPTICON 2022. It's an amazing privilege you have given myself and Dr. Anand to be part of. Thank you all.
Thank you.
Hope to see you next time with greater performances. Thank you.
Thank you. Thank you, everyone. So are we...
Yeah, congratulations winners. Please be here. It's not over. Yeah, first of all, I would like to call all the participants to receive participation certificate. I'll call the names. So I request our quiz masters to give participation certificate to them. Dr. Anand Patil and Dr. Melvin George. As I call the names, please come and collect your participation certificate. Dr. Mahesh and Dr. Abraham Joseph Kodikal from AIIMS Raipur, Chhattisgarh. Dr. Soham Sinha and Dr. Komal Kurade from Seth GS Medical College, Mumbai. Dr. Arvind and Dr. Christy George from Government Medical College, Kottayam. They left? Yeah. Yeah, please come and take your participation certificate. Dr. Palak Vania from Government Medical College, Bhavnagar, and Dr. Aishwarya Vodaya from Kshema, Mangalore. Please, quiz participants, be here, don't leave. Dr. Tamil Selvi and Dr. Abhirami. Dr. Tamil Selvi is from AIIMS Nagpur and Dr. Abhirami is from Tirunelveli Government Medical College. So now we are going to distribute the prizes. First prize, so please participants, Dr. Tamil Selvi and Dr. Abhirami, please be on stage. I request our quiz masters to give away. So we have a trophy and a cash prize of 7,000 with the certificate to the winners. Yeah, please. The winner certificate. Yeah, second prize, Dr. Mahesh and Dr. Abraham Joseph. Request Dr. Padmaja Uday Kumar and Dr. Kamal sir to come and give away the mementos to our quiz masters. Dr. Anand Patil.
please give a big hand to the quiz masters. Number one in the country probably, in my opinion. They have taken this activity, quiz activity, to almost next level. Probably you have all we have experienced it together. In such a nice way they have conducted this quiz, and this quiz I'm literally getting so many messages. Please send me the pictures related to quiz. I immediately posted. You will be able to follow in the WhatsApp group. People are literally wanted to see how the quiz is going on. It is a marvelous success, and I would like to congratulate all the quiz masters for this success. Thank you very much for being us and for accepting our this invitation. You can continue.
Yeah, ma'am, please give it to Dr. Anand Patil, sir. So please hand over to the Dr. Melvin, sir. And also I request ma'am and sir to give away the memento to our judge, Dr. Naresh Jyoti. Thank you, Padmaja ma'am. Thank you, Kamal sir. Thank you so much, quiz masters, for taking up this responsibility and making this the first NAPTI quiz 2022 a big success. I know I could see the audience, the big number, and thank you, participants. Congratulations, winners and runners once again. Thank you.
Most welcome. Please. From GPM, sir will give introduction and some suggestions also.
Thank you, sir. My name is Dr. Sushil Sharma. I'm from AIIMS Mangalore. And sir, I have a suggestion that we should have a couple of prizes. Like we could have certain sponsorships. If one of the members or somebody is willing to sponsor a prize, and so we can start some prize, maybe one prize on maybe medical education section or any other prize, and we can ask people to sponsor, and the name can be given by the person who is sponsoring the prize, and that will be a good start, sir. That I think we should have that, sir.
Point taken.
Myself, Dr. Pramod from AIIMS Patna. Thank you, ma'am and sir, for wonderful conferences. I would like to suggest this NPT. It is a nice organization. So yearly we will do this NAPTICON, but I will suggest that on quarterly basis we will focus some aspects of pharmacology, like some at least in a webinar like series we can do some educational activity on biostats, GCP, some webinars on animal experimentation, which are the core pharmacology, as well as therapeutics. Okay, so it every quarterly basis, like there are 4 papers in pharmacology. So one focus on the pharmacology, second will be on the screening part, third on the animal experimentation, fourth on the biostats and recent advances. So by this, the society, our society will be active on quarterly basis, as well as more members will know about ourselves and they will join us. Thank you, sir.
Thank you, sir. Your suggestion is well taken, and probably I would like to inform you again that already we are involved in doing activities like GCP training and competency-based medical education. Probably you want to convey that we need to increase its frequency.
Yes.
We need to include more of the topics.
Yes.
Your suggestion is well taken. I would like to request our Joint Secretary sir to note it down.
Thank you, sir.
Yeah.
Hello. In Dr. Sharma's context, I want to add one thing. We can have an oration. Oration is a lecture of a senior professor or senior colleague, senior pharmacologist, and we can name it oration on some scientist of our pharmacology field and all. So this we can start a oration.
Okay. We have already started one prize you are already aware of, Lifetime Achievement Award that is awarded by the National Association of Pharmacology and Therapeutics. As far as the oration is concerned, we are looking at sponsorship options also. If there would be sponsorship options also, we will consider it. And touching the name of the scientists in the name of the orations, you know it, there is a pandara of dispute also. Some of the people are saying why you have started. That's why even Lifetime Achievement Award, there was a suggestion to start on the name of a very senior pharmacologist. But in the council meeting, we have thought it better that we will be giving it on the behalf of the association, because there is no dispute in the name of the association. That is one factor. But if somebody is offering us the sponsorship option, we will definitely consider it.
Sir, my name is Dr. Ajay Gupta. My suggestion is, when we are organizing such a big program, we must have a feedback form or some e-feedback.
We are having feedback form, e-feedback as well as hard copy. I think they'll be giving it to you very soon.
Okay, ma'am. अगर आपकी परमिशन हो तो एक शेर भी है. आपने बढ़ा दिए हमारे खर्चे शराब के, नाम ही हटा दिया वहां से जहां जरूरी था. For A1 Logistics, रजिस्ट्रेशन में से नाम हटा दिया.
What is this?
Good afternoon, everybody. First of all, congratulations to the organizers, Madam and Sir, and all the team for a wonderful conference, first conference. I am Dr. Shashidhar Banare from Bagalkot Medical College, Karnataka. Just one suggestion, since it is our national body, we can communicate to our MC or currently NMC, because during whenever there is a staff recruitment, there is one of the criteria like marks and all. Those who got a gold medal, they will get a 1 point. But there are universities where there are no gold medal for MD pharmacology, but there will be person with a first rank, but they don't consider it as a equal. There is some gray area. So we can, as a national body, we can communicate to the NMC on this issue, sir.
Yeah, your suggestion is well taken. Madam will also elaborate.
Yes, we can do it. Anyone else? Any other suggestion?
I'm Dr. Shivamurthy, Associate Professor, Bangalore, Chandrama Dayanand Sagar Medical College. Actually, while having the student session, one of the students said that therapeutics and involvement of pharmacologists in the treatment aspects can be initiated like that. At that time, even I am the coordinator of pharmacovigilance. And in the last 2 years, I have accumulated good amount of information about various adverse drug reactions and their treatment methodology and signs, symptoms and all those things. So over a period of time, we will be experts in which drug produces, what adverse event, what is the expected, and how to treat. So if we can take this as a lead and then go and propose to the management saying that we would like to serve as the physician, pharmacovigilance physician, and take the responsibility of managing these adverse events so that we can take out some chunk of clinical cases in our sector. In that way, we can directly start doing the emergency services and doing the treatment and taking the responsibility of even reporting already we are doing. So we can come up with, in this way we can become a service department. Maybe association can think in this way, so that we are already having 500 medical colleges are having AMCs. So in this way, we will become a service organization, we will become clinicians. Not only, like it's now considered as non-clinical. We get hurt when people, call us as non-clinical people. We are clinical, we are only paraclinical. But, like in this way we can become clinical and our pharmacology can get a very good hold.
Point taken. Point taken, sir. He has noted. Thank you. Any another suggestion from the general body? We will be waiting for 30 seconds, and after 30 seconds there is no question is coming. We will be considering that there is no suggestion. And as far as feedback is concerned, Madam has announced that there is a hard copy of feedback and feedback in the e-form that can be accessed through the website and WhatsApp also. A1 Logics have made a website and they have put a feedback. We are eagerly waiting your feedback. Your feedback will improve to organize these programs further. We will be waiting 30 seconds. After 30 seconds, if no suggestion is coming, we will be concluding this general body meeting. Thank you. Soon we are starting the state branches also, but since it's the upcoming organization, and you will agree with me, those people who are quite senior, those who are experienced one, they know it very well that to register an organization, to shape up of an organization, to reach a certain stage, Dr. Sushil Sharma will be able to explain you in a better way. He was discussing me. I was getting his phone call. He was telling me maybe compliments that the kind of the activities and we have rose it at least for ordinarily an organization take at least 10 years and we are able successfully doing it in a 1 year only. So definitely we will be take all the major activities like state branches also, but it will take some time.
Is it possible to merge all and make only NPT like that?
We can discuss this point among the executive council through an online meeting. It is going to be organized next month. We will keep the proposal and you can send me the proposal through direct WhatsApp, email or phone calls. Definitely in your name I will forward this to governing council as well as the executive council, and whatever their feedback we will be conveying you. So thank you all, and in a diplomatic way, in a diplomacy and in a democracy also, to give opportunity to all, I feel we are giving good opportunity to speak to us. We are taking all the suggestions well in advance. We are working on it. But you will agree with me that an organization to need to grow takes some time. If you are keep on cooperating us in this way that you have cooperated in the past 1 year, I'm fully confident it will grow like anything. And Madam has again ensured it that we will be dedicatedly working towards it. Thank you.
Thank you everyone for your suggestions and for your presence. Thank you.
Dr. Shirish Joshi is an additional professor at Department of Pharmacology and Therapeutics, Seth GS Medical College and KM Hospital, Parel, Mumbai. He is a joint member secretary of IEC, member of Transplant Authorization Committee, Nodal Officer for COVID Vaccination, lifetime member of ISER and Association of Medical Consultants, Mumbai, accredited speaker of Maharashtra Medical Council and also member of the Indian Pharmacological Society. He has delivered several guest lectures, conducted plenty of workshops and conferences, has multiple publications along with being an enthusiastic sports person. We welcome you, sir.
Dr. R. Ratish is Associate Professor, Department of Pharmacology at KMCT Medical College, Calicut. He is a renowned exuberant pharmacologist, industry consultant for pharmaceutical firms, and TEDx guest speaker at various medical colleges. He completed his MBBS from Jawaharlal Nehru Medical College, Belgaum, and MD from Kanachur Medical College. He has many original articles in famous peer-reviewed international journals and has presented research works in distinguished international conferences. He was also instrumental in controlling the COVID-19 pandemic in association with Honorable District Collector's Office, Kozhikode, being the nodal and liaison officer for 450-bedded KMCT COVID Hospital, Kozhikode, Kerala, as well as nodal officer for COVID data management. He was actively involved in ATR monitoring in the COVID hospital. His special area of interest is pharmacovigilance and eco-pharmacology. It is indeed a great pleasure to have you with us today, sir. I request our honored chairpersons to kindly take stage.
We have with us our first speaker, Dr. Aisha, Assistant Professor, Department of Pharmacology, Government Kilpauk Medical College, Chennai, whose talk is on DARPins.
So, good afternoon, respected dignitaries on the dais and off the dais, the remaining 5 speakers of the YPS session, and our supporters, of course, and the organizing team, not to forget them. A very good afternoon.
I think you are aware of the rules, 8 minutes for presentation,
Yes,
2
sir.
minutes for question. So you can proceed with the topic.
Okay, sir. Thank you. So, I'm going to talk on DARPins. So, I would like to ask a question. Have you heard of DARPins before? Okay, I will take it as a no, and I like to continue. See, DARPins are nothing but designed ankyrin repeat proteins How does this work? Sorry, it's not working. Yes, so designed ankyrin repeat proteins. So
Technical team, can you help her, please? We are already overshooting, short of time. And
So designed ankyrin repeat proteins are a new class of potent, versatile small protein therapeutics. These are going to revolutionize the field of not only research but also diagnostics and therapeutics. And these are going to be the real-time sensors of protein conformations, and they also act as crystallization chaperones. Crystallization chaperones, I would like to tell one thing, that they are going to be used as a binding protein by a method of crystallography. So DARPins, they can be agonist, antagonist, inverse agonist, enzyme inhibitors, or simple binders for the target proteins. You name any protein in the body, A or X, you can make a DARPin act against it. Okay, it can be either an agonist, an antagonist, an inverse agonist, an enzyme inhibitor, or a simple target. So DARPins are nothing but genetically engineered antibody mimetic proteins. especially the advantage of these DARPins is going to be high specificity and affinity for target protein binding. I'll tell about this in the later slides. And I told you that these are going to be sourced from the natural ankyrin repeat protein. So what are these natural ankyrin repeat proteins? These are nothing but the proteins which naturally occur in the body. They are responsible for the normally occurring cell-to-cell interactions and regulation of cellular function. So these DARPins actually originated before 30 years ago approximately, and they were first manufactured in the laboratory of Andreas Plukten, University of Zurich, Switzerland. So we'll see a bit about the structure of the DARPins. DARPins, they are going to have at least 3 repeats or the internal modules, which is going to be capped on the either side by the N and the C terminal modules, and this N cap and C cap is going to protect the inner hydrophobic protein core. So N2C DARPin is going to have 2 repeats, whereas N3C DARPin is going to have 3 repeats. DARPin has to have at least 3 internal repeats for it to be stable. Okay, a DARPin has to have a 3 repeat to be a stable tertiary structure. And the molecular mass of the DARPin is usually going to increase with the number of the internal repeats, and the stability is also going to increase with the same. That is, the increased internal repeats will be associated with increased stability and molecular mass. So this DARPin, the speciality of DARPin is it's going to have a very large interaction surface or the target binding surface, which is its advantage. So DARPins, because they are going to have a very small molecular size of approximately 18 kilodaltons, it is very small when compared to a monoclonal antibody, and hence this is going to be an advantage for its easy penetration into the tissues. And also because it's small and it's going to have only the internal repeats, is going to be very highly stable even for any high temperature. And specificity, it can just go and bind any target protein in the body. It's going to be high potent, that is potent at very low concentrations, and affinity is also going to be very high. And because of the 3D structure, it's going to have a robust folding and a flexible architecture, and its tendency is it cannot aggregate. So that is why it's going to be stable. So one more point in this, it's going to have a rapid and a cost-effective production, which means it's going to be produced in vitro in the laboratories, and you can have a very large yield. And adjustable pharmacokinetic parameters, which means that you can just tune the pharmacokinetics, you can increase or decrease the half-life of any DARPin by combining it with a pegylated structure, pegylated form, or you can either combine it with a human albumin to increase the T-half. So there are so many recent advantages in DARPin design and properties illustration that has been done nowadays. So I would like to remind again that this is going to have very high affinity and specificity, and again multi-specificity, because it's going to act against any target. And a small size, again it's going to have easy tissue penetration, simple high yield manufacturing, one DARPin which is going to have one function and a long shelf life and stability. Again, because it's not going to aggregate. So DARPin generation, you usually generate DARPins from libraries via sequence alignment of several thousand natural ankyrin repeat proteins, about 33 amino acids, which is combined with structure-based design or recombinant DNA method. So this is going to be the first step where a DARPin library is going to be selected, okay, with over 1 trillion different DARPins. You are going to select a DARPin which is going to have affinity for the target. So the gray one is the target, the upper one is going to be the DARPin library, and you are going to select the DARPin. And creation of multi-domain DARPins, which means you can have many DARPins. I told you one DARPin is going to correlate one function, so you can combine as many DARPins as you want by means of a linker, so that you can affect any pathway, any disease pathway, which is mostly because of different pathways, whereas conventional methods usually target only single pathway. And then you can add any therapeutic platform which you want to make the DARPin finally to have a tunable half-life, or you can immunomodulate it, and then you have the winning therapeutic design, and this is how a DARPin is selected. So types of DARPins, it can be either single domain DARPin or multi-DARPin, which is again single domain DARPin will have only one DARPin molecule, whereas multi-DARPins will have many DARPin molecules which are linked together. So I would like to just reiterate by saying that DARPin applications is especially in cancer chemotherapy, multiple myeloma, refractory multiple myeloma, and non-squamous cell carcinoma, and it is going to act as an antiviral therapy. And
please do conclude in.
Yes, sir. Yes, I am going to conclude. This is the last slide. So the applications of DARPin is going to be especially in cancer chemotherapy, multiple myeloma, non-squamous cell lung cancer, and antiviral against SARS-CoV-2 virus and COVID-19. It was also tried. Then you have in eye, we have two important conditions, that is age-related macular degeneration and diabetic macular edema.
Thank you, Dr. Aisha, for a nice presentation. Session is open for the questions. Any questions? Dr. Aisha, what is the current status of DARPins from FDA?
So currently only two DARPin candidates have been tried, so only have been come out. One is the DARPin which is used for, it's called as Abicipar, which is used for wet AMD, but still it's pending FDA approval because of the intraocular inflammation it causes. Its approval is pending, and we have a second candidate which is, the number is going to be MP040, which is tried in multiple myeloma and non-squamous cell lung cancer. The first candidate is used for wet AMD and diabetic macular edema as intravitreal injection. It's long acting, but still it's pending FDA approval because these trials have been non-inferior to the conventional therapy, that is ranibizumab, and intraocular inflammation is going to be a problem with this DARPin. But still there are so many DARPins and which are in preclinical stages of development, and this will definitely revolutionize the treatment of cancer chemotherapy because it can be used as agonist, antagonist, inverse agonist. You can use it as just for a targeted drug delivery, or you can just act as a chaperone, that is crystal chaperone.
Thank you. Thank you.
Yes, thank you for your patient listening. I would like to leave you all just a minute, sir. It will not take a minute. I will leave you all by saying a quote from Thiruvulluvar. Yes, 1 minute, sir. Vellathanaya malarneetam mandartham ullathanayadu unarvu uyarvu, which means the stalk of the water flowers are proportionate to the depth of the water, so its men's greatness are always proportionate to their minds. Thank you.
Thank you. I now request Dr. Shirish Joshi to kindly hand over the certificate to our speaker, Dr. Aisha. Thank you, sir.
The next speaker for the session is Dr. Venu D, Senior Resident at JSS Medical College, Mysore, who will be talking about diabetes without needles, advances in insulin delivery.
Good afternoon all. Myself, Dr. Venu. I am from JSS Medical College, Mysore. So my topic is advances in insulin delivery. So we all know the diabetes is the one condition, it is an intractable chronic disease manifested as high blood glucose levels. So we give exogenous insulin supplement is essential for both type 1 and type 2 advanced cases. Sorry. Okay. So we know insulin taking is very hectic and painful process. Most of the patient will feel it as irritating. So there is always a search for the painless and less painful measures from our pharmacology and therapeutics. So we came across so many types of administration. One is nasal administration, pulmonary administration, oral administration, and transdermal administration. So I want to discuss about the insulin pumps and their evolution. First, there was a tethered pump where the insulin is stored in a compartment, then it is given through the cannula. It was attached to the skin through the transdermal patch. Then came the patch pump. In this, we can see there is a insulin storage is also kept in the transdermal patch only, so that the delivery will be easier. And the next evolution was the how much amount of the insulin should be delivered. That was connected through a mobile app, and it was monitored through the Bluetooth. So when we come to the insulin pump, we want to know why insulin pump is better than any regular insulin. The one thing is, we can see when we give a insulin, the basal insulin, and when the patient is on his own routine, like that in the morning he will take the breakfast, his sugar levels will be increased. So the basal insulin will not serve the complete purpose as our physiological insulin. So when we give the insulin pump, it can alter the dose of basal insulin so that it will match with our bolus of food, so that the glucose alteration in the body is smoothened. Then comes with the exercise. When patient do a hard work or some exercise, his blood glucose levels will go low. So when the blood glucose level will go low, he might feel hypoglycemia. So that when we give insulin pump, his basal insulin can be decreased so that it will get adjusted to his normal levels. Then comes in the diabetic patient, there might be a chance of gastroparesis, right? So that is a counterpart of our diabetes itself. So in that, the glucose levels will reduce in the body slowly. So it is not very normal like any other diabetes patient who is not having gastroparesis. So in this patient, the insulin pump will act in a different way, and it will minimize his blood glucose levels. So after this, there is a evolution. Then comes the sensor augmented pump therapy, where the sensor will count how much blood sugar level is there in the body, and then the insulin pump can be given with a adjusted dose. This is done manually. The next one is hybrid closed loop insulin therapy. In this, the mobile or any device which will sense the glucose level in the body, later the data is transferred to the insulin pump, and this data is transferred and analyzed by the what we call the algorithm, where it will calculate how much insulin should be given on its own. So it's all computerized. It's called closed loop. There is no intervention manually. We call this as automated insulin delivery or artificial pancreas. In the later studies in NEJM, I saw this article where it's telling randomized trial of closed loop control in very young children with type 1 diabetes. So this study shows closed loop is working better than our sensor augmented insulin device. So in the closed loop, if you see, there was a 72% time if the target glucose range was achieved, better than sensor augmented pump period. Then time of hyperglycemia was decreased with a closed loop pump. So the next one is another article which from Science Direct. Even this article shows closed loop pump was better. When you come to the closed loop, nowadays there is advancement where it will measure long-term HbA1c management. So we can achieve a relaxed control or a tighter control, and it will take the dynamics to consideration, that is higher insulin sensitivity or higher insulin resistance. Then come to the different groups. When we say the insulin pump, it should be different in variability in location for pediatrics, pregnancy and athletes, and the location of the insulin pump can be decided. And then later on, there is an advancement that these additional sensors, such as for some other hormones like cortisol and melatonin, so that shift of the work and the routine of the patient can be monitored, and accordingly the glucose level can be altered. Then comes the monitors are different for pediatrics and athletes, and the lactate sensor for athletes. So these all advancements made the control of the sugar very tight and we can make the aggressive control. So these are some few insulin devices in the market. Then there are variety of insulin sensors which are chemical based. The chemical based are the glucose oxidase, then PBA and concovalin A. Among these, glucose oxidase is considered as very authentic glucose sensor for the sensory devices. Then comes pros. What are the positives? It is convenient for the patient, then it's consistent and adjustable insulin delivery and fewer insulin injections, the flexibility and privacy and improved blood sugar levels. And alarms if a person below their preset glucose value, then waterproof protection. And always any device will come with some cons, that is inability to hide the tubing or pump and higher cost than the injections, pumps breaking and tubing became disconnected. And apart from that, infusion site infection can happen, erythema, induration, tenderness to palpation can happen. Apart from that, hyperglycemia, diabetic ketoacidosis, hypoglycemia can happen like any other OHCs. But the thing is, this is always better than any other diabetic treatment. The insulin pumps are far better and they won't cause these complications. Then comes the implanted insulin pump, which is already not much entertained these days because it's of difficult procedure to install. Then I went through some peculiar mechanism which is tried for insulin delivery. As we know, we cannot take insulin orally. There is one discovery made by Harvard Medical School. This was actually inspired by the tortoise, the sea tortoise, which will always stay flat. Like however you throw the tortoise, it will come to the normal position because of its shell shape. So inspired by that, they designed a shell and it will contain the insulin. And it's called self-orienting millimeter scale actuator, SOMA. So when we give this SOMA, it will go 1 second inside our stomach, then it will get located in the gut wall, like inside the lining of the gut. Then in 1 minute it will inject the insulin, in 1 hour it will dissolve. So in this way we can give oral insulin, and it was experimented on pigs. So the insulin uptake was almost near to the subcutaneous injection. When we come to the blood glucose drop, it was better than subcutaneous injection. So when we come to the economics, the insulin delivery device market is USD $24 billion, so it has very huge market. I will come to the conclusion. There is a big scope of research in this field, and participation of pharmacologists in giving inputs to the algorithm is needed. Then bringing cost of these devices, bringing down the cost of these devices, and vigilance of new pattern of ADR that can be happen because of these devices is needed. Thank you.
Thank you, Dr. Venu. The session is open for questions. Any questions? Dr. Venu.
Yes, sir.
What is the cost of patch pump?
Sir, patch pump is nearly, Indian market it is 15 to 20,000, sir, as of now.
Okay, yes. All right. Thank you.
Welcome, sir.
I now request Dr. Ratish to kindly hand over the certificate to our speaker, Dr. Venu. Thank you, sir.
The next speaker for the session is Dr. Manish Mohan, Senior Resident, Department of Pharmacology, Believers Church Medical College Hospital, whose talk is on GLP-1 receptor analogs, Basics to Guidelines.
I am Dr. Manish Mohan from Believers Medical College, Tiruvalla, and my topic of presentation is GLP-1 receptor analogs from basics to guidelines. Thus, GLP-1 analogs are one of the most discussed agents nowadays for the management of type 2 diabetes. So in this session, I will briefly introduce these drugs and will talk about some of the proposed physiological benefits, pharmacological properties, adverse effects, current guidelines and their suggestions regarding these GLP-1 analogs. So I would like to start with incretins. Incretins are hormones that are released from GI tract in response to the oral intake of nutrients. And incretin effect, it is the phenomenon where oral glucose eliciting a higher insulin release than IV glucose infusion. Here you can see that incretin effect is reduced in type 2 diabetes. Later researchers found out that incretin effect is due to two hormones because of GAP and GLP. GAP is also known as glucose dependent insulin releasing polypeptide. Previously it was known as gastric inhibitory polypeptide. GLP-1 is glucagon-like peptide. We already know the differences between GAP and GLP. Both these agents stimulate insulin secretion and promote beta cell proliferation, whereas GLP-1 inhibit glucagon secretion, reduce food intake, and stimulate insulin biosynthesis. The secretion of GLP-1 is reduced in type 2 diabetes mellitus, but its response is preserved, unlike GIP. Human GLP has a very short half-life because it will be immediately degraded by an enzyme called dipeptidyl peptidase 4. Next, I will discuss some proposed physiological benefits, what make them special apart from other agents. So the first one is they enhance glucose-dependent insulin secretion. Unlike some insulin secretagogue agents such as sulfonylurea, these release insulin only in presence of hyperglycemia. So there will be lesser episodes of hypoglycemia. Second point, they restore appropriate glucose-dependent glucagon secretion. Like beta cell, alpha cell function is also impaired in type 2 diabetes mellitus. During fasting and also post-meal, there will be an elevated glucagon level will be seen in type 2 diabetes patients. Such problems won't happen with GLP-1 analogs. And third point, it delay gastric emptying, especially short-acting GLP-1 analogs. And they improve satiety, which helps in weight loss. And in heart, it has anti-atherosclerotic effect through different mechanisms, such by reducing this metalloproteinase action. And these agents improve endothelial function. It has anti-inflammatory effect. Also, they modify risk factors. Also, they reduce infarct or injury size. Renal benefits is mainly through preventing new onset macroalbuminuria. So, come to the overview of drugs and pharmacological properties. Exenatide is the first approved GLP-1 analog, that was in 2005. Later, liraglutide approved in 2010, exenatide in 2012, albiglutide and dulaglutide in 2014, lixisenatide in 2016, and semaglutide in 2018. Recently, they launched oral semaglutide. It is the only one oral GLP-1 analog. All others are subcutaneous preparations. And exenatide was developed based on a protein called exentin 4, which obtained from a venomous lizard called Gila monster. It eats only 4 times a year. While fasting, it shut down the pancreas and stopped insulin production. But it's time to eat, it restarts its pancreas with a substance called exentin. Exentin is almost similar to GLP-1. And based on the duration of action, these agents are classified into short-acting and long-acting compounds. Short-acting include exenatide and lixisenatide. All others are long-acting compounds. These long-acting compounds strongly reduce fasting blood glucose and HbA1c, but short-acting compounds strongly reduce postprandial hyperglycemia and significantly decelerate gastric emptying time. And this graph shows the time to reach maximum plasma concentration. Here you can see albiglutide will take almost 96 hours to reach peak plasma concentration. Short-acting compounds reach their peak plasma concentration soon after administration. Dosing schedule is different for these agents. Here you can see semaglutide, oral semaglutide, we will initiate therapy with 3 mg and we can escalate the dose up to 7 mg, and if glycemic level is not controlled with that, we can escalate up to maximum up to 14 mg. But in case of dulaglutide dose, we can start and we can maintain with 1.5 mg once weekly. And these are the currently available pen devices for injecting this GLP-1 receptor agonist. Next, I would like to show you some clinical trial reports. Because of rosiglitazone, FDA made it mandatory that all anti-diabetic drugs need to undergo these cardiovascular outcome trials. As a class, these GLP agonists reduce almost 14% of cardiovascular events, 12% of all causes of mortality, 11% of heart failure hospitalization, and 21% of composite kidney outcomes. Here, if you look, you can see the upper row shows clinical trial names with different agents. Elixir trial with lixisenatide, Excel trial with exenatide once weekly preparation, Leader trial with liraglutide, Rewind with dulaglutide, Sustain 6 with semaglutide, and Pioneer 6 with oral semaglutide. A greater HbA1c reduction noted with oral semaglutide. And the drug in green boxes showed clinically significant report, especially in reducing MACE events. MACE events means major adverse cardiovascular events. And it is a forest plot showing the effect of GLP on composite renal endpoint. Leader trial with liraglutide, Sustain 6 with semaglutide, and Amplitude trial, that with FP glinatide, not approved yet. And what are the precautions and GLP-1 agonist precautions? Precautions include adverse effects are mainly GI adverse effects. The major cause that lead to the non-compliance is mainly nausea. And in preclinical studies using rodents, it resulted in medullary thyroid cancer. So these GLP-1 agents carry a black box warning. And pancreatitis is also an adverse effect. So these agents are contraindicated in patients with previous or family history of medullary thyroid cancer and in patients with history of pancreatitis. So guidelines. In guidelines, metformin is the first line of management. In case of metformin intolerance or metformin failure, we can start with GLP-1 analog, or we can add GLP-1 with metformin. So according to ADA, in patient with established cardiovascular disease, we can start with GLP analog, and also patients without established CVD with high-risk indicators, such as age more than 55 years, carotid lower extremity or coronary stenosis more than 50%, and in patient with left ventricular hypertrophy, EGFR less than 60% or albuminuria, we can use GLP-1 analog. According to European Society of Cardiology, they also recommend GLP as first line of management in patient with established CVD. Already discussed this is the latest ADA guideline. So what are the barriers of GLP-1 receptor analogs? First one is cost. If we compare the cost is little bit on higher side. Especially in a country like, developing country like India, cost is a major factor. And rather than daily or weekly injection, we prefer oral medication. Most of these GLP-1 analogs are subcutaneous preparation. Only semaglutide is available as oral preparation. Then contraindications and GI adverse effects. So I want to conclude by saying this is a wonderful agent with multiple mechanisms in case of metformin failure or intolerance.
So the session is open for question answers. Nerurkar sir, can you pass on the mic please?
Among all the GLP analogs you have told, semaglutide is actually peptide. Now it is very surprising that we have learned in physiology that peptides cannot be absorbed as such. So yet it is the only one which is given orally.
Yes, sir.
So is there any data about the semaglutide as to what is the bioavailability, half-life, etc.?
Half-life is almost 5 to 7 days, sir. 5 to 7 days.
5 to 7?
7 days.
That's the half-life?
T-half.
T-half. And bioavailability, because any data? How much is absorbed? Because proteins are not absorbed. I mean, this is what we learned in, that's why insulin cannot be given orally. It's a peptide. So this is also a peptide. I think it's very less. It's less than 1% which is absorbed. But this is the, exception to the rule. That's just a comment, not a question.
Thank you, sir.
Thank you.
My question is, amongst the drugs, GLP analogs, which you have mentioned, which of them are available in India for Indian patients?
Except albiglutide, dulaglutide, liraglutide, semaglutide.
So they
most
are
of them are available in India, sir.
And you not mentioned in the contraindications about pregnant women or lactating mothers. So is there any specific contraindication to that?
No, sir. Specific contraindication in pregnant ladies, mostly we won't give, sir.
Okay.
What is the cost?
Costs are these Victoza, that's liraglutide, it will cost around 6,000 per injection. But oral preparation, oral semaglutide tablet will cost around 315 rupees. So for 1 month therapy, we require almost about 10,000 rupees we need. Cost is little bit high, sir.
Sir, I vaguely remember that semaglutide, they have adopted some newer technology to deliver the peptides through oral route, and it will facilitate the better absorption. Actually, I forgot that exact how and all, but if we read, we will remember. We will be able to get that. They have collaborated with, actually no one nowadays, they have collaborated with one company internationally, and they have come up with that even oral insulin also they are trying now, sir. Similar technology only. So they are trying that, and they have successfully able to establish better bioavailability.
Thank you, sir. I now request Dr. Shirish Joshi to kindly hand over the certificate to our speaker, Dr. Manish Mohan. Thank you, sir.
The next speaker for the session is Dr. Madhusudan Singh, Resident, Department of Pharmacology, All India Institute of Medical Sciences, Raipur, whose talk is on newer beta-lactams, beta-lactam inhibitor, combination future implementation and rationality.
Good afternoon, everyone. Today I am here with an important group of drugs which are largely ignored, often confusing, and generally neglected by UGs and PGs, as it is meant to be there in the last paragraph or the last page of beta-lactam antimicrobial chapter. So there are a lot of things people around the world are doing to prevent antimicrobial resistance by either stewardship practices or developing newer drugs or drug groups. Similarly, bacteria are also not taking rest, developing novel mechanisms to neutralize all the efforts. So these MDR pathogens, they lead to increased use of cephalosporins and carbapenems, resulting in more and more resistance against these agents, and there is need for newer options for treatment and other carbapenem sparing options. So let's just have a quick review of beta-lactamase enzymes. These are mainly produced by both gram-positive as well as gram-negative organisms and are transferred to another bacteria quickly by conjugation and transduction. And they hydrolyze beta-lactam rings of these penicillins, cephalosporins, carbapenems, and monobactams. So practically we have two classifications, Ambler classification and Bush-Jacobi classification. Ambler has classified these on the basis of active sites as serine beta-lactamases and metallo-beta-lactamases. Serine beta-lactamases constitutes of class A, class B, and class C, and metallo-beta-lactamases is class B or MBLs. This ESBL, extended spectrum beta-lactamases, oxacillinases, A and D constitute of group 2 of Bush-Jacobi classification, and this class C, that is AMP-C or cephalosporinosis, is group 1 of Bush-Jacobi classification, and class B MBLs is group 3 of this classification. Clinically, the important point is the drugs like clavulanic acid, sulbactam, and tazobactam are only active against ESBLs, that is class A. And MBLs and oxacillinases that are produced by Enterobacter, Klebsiella, E. coli, gram-negative organisms are very difficult to treat because of MDR, because of multi-drug resistance. So coming to the BLIs, There are 6 BLI that are approved by US FDA, 3 older ones and 3 newer ones. Older ones like clavulanic acid, sulbactam and tazobactam, and newer ones like avibactam, vaborbactam and relabactam that is approved in 2020. So when we compare these older beta-lactamases, their mechanism of action, apart from inhibiting beta-lactamases, this sulbactam, it is having inherent antibacterial activity. That is, it is seen in certain bacteria because of binding to penicillin binding protein. Similarly, these clavulanic acid and tazobactam, they are also having some amount of this activity, but it is very limited. They inhibit the enzymes like, they inhibit ESBL type TEM and SHV1, which is produced by enterobacterials like these organisms. Sulbactam, it is also having some inherent antibacterial activity against Bacteroides fragilis and Acinetobacter. And similarly, clavulanic acid is having action against pneumococci. So, the main problem today is this CTX-M type of ESBL is the most common culprit behind the resistance which is seen in gram-negative organisms nowadays in whole world. And there is very little or no activity of these agents, clavulanic acid, sulbactam and tazobactam against this CTX-M. So, what is the solution? The only solution we can do is to establish labs for detection and molecular categorization of the type of ESBLs so that we can select BLBLIs on the basis of this tests. Now coming to approved and available combinations of clavulanic acid in India. These are amoxicillin, ticarcillin, cefadroxil, cefuroxime, cefpodoxime and cefixime, and only amoxicillin and ticarcillin is approved by US FDA. Amoxicillin clavulanic acid has been added in NLEM 2022 and 21 list also, and WHO 2021 list also. These are approved for these many indications like aspiration pneumonia, CAP, acute bacterial rhinosinusitis, UTI, and these infections. The important thing about this clavulanet is it has been added by PMDT 2021 guidelines by National Tuberculosis Elimination Program as a companion agent along with meropenem and imipenem cilastatin. The reason behind this is the Mycobacterium tuberculosis, it also produces beta-lactamases to acquire resistance against beta-lactam antibiotics. But contrary to this, this clavulanic acid plus meropenem or imipenem cilastatin is not approved in India by CDSCO as well as by the US FDA. Also, this clavulanet, it is not available as a single agent to be added to this meropenem and imipenem at present. Next, the combinations of sulbactam, this ampicillin and sulbactam is the only combination which is approved by the US FDA, and in India, various other combos are also available. The sulbactam has been combined with 3rd and 4th generation cephalosporins and as well as with the meropenem. Other combinations which has been studied for their activity along with sulbactam are with fosfomycin, cefoperazone, minocycline, aminoglycosides and colistin, and they have seen increased efficacy due to intrinsic activity, antibacterial activity of the sulbactam. Tazobactam in India, it has been approved along with the combinations with piperacillin, ceftazidime, ceftriaxone and cefepime, and only piperacillin tazobactam is also approved by US FDA, and it is also there in NLEM and WHO lists. It has been approved for various intra-abdominal, SST, gynecological, CAP, VAP and HAP infections. Other combinations approved by US FDA but not available in India is tazobactam plus ceftolozane. It is approved for complicated IAIs, complicated UTIs and hospital-acquired pneumonia and ventilator-associated pneumonia. But I want to stress on some issues here. The combinations of sulbactam and tazobactam, they are they are not approved in India, but they are available in Indian markets. These are sulbactam plus cefixime and sulbactam plus linezolid and tazobactam plus cefoperazone and tazobactam plus meropenem. You can buy them on any fine day through online websites like 1mg. Other thing is piperacillin alone versus this piperacillin tazobactam, they are equally effective against Enterobacteriaceae and Pseudomonas aeruginosa, as tazobactam is not effective against metallo-beta MBLs. But piperacillin almost always comes along with tazobactam, and it unnecessarily exposes the patient to tazobactam, and it increases the cost of therapy, and a risk of ADRs. Now coming to newer beta-lactamase inhibitors, when we see here, this avibactam and relebactam, they are active against classes A, C and D, and vaborbactam is active against class A and D. Vaborbactam is not active against class B and D, that is OXA and MBLs. Avibactam is not effective against MBLs and anaerobes, and relebactam is not effective against MBLs produced by Acinetobacter baumannii. None of the combinations are available in India, but US FDA has approved combinations like avibactam plus ceftazidime, vaborbactam plus meropenem, and relebactam plus imipenem cilastatin. Former two are also there in the WHO EML 2021 list. You can see again here, this avibactam and ceftazidime, it is not approved in India, but it is available in the Indian market. And all these three, they are active against Enterobacteriaceae and Pseudomonas aeruginosa. And vaborbactam plus meropenem, it is also having good action against Acinetobacter. Avibactam, it also comes in combination with ceftaroline and aztreonam. Now these approved combinations, this avibactam plus ceftazidime, it is a good valuable option, carbapenem sparing option, and they are approved for complicated IAIs along with metronidazole, UTIs and HAP and VAP. And this vaborbactam plus meropenem, it is approved for complicated UTI including pyelonephritis on the basis of Tango 1 trial, and they have seen that it is also good good in the conditions like complicated IAIs, HAP, VAP and BSI caused by carbapenem resistant Enterobacteriaceae on the basis of Tango 2 and Tango 3 trials. Non-approved, so this is last slide, non-approved beta-lactamase inhibitors like zidebactam, nacubactam and enmetazobactam, they are in various trial phases. This zidebactam plus cefepime, it is in class 3 phase against in cases of complicated UTI, and this nacubactam, it is having in vitro activity against oxacillinases and MBLs, and it is in phase 1 trial along with meropenem, cefepime and aztreonam. So to conclude, we need newer options against MDR organisms that are producing MBLs and oxacillinases, and it is urgent need to find carbapenem sparing options. The only new combination available in India is avibactam and ceftazidime, which is not approved by CDSCO, and WHO EML list, they have added vaborbactam plus meropenem and avibactam plus ceftazidime. So India can at least approve these drugs so that they can only be prescribed in MDR pathogens for better patient care. Thank you.
Session is open for the questions.
What is the cost of these ESBLs, newer extended spectrum that you have talked about?
So ESBLs?
Yeah, the newer beta-lactams along with clavulanic acid combinations. What is the cost of?
So cost of newer like avibactam and
drugs. Is there any price range?
Sir,
are they affordable, cost effective?
So they are very costly. 2,500 around one vial it is of 2,500 or 3,500. So they are costly, sir.
And you mentioned only one of them was actually effective against anaerobes. So I mean, generally when we talk of anaerobes, we talk about metronidazole.
So they have did the study along with metronidazole only. So they have approved along with metronidazole they are giving only.
So efficacy wise, which is better? I mean,
yes. Sir, it is not established. They should be given along with metronidazole. It has also only been given, sir.
Not in isolation.
Yes, sir.
If there are no more questions, we can proceed to the next session. Thank you very much.
Thank you, sir.
I now request Dr. Ratish to kindly hand over the certificate to our speaker, Dr. Madhusudan Singh. Thank you, sir.
The next speaker for the session is Dr. Shambhavi Kulkarni, Assistant Professor, Department of Pharmacology, SNMC and HSK Hospital, Bagalkot, whose talk is on neuroscience-based nomenclature of psychotropic drugs.
Warm greetings to all. I am Dr. Shambhavi Kulkarni from S. Nijalingappa Medical College, Bagalkot. I am going to speak about a new way of naming the psychotropic drugs, which is the neuroscience-based nomenclature. I'll be speaking about the current classification and about the new classification or the nomenclature and what is the need for this and what are the challenges at the level of implementation of this new nomenclature. So the current nomenclature of the psychotropic drugs does not follow one single mode of classification. That is, many of our antipsychotic drugs are being used for depression, many of the antidepressant drugs are being used for anxiety. So there is a kind of mismatch between the terminology that is given and the indication for which it is being used. So the current classification, that is anatomic therapeutic classification given by the WHO, was first started in 1976, which dates back to around 60 years from now, which classifies the drugs by these names, that is antipsychotics, anxiolytics, so on and so forth. So I'd like to take the example of the drug quetiapine, which is currently classified as an atypical antipsychotic under the ATC classification. So if we see the receptor occupancy, it is a D2 receptor antagonist, it is 5-HT2 receptor antagonist, and a norepinephrine reuptake inhibitor. So based on this type of action, it is used in different doses for different indications. So as per ATC, we could group it under antidepressant or as an antipsychotic or for mood disorders, etc. So this leads to a lot of confusions among new learners and among new prescribers as well. So the current ATC classification lacks a kind of orderliness. There is no information regarding the pharmacological aspects when we see the name of the group of the drug. One of the key authors of the NBN puts that ATC classification is confusing and confused. And there is a stigma associated with these drugs when a patient of depression is being prescribed antipsychotic drugs. He might always come back to the doctor and ask, is my diagnosis correct? And there are few problems with new drugs. Where to place the new drugs under the existing classification of the naming? So hence, there is a need for neuroscience-based nomenclature of these drugs. The NBN nomenclates the drugs on the basis of the pharmacology and the mode of action, the pharmacological domain and the mode of action. So it provides physicians clear alternatives when they are selecting a drug or they are altering the therapeutic regimen. It expands the vocabulary of these agents and helps improve the treatment and also adherence to the therapy. So this is neuroscience-based nomenclature, which is a new system of classifying the psychotropic drugs based on the pharmacological domain and its mode of action. It was developed to replace the current indication-based nomenclature. It is a publication and also there is a web-based application which is released, which helps in transitioning from the current classification to the new nomenclature. So currently we are in the second revised edition of NBN. The work of developing this new system started way back in 2008 and the current second revised edition has around 130 drugs. It is also being translated into various other languages and there is also a nomenclature system for children and adolescents, the drugs used in child and adolescents as well. So the task force was created with these 5 international agencies, which is led and majorly funded by the European College of Neuropsychopharmacology. It also includes American College, Asian College of Neuropsychopharmacology, College of International Neuropsychopharmacology, and our very own IUPHGR. So the principles are to move from indication-based nomenclature to a neuroscience-based nomenclature, to incorporate neuroscience insights and the recent findings, to replace 60-year-old nomenclature system and bring an updation, and to make this classification future-proof. So as and when a new drug comes, there should be a place to accommodate the new drug as well. And to provide an important teaching tool that reflects the depth and richness of the neuroscientific basis of these drugs. So the nomenclature considers these important aspects. So the pharmacological domain could be one of these 11 chemicals that act in the central nervous system, and the mode of action could be one of these 9 actions. So for example, we could take the example of acetylcholine, and we have important drugs like donepezil, galantamine, etc. So donepezil would be called enzyme inhibitor of acetylcholine esterase. So that is how we would be nomenclating these drugs. Along with the nomenclature, it gives information about 4 additional domains, that is the approved indication of these drugs, the efficacy and side effects, the generic and brand name, and the former terminology so that we can easily correlate with the existing names. So this is what the NBN3 mobile application would look like. We could download this NBN3 app from the on both Android and the Apple phones. So I just gave the search of drug like aripiprazole at an upper dose. So it gives all the required information for the clinicians and for the learners to get all the basic information about the drug related to efficacy, mode of action, adverse effects, etc. and even some of the other data which is clinically important for the physicians. So there have been certain challenges. There is occurrence of confusion whenever there is a new change or some change being implemented. And the task force does accept that this is not a completed work because there are new drugs being emerging every other day. So as and when the drugs emerge, those are going to get incorporated in the NBN, and this is getting revised twice yearly currently. The drug approvals by regulatory bodies and insurance reimbursements may face certain challenges until it is widely accepted. And to overcome these challenges, there is a need for welcoming of this from the scientific community, which will be even better if international organizations like WHO back this kind of changes. So implementation is being done in a stage-wise manner. Currently, the chief editors of 22 scientific journals have implemented this from 2017 July onwards, wherein the journals require the authors to mention the NBN nomenclature of the psychotropic drugs which are coming in the articles. So to make the publication searchable, the drugs mentioned by this nomenclature should be added to the keywords section. And the journals are encouraged to add an NBN subcategory to the keyword search, and the number of keywords allowed have also been increased to accommodate the NBN nomenclature. So to summarize, implementation of the NBN will help clinicians to improve the communication with the patients. It will increase the understanding of the subject for the learners, for the basic scientists. It will be useful for describing the drugs in literature and in academic discussions. And when we identify a drug by its pharmacological action, the rationale for its use also increases, and definitely the undergraduate and postgraduate students will be able to understand the pharmacological aspects of these drugs much better than with the current nomenclature, wherein we are putting different drugs of different mechanisms of action into one group called as atypical antipsychotics. Lithium being classified as an atypical antipsychotic is a great example why we need a change of this sort. Thank you for your patient hearing.
The session is open for discussion. Any question, answers? Nerurkar sir? In one of your slides you mentioned that we should move from disease-specific treatment to pharmacology-driven, but most of our treatment essentially rests on pathophysiology.
Yes, sir.
So it ultimately we go to the pathophysiology of the disease and then plan our treatment protocol accordingly. So would you like to?
Sir, I do agree that we need to treat based on the pathophysiology, but the nomenclature part only is what we are considering. The nomenclature should be giving an idea about what we are trying to treat. There is no harm in keeping the indication of the disease, the drug, but currently because of lot of confusions, to avoid that, maybe we could add some kind of basis for the drug therapy is what the task force asks for.
Will this nomenclature help for PTSD patients? Like which PTSD were mixed anxiety on one hand, depression on another hand. So will that nomenclature will be, that's also very common post-traumatic stress disorder. So any particular take on that?
I think completely eliminating the current system will be very difficult and may not be judicious also. Maybe a uniform mixture of both would help everybody.
You mentioned about software. All the drugs are mentioned in that?
Yes, sir. Around 130 drugs which are commonly being used by the psychiatrists all over the world has been included.
It's being regularly updated?
Yes,
software.
sir. Twice yearly it is being updated.
Software also?
Yes, sir. Towards software as well.
Right. Thank you.
Thank you.
Thank you, sir. I now request Dr. Shirish Joshi to kindly hand over the certificate to our speaker,
Is it
Dr.
something,
Shambhavi.
like we have already so many classifications of drugs, means nomenclatures of drugs, like proprietary, non-proprietary, ATC coding, so many things, and again this will be another one. And so this is for psychiatry, and gastro will start something another. So is it allowed?
If it is helping, maybe we could.
That's a matter of debate. But we have to get it validated and approved from authorities. Anyway, thank you so much.
Thank you, sir.
The next speaker for the session is Dr. Pramod Kumar, Assistant Professor, Department of Pharmacology, All India Institute of Medical Sciences, Patna, whose talk is on pharmacogenomics, a tool for precision medicine.
Good evening, one and all. The theme of NAPTICON 2022 is pharmacology expertise for changing times. So I wish to talk about pharmacogenomics, a tool for precision medicine. So I'm not going to give the full recipe of these objectives. I will briefly touch these topics with these outlines like definitions and history. Why does drug response vary? What are the types of genetic variations? What are the clinical applications of this pharmacogenomics, and what are the clinically available pharmacogenomic tests? What are the resources for pharmacogenomics information? And what are the advantages and barriers with the take-home message? So let us begin with these simple questions. Does one drug or dose fits all? Wonderful. So, no any drug or dose fits to all. We have to customize drug and doses for individual patients. Okay. So let us see a picture. This picture shows the differential drug response and nature of responders. When we give a drug, here the group of patients having the similar patients, what the clinician do, this is the current approach of treatment is, they make a same diagnosis, patients having the similar symptoms, and prescribe the almost same drugs to all the patients. But what happened, majority, about 60% of the patients respond. Rest of the patients show different type of response, like they have poor responders, some are toxic responders, and some are, few are, even they are non-responders because they have no receptors or target molecules on which the drug is going to work. Okay, so this is the differential drug response. So what is the solution? Okay, so we have to precise the medicine. We have individualized therapy. We have to personalize medicine. As we know, there are various factors which modify the drug action. Among these, one is the genetic factor, genetic variation. So we can customize this therapy in the form of precision medicine by study on the genetic variations. Okay, so then we can customize. Some needs standard doses, some needs lower doses, some needs alternative therapy. Okay, so that is why I have chosen this topic. So pharmacogenomics uses the use of genetic information to guide the choice of drug and dose for on an individual basis. So what is pharmacogenetics and genomics? So initially it was thought that the single gene responsible for variation in drug response, but now later on it has been seen that there are multiple genetic variations which is responsible for various drug response. So genetics refers to monogenetic variants affecting the drug response, whereas pharmacogenomics surveys the entire genome which is responsible for variation in the drug response, whereas precision medicine is the aims to customize the therapy. It is precision medicine is also known as personalized medicine or individualized medicine or individualized personalized therapy. So this is pharmacogenetics or genomics, abbreviated as PGX, and precision medicine. So if we look at the history status as far as back since 510 BC, when Pythagoras observed that ingestion of fava beans may lead to this deficiency of anemia due to hemolysis. It was, but at that time it was not known that it is due to the deficiency of G6PD in some individuals. This then the various landmarks shape the field of pharmacogenomics, like this Mendel's established the rule of inheritance. Then this Watson and Crick described the double helix DNA model. In 1959, this Frederick Vogel introduced the term pharmacogenetics. So why drug response vary? We all know non-genetic factors like age, sex, species, various pathological conditions affect the drug response. Along with this, the genetics also greatly modify the pharmacokinetics and pharmacodynamics, because all the drug transporters, ion channels, drug receptors, their couplers and effectors are controlled genetically. So on the basis of the enzymes, we can, the patients or individuals can be divided into 4 groups: poor metabolizers, intermediate metabolizers, normal metabolizers, and rapid or ultra rapid metabolizers. So what happens in poor metabolizers, there is almost no enzymes that is going to degrade the drug, so there will be toxic response. In intermediate metabolizers, that there is deficient enzymes, so again there will be they need the lower dosage of the drug, whereas in normal metabolizer we can give the standard dose. In rapid or ultra rapid metabolizers, we can there is rapid metabolism of the drug, so there will be therapeutic failure, chances of therapeutic failure. They need some higher doses. So these are the examples which affect the various drugs. Now, what are the clinically available pharmacogenomic tests? So these are the name of the drugs along with their variants, like Abacavir HLA-B*5701. Now what the genomic lab, they doing the test like panel, respiratory panel testing, cancer panel, cardiovascular panels. But we should know what are the important drugs and their specific tests. So no clinician can be expected to remember all the variations and gene responsible, so they can use this electronic data system for getting information. So we have this clinical pharmacogenetics implementation consortium, which gives evidence-based pharmacogenetic clinical practice dosing guidelines. Other resources are available. So what are the advantages? So ultimate goal is we have to maximize the drug efficacy or minimize and no harm to the patient. So we can, it can be achieved by this pharmacogenomics, but there are various barriers. First one is we have to identify the multiple gene variants, drug-specific gene variants. Screening of multiple drug-specific gene variants is a difficult task, and also millions of SNPs must be identified and analyzed to determine their involvement in the drug response. So this is the summary. If we use the pharmacogenomics, there will be low cost, overall cost will be reduced. It will be more beneficial. It will be safer and predictable. And the current, but the disadvantage of personalized medicine is it is difficult to screen which drug-specific genetic variants. It is a time-consuming, expensive, and this above one shows the current model of therapy where we don't use this pharmacogenomic testing, which is advantage is less expensive, do not need more tests. The treatment could be initiated rapidly, but there is a disadvantages that there may be different side effects. The treatment is beneficial or not, we are not sure, and what is the optimum dose, it is again questionable. So thank you so much for patience hearing. I will be happy to answer. And so this is the conclusion. Even after the test has great potential to optimize the drug therapy, and this topic also has a great potential for we can establish the pharmacogenomic lab in the Department of Pharmacology. We can do various research. We can collaborate between the patients, pharmaceutical industries as well as the patient. So that is why I have chosen this topic. Thank you so much.
Thank you, Dr. Pramod. I think it was a very interesting lecture, and I think pharmacogenomics is the future. And I just saw a couple of days one small cartoon where a patient is carrying a QR code to the doctor, and when the doctor scans that QR code, his entire genome actually comes. So maybe there will be time when we reach that. My question here is, what is the cost of this? Like for example, for warfarin, we have to go for VQOR-C1 and CYP2C9.
Yes, sir.
So what exactly are we looking at? What is the cost?
Yes, sir. Sir, basically the genomic lab, they are not doing the individual, they are doing some panel-like investigation. So 25 to 30,000, I have queried some in Delhi based some lab queried. 25 to 30,000 per panel is the cost of this testing, sir.
Any other questions? Pramod sir, what is the current status of pharmacogenomics in India?
We are, sir, in baby step. I am at INI like AIMS. I am also in connection with the other AIMS also. So still the last slide is still some simple spot test like G6PD we are doing. So that is why this topic has a huge potential. We can just do the prescription audit of the determinants of the genetic variations of commonly prescribed drugs, whether it is done or not. So that is why I think this is the last presentation, and I hope in the next NAPTICON this pharmacogenomics will be the first presentation. Okay.
Great. What is the cost of this G6PD testing?
Exact costing they are doing some panel wise, 25 to 6,000 for recipe panel, cardiovascular panel, anti-cancer panel. They are not doing sir, drug and a specific drug or this.
Okay. And do all AIMS colleges do genomic testing? In AIMS Patna, you are from?
No, sir. No any practice is done on the basis of simple spot tests are going on. So, I have queried my medicine colleagues, people working on the oncology department, they are not doing at all.
Thank you. So we conclude with today's session. Yeah.
I now request Dr. Ratish to kindly hand over the certificate to our speaker, Dr. Pramod Kumar.
We have now come to the end of the YPS session. I once again thank all the speakers for their excellent presentation and our honored chairpersons for their valuable time. I request Dr. Sushil Sharma, Professor and Head, AIMS Mangalagiri, to hand over.
Everyone, whatever you vividly imagine, ardently desire, sincerely believe, and enthusiastically act upon must inevitably come to pass, says Paul J. Mayer. Firstly, I'm just so grateful and extremely overwhelmed that pharmacologists from all over the country came together under the same roof to celebrate one fest, that is NAPTICON 2022. We celebrated togetherness, unity, adoration, harmony, and I feel quite ecstatic about this, and the presence of all of you has made this event even more extraordinary. Welcome you all gathered here. Special salutation to our beloved Dr. Padmaja Udaikumar, the President of NPT and the Organizing Secretary of NAPTICON 2022. Ma'am, it gives me immense pleasure to say that you have always given us a reason to keep adoring you, and the success of NAPTICON 22 is just another golden feather in your cap. Thank you for bringing us together and making sure this conference was a magnum opus. Hearty welcome to ma'am. A warm welcome to all the dignitaries present here, and I would also welcome our sponsors, speakers, chairpersons, judges who are present in the valedictory gathering. And any event cannot be successful without people who devote their resources and time to ensure that everything is flawless. I take immense pleasure to welcome the organizing team members to this valedictory function. Welcome you all.
Thank you, Skanda. Feedback is an integral part of a continuous improvement. With this, I now request the August gathering to kindly give their honest feedback to us during this session. We have Dr. Sushil Sharma, who stayed with us for a very long time till this very end. So I think we'll hand it over to you, sir, and even to Dr. D.D. Gupta.
Hello, good afternoon to everybody. First, I congratulate Madam Padmaja Udaikumar and her efficient team for grand success of this conference. And I think everything was excellent, food was excellent, and scientific contents were excellent. And this whole was due to your endless efforts throughout this, I think, period you have spent in the last 1 or 2 months.
Not 2 months, sir, but 6 months more. January we started our efforts. So, yeah, sleepless 1 month, yeah.
Thank you very much. And I think I second Dr. Gupta's words. And, I'm attending a conference after so many years. And I don't know, this is one association which feels like home, basically. We have had other associations in the past, but somehow the kind of feeling that we get with NPT, I think it's totally different. And I'm very happy that I could manage to come here and meet all of you. And it was a wonderful conference, madam. We have made some good friends. We have heard so many good talks. We learned from each other, and we hope to take it forward. And I think your team of, someone told me that if there are 7 ladies in a team, it's going to be really difficult. Like my sister is in one of the medical colleges, and it's a full ladies team. And every day she says it's a torture. But this is a total, different thing which I am seeing here, and I think it's totally because of the leadership qualities of madam. I think she is the one who is able to take the entire team, forward in a very cohesive manner, and the result has, shown, and it's visible to all of us. So thank you very much, and I'm really thankful to be here. Thank you.
Thank you, sir.
Thank you so much, sir. Our efforts are one thing, but it's the feedback which gives us the motivation to do better.
I've already said it is a wonderful conferences. I have attended till now. Thank you so much, ma'am.
Thank you, sir, for your feedback.
I must congratulate the women power of this Father Medical College. It's a great effort, unmatched. I am also attending a conference almost after 6 to 8 years, and it is an academic feast. All the scientific sessions were excellent topics which were chosen for young pharmacology speech, which was a new thing, and it will encourage the way Dr. Kamal and Dr. Padmaja Udaikumar handle the these events and is admirable. and it was an academic feast, and it was a great stress buster. The yesterday's event, cultural event, was very, really nice. I enjoyed, and I'm very happy that NAPTICON has started in a big manner, and it will go on. The kind of people who are there in the organizing committee, and it's a wonderful feeling. Thank you all, and wish you all the best, and we look forward for the next conference.
Thank you, sir.
I would like to say a few words. I have never seen, I have attended many conferences, workshops, but never seen a very cool, calm, honest and humble person, okay, like Dr. Padmaja Uday Kumar. So hats off to you, madam. And from all of us, a big hip hip hooray to madam, 3 times. Hip hip.
Hooray!
Hip hip.
Hooray!
Hip hip.
Hooray!
Thank you very much.
So, first of all, I would like to congratulate the organizers. It is really a great opportunity to meet all our colleagues. And I would like to say that, very nice to see that first conference happened in Karnataka. And congratulations to madam and the team, entire team. They worked very hard. I have been talking to madam, and she is very, encouraging to everyone, and I would say whatever it is organized, it's entirely, perfectly. I would say, all of you please clap. It's one of the perfectly organized program. We could not see any flaws. Such a big, nearly around 500 people attended here, and everything is properly planned. Thank you, madam. It's really nice.
Thank you so much, sir, for your feedback. It means a lot to all of us. Yes,
Good evening, everybody. Myself, Dr. Shashidhar from Bagalkot. First of all, thank you to the organizers, madam and sir, and their team for such a marvelous, wonderful conference. I had when I was PG long back, that time when will be our only MD pharmacology conference, pharmacologist, and that wish has come true. Thank you, madam. And I think this is just a beginning. We wish we will grow together. Hope for the future. All the best, ma'am.
Yes. Thank you so much. Thank you for your feedback. Mia Hamm, a female footballer, the winner of 2 Olympic and 2-time FIFA Women's World Cup winner, said, "I am a member of a team. I rely on my team. I defer to it and I sacrifice for it. Because it is this team and not the individual that is the ultimate champion. And it is this teamwork that does make teams work and dreams work." So with these words, I would like to call upon Dr. Padmaja Uday Kumar and kindly request her to give a memento of appreciation to the organizing members. Dr. Reena Parveen. Can we have a big round of applause? Dr. Chandini. Dr. Bhavya. Dr. Sheetal. Dr. Sneha. and Dr. Shreya. I request Dr. Bhavya to take Dr. Shreya's memento of appreciation on behalf of her. She is unable to join us at the moment. Thank you. Miss Lakshmi, a big round of applause to the person who taught us dance and helped us move all our left feet. Mr. Praveen. Renita. And Chitra. Thank you, ma'am. I also request ma'am to kindly present a memento of appreciation to our sponsors, Mr. Jwalant and Mr. Bharat from Delta Healthcare. A round of applause to them for supporting us in our conference. Mr. Nitin Gaikwad from Medimation Limited. Dr. G. Shankar Family Trust, Udupi. Thank you so much, ma'am. With this, I now request the President and the Organizing Secretary of National Association of Pharmacology and Therapeutics to kindly give us the closing remarks and also propose the vote of thanks.
We have been speaking enough. There is nothing much to give as closing remarks. It was a wonderful experience for me to organize this conference. As I told yesterday, like, you should also have the experience of organizing a conference in your lifetime. But I have to place on record the excellent support given to me by my team. I have no words to say, to express how they have been working. All of them have been behind me, with me, supporting me as so many pillars of support. We really worked as a team, and it really showed how teamwork could be of help. And I thank all of you for having been here, and particularly for staying so long for the valedictory function. And day 2, in fact, someone was telling me, I have never seen any conference, because those who arrange conferences, they know there will be 2 days conference, but on day 2, I have not seen more than 10 people, but in your conference, what is this afternoon for lunch, there was 400 people eating. So they were really surprised. So this is the first conference that we have so many people coming on the second day also, and so many of them stayed till very late. Only now, just a few have left and a few are remaining. So it was a great experience, and I'm really grateful to all of you for being with us and supporting and coming here and giving us a helping hand and supporting. So it's, as sir said, it is like we have become like a family. We made many friends, and we are looking forward to meet all of you in the next NAPTICON. Thank you all, a big thank you. Long live NPT, long live pharmacology, long live NAPTICONs to come.
Thank you so much, ma'am.